So that is what it is. And we found that if we really inhibit this pathway, we could really target this GBM. So that was the study which was published. The second story, as I told you, I'll within 15 minutes, I'll tell you 3 stories. So one story I finished. The second one is basically on the perfusion and the cancer stem cells. So now we have a cool technique called as PFDLC. So can we try to understand in vivo there are these so-called cancer stem cells or the quiescent cells or the drug tolerant cells or the persister cells, which are the cells which evade therapy. Again, more and more studies have come up that this is again not a genetically hardwired, but it is also plastic in nature. So they are changing, and our hypothesis, again coming from a vascular biology perspective, is that the environment is influencing them, and based on that they are responding to it. So the question is the stemness hardwired or adaptive? So what did we do is basically we used a marker independent approach. So this is basically called as a dye labeling approach, or exploiting its standard function of quiescence. Stem cells are what? They are quiescent in nature because they don't want to grow more unnecessarily. They want to conserve everything, and because of this, if you just stain them with a particular dye, if there is an uniform division, the dye will be distributed uniformly and it will just get diluted. But if you have some cells which are biased, they will retain the dye because they are not dividing so much. They will still have their dye. The rest of them will divide and lose these. So we use this approach, and we saw that indeed there are some cells, in the spheroid assay, which will retain this quiescent property and they can be identified. So we went on in vivo, we did the same thing, and we merged this with the PFDLC technique that we had, and we found out that there are a lot of these white cells are the ones which are quiescent cells or the cancer stem cells. So now, because we have this approach of identifying if it is in the perivascular region or not, we can use this method to again isolate these cells which are functionally quiescent, so to say, and characterize them to do all the omics that we want. So I want to again show a cool video, which will again take me a second. So the white ones are the cells, and if you see, the blood vessels are the red and the green is the tumor. It reminds me of cancer stem cells which are holding on to the blood vessels like an epiphyte. You know what is an epiphyte? Basically a plant which depends on the tree for the nutrients, right? So that is what is happening. So we were interested, again being vascular biologist, sorry for my bias, but we were interested what exactly is the influence that it is having. So we went on to do some transcriptomic studies. There are series of markers or series of genes which we found out, and we did some gene set enrichment analysis. Of course, we had mTOX signaling, the G2M checkpoint. So basically the quiescence is there, and the ABC transporters. Why ABC transporters are important? Again, right, these are the ones which basically fluxes the drug out. So they are there. These are all enriched in these cancer stem cell fraction or the label retaining fraction. And we took one of these interesting molecule from here, and what we did is basically we found out that CD271 is one of the markers which is overexpressed specifically in these cells, and when we knocked down those and we implanted these with the into the non-skid mice, we could see that the tumor initiation ability was reduced significantly. So blood vessels are important for tumor heterogeneity. Blood vessels are important for cancer stem cellness. And also what we quickly did is, if that is indeed the case, what is the role that endothelial cells have? So we performed a cool co-culture technique, which again I'm not going to go into the details, but we found out that there are some signals which are being told by the endothelial cells to the cancer cells, as well as there is a reciprocal talk, and we found out there are some cancer stemness related genes here which we are validating. Third question, so I'm done with the 2 stories. The last story, what is happening? Why is there this driver of intratumor heterogeneity? Definitely it is not a genetic factor, because these were all clonal population and they're just spatially different, enjoying different perfusion. So we focused on the epigenetic changes, the DNA methylation mainly. When we looked at the H3K27 trimethylation, which is one of the classical repression-based methylation. You can see that again, this is the perfusion-rich region, right? And these magenta ones cells which are positive for this H3K27 trimethylation are all complementary to this. So basically, they are enriched in the hypoxic region. So there is some kind of a methylation which is happening specifically in the hypoxic region, which is changing some of the things. So what we did is basically, again, the advantage of this PFDLC method, we could do multi-omics. So we did a methylome array of around 850K, all the pan genome CPGs. We checked it out, and you see how beautifully the principal component sits. So these are again same population. These are perivascular, intermediate, and the hypoxic, and they're all segregated. So the methylation is very different, and that is why everything is happening. We could get the heat map in which there are some which are enriched here, there are some which are enriched in the other population. And the coolest is the highest bivariance is between the perivascular and the hypoxic, right? These are the ones which are the farthest. So again, it is not because I'm just a vascular biologist, but it is indeed the case that it is because of the vasculature or the perfusion that it is happening. And we did a volcano plot, as well as we were interested specifically in methylation in the promoter regions. And when we looked at the promoter regions, we see that this is the red ones. The hypermethylation is mainly in this region, So again, why is this important? These are the ones which decides whether some genes has to be turned off or on. The last slide, I promise. So we did an integration of the methylome and the transcriptome that we have, and we could get some interesting candidates. And these are all unpublished data which we are still working on. And we could find that there are some hypomethylated upregulated genes. And there is one, again, I'm coming back to the ABC transporters, ABCA1, which is in the hypoxic niche, which is upregulated. And we are interested in investigating how is this really playing a role in all that I explained. So in summary, I showed you that we have devised a methodology to identify, cancer cells based on the perfusion, and differential exposure of vascular inputs generates multiple metabolic niches and results in heterogeneity. mTOR is the key driver there. This is the first spatial methylome which has been done at in an in vivo level, and these methylation changes result in perfusion-induced plasticity. So I would like to thank my lab members. I'm just a messenger. All the work was done as part of my postdoc and being carried into the IIT lab which I've set up. And this is primarily the work of, the last part of the work is from my PhD student Neil. And thanks to my collaborators and funding agencies. So I'm happy to take any questions if they are there. And thank you again for coming back at 9:30 and listening to me. Thank you. Thank you. Mm-hmm. Mm-hmm.
Second question is, you also did this methylation study. Again, regions identified by the perfusion study, and you showed different methylation. Previously, it is known whole tumor study identified based on methylation, GSIM plus and minus having two additional groups. So any of those whole tumor methylation study based 3 different groups is matching with your division based on your study.
Okay, interesting. Yes, I'll answer the second question first because it's easier to answer. We haven't done this, but yes, good point. We haven't done the whole tumor and then looked at it, but I understand that yes, there is a differential methylation which happens. But what we are interested in is the intratumoral heterogeneity, and that is the reason why I did not do it. But it would be an interesting this one to look at our signature and see how it is mapping with this one. What we have done retrospectively is basically there are subtypes of tumors, and we have tried to take that and see where our signature is matching, and there is some interesting studies which are ongoing. And to answer your first question, yes, we did that. It is very interesting. What we see is basically these cancer stem cells I think I would like to classify them as two. They are mainly fall into the perivascular, also the hypoxic. So I think there are two classes there, but again, the time and this one which we had, we could only characterize the perivascular. Now the whole focus of mine is to basically look at the hypoxic and see what is happening to those cancer stem cells, because also the whole theory popularly is quiescence, hypoxia, all these are related and intertwined. So yeah, thank you. Please.
How long is stably fluorescing?
Yeah, so all these assays, we try to do it as quickly as possible, but this will sustain even for few hours. But again, because it is a perfusion dye, the more you leave it, the more it will spread, and then the chances of the mess. So what we have done is basically all the assays that are there, 5 or 10 minutes, and then post fax is basically hardly 30 minutes to 1 hour. Yeah.
Oh, sorry, which dye, sir?
It's the Hawks dye. Yeah.
And why I asked the question, because there are lots of people, even I'm also trying for certain bioimaging markers, which we can use it for stable fluorescence. Because mostly what happens when you fluorize them and keeps them under any of the light, and gradually it quench. So again, when we look back into the same location where we are primarily looked into, it will not fluorize again. That is why I was asking for this question.
Understood. We can take this off table. There are other types which are correct.
Okay, sir, sure.
One more question?
Yeah, one.
Sure.
Nice talk, sir.
Thank you.
So one question is, you told that differential methylation you observed in the hypoxic region. So did you also check the methylation? Because cancer stem cells, the glioma stem cells mostly reside in hypoxic region, as you are telling. Did you also check in the cancer stem cells about the methylation pattern?
Yes. So basically, no. The reason being, cancer stem cell population is a challenge to do the methylation, the amount of cells you will need and all that, because it's post sorting and all. So just for the technical reasons, we haven't done it. But it's a good point. What retrospectively, what I'm thinking is basically we could look at the omics and check if there are any signatures for the methylation related changes that would have happened. We haven't done it, but this is an interesting point actually. Yes, thank you. There is, I think, one more question.
I too am interested in a little about the dye, mostly because one of the ways we enrich CSCs in the lab is to enrich for dye negative cells because of the ABC transporters. So there must be some background work you did to, and if you could explain that to us about how yeast is able to stay within the CSCs.
Yeah. So two things here. Yes, for the site population studies, HOX3 is the dye that is used, but what we have done is basically we have oversaturated the dye here, one, and the time limit that we have, it cannot actually actively pump out the whole dye at that point of time. So even if it is doing, it is already an underrepresentation of what we have already done. So it is that way. So, but we have done some characteristic features. We are still not missing the point here. Yeah, thanks.
Thanks so much. Felicitate everyone at the end. All right. And so it's my pleasure to introduce the next speaker, which is Dr. Puneet Prasad from the Institute of Life Sciences in India. He's going to talk to us today about insights into epigenetic mechanisms in AML. Dr. Prasad.
So by the time this is setting up, yeah. Thank you, organizers, for having me here, and the session chairs, and especially the volunteers who have been working tirelessly to make our stay even better. Okay, so without any delay, what I'm going to do is to today discuss epigenetic mechanisms about leukemia that the lab has been working on for now 6-7 years. And the way I have designed my talk is to basically go through, briefly introduce, as I see, there's a lot of students over here, uh, and the lab is working a lot of big data. So I'll try to discuss about that first, and then show that how this data can, actually help you to, streamline and identify the factors which can be taken to the lab and then work further. So this is the lab uh, with so many students now, expanded a bit. We have good collaborations with the clinical partners, which is helping, and the financial assistance, as you can see, from different funding agencies, and big thanks to them. So, hematopoiesis is a very complicated procedure, and it starts from hematopoietic stem cells, and then it generates multiple progenitors, which then goes into lymphoid or the myeloid lineages, thereby releasing or doing so many different types of cells and releasing different factors, which is then helping in different kind of functions. Now, any genetic aberrations, primary, secondary, tertiary mutations, which actually leads to this aberration in the renewal property, and then it results in excessive proliferation, loss of maturation, and which results in leukemic phenotype. Now, this is primarily done by a bunch of players, which is epigenetic factors as well as transcription factors. And you see, there's like so many different mechanisms that is involved as to how this chromatin is regulated in order to define a function. And this any misregulations can lead to different functions. And this can be basically classified as writers, readers, and erasers. And the lab is primarily focused on epigenetic factors, and that too a bunch of chromatin remodelers. So how the lab work is that we use a lot of big data to basically churn the different factors that is involved in differential gene expression, and then understand the epigenetic mechanism through DNA methylation, chromatin accessibility, histone modifications, and so on. And once from there, whatever factors we get, we go and we take it to the lab and do in vitro, ex vivo models and screen them and find out their functions. So this is one of the analysis that we have started. It's unpublished, but it is almost finished, ready to be submitted. So we started with TCGA data. We took DNA methylation data. We took gene expression data. We classified the patients into cytogenetic risk based on poor prognosis, good prognosis, or favorable and intermediate, and the FAB classification, which is primarily based on different levels of maturations of the cells. Then there is series of different ways we have analyzed by looking at the standard deviation, looking at multifactorial analysis, MOFA analysis, then using weighted gene expression analysis, network analysis, and then we get several outcomes. So there's a lot of data behind, but I'm going to give you the snapshot of the study or the key figures that we have. So when we did this, you see this is the survival curve, and what you see here is that the good prognosis survival is good. We know, everybody knows, but we also see that M3 classified FAB, French, American, British classification, M3 also has a very good survival rate. So we asked a question if there is any relation in terms of gene expression among them. So, so what you see here, this is the heat map, and what you see is this green. Just focus on the green part, where you can see that the FAB M3 as well as the good prognosis is clustering together. What it suggests? It suggests basically that there is this bunch of gene signatures, differential expressed up or down, which defines their prognosis factor. However, the poor and the intermediates are clustering in a different way. And in order to understand that, what we did, we also took the literature curated genes and the genes that we got from our own analysis. And then we just did a Venn diagram to figure out what are the common and the unique genes. And then we went ahead with the common genes as well as the unique genes. Literature curated, we ignored because they are already reported, so we're not interested in them. And out of that, you can see that both common genes and unique genes are showing similar clustering pattern that suggests that these genes are somehow involved in prognosis factor or the classification factor, right? So we took these genes, we wanted to validate. ECG is a small data set. We went ahead and validated in BTML data set, and with multiple regressions and an analysis, what you can figure out, see that both common gene sets and unique gene sets are showing that the genes that are upregulated have poor survival compared to the one that is low expressed. So this again suggests identifies that the genes that we are looking and we are trying to focus do have some important aspects in terms of survival outcome. So after this, because the lab was interested in epigenetic factor, we just segregated all the epigenetic factors that we have churned from there, and we did a simple pathway analysis. And what you see here is primarily that the gene expression regulation is the key aspect that is getting affected. So now that this is done from a lot of different patients, this in 2017, Ravi Majeti's lab and Chang's lab published a very good multi-omics data set in Nature Genetics, where they have used very highly purified cell populations in order to study the epigenetic mechanisms. So what I did, we segregated epigenetic factors, transcription factors, and a combination of both. I wanted to see if we just take these factors, if the expression itself will be able to cluster them in some kind of pattern. So the good thing is, what you can see that the patient HSC, we can see BLAST, LSCs over here, they are all getting very nicely clustered. Transcription factors, we took more as a positive control because they are known, they have a DNA specificity and so on. So they are also clustering. But the good thing is that when you combine this epigenetic factor and transcription factor, the clustering becomes even stronger and better. Where the BLAST, as you all know, is highly heterogeneous. Even the blast cells are getting very nicely clustered together, despite of different clonal heterogeneity and so on. Again, giving us an indication that these epigenetic mechanisms are indeed an important player. And this is another representation, PCA, where you can see that leukemic stem cells are clustering nicely. Blast is also clustering, and patient HSC and normal HSC are clustering in a different way. Okay, so we are not done yet here. So we took all this data, we said, okay, all these are biased analysis. Let's do an unbiased analysis. We took all these genes and then did gene-gene correlation assays. And then we identified modules in that. And we wanted to see if those modules gives us any signature on that. And this is modules. We have segregated all the epigenetic factors. And what you can see, we have got 3 different clusters, and cluster 2 is the most important, where you can see the median expression patterns of different cell types, leukemic stem cells and others, are actually varying. Now we segregated, we looked, they are highly significant. HSCs, the epigenetic factors, and the transcription factors that are upregulated, and the one that is downregulated. Now we are looking into those. One of it I will show you how it looks. Before that, for the everyone, chromatin remodelers, as we heard yesterday also from Sagar, they are involved in altering chromatin structure and in a multiple mechanisms as shown over here. I'll not go in detail, but the overall is either expression or repression. So what is interesting is we thought that let's see the genetic mutation or the frequency of mutations. And what you see here, there's this color indicates deeper the color, more is the frequency of mutations. You can see this mutation frequency of these chromatin remodelers are very high in all the solid tumors. You will not find leukemia over here. So we segregated leukemia and we saw this. Now, because the lab was focusing on SpiceLeaf, I'm just showing SpiceLeaf, but we have for other data as well. The range is from 0 to 0.2%, which means these factors are not mutated. Okay. So during that time, we searched literature and we found, yes, SpiceLeaf is heavily involved in multiple and its subunits are also involved. So let's see if we get anything unique in this. So this is 3 different data sets, and in all 3 different data sets, we got this marker 1, which is an auxiliary subunit of Swiss-Smith factor. And what we saw that they are highly expressed in progenitors and stem, but not in the mature cells. They said, okay, now this is good. Let's see if it has any functions or not. So during that time, there are 3 isoforms marked 1, 2, and 3, also called BAP60A, B, and C. And only the 2 back-to-back Nature Genetics paper, which shows that SMARCA2 is involved in neutrophil development. There's no other. They are all involved in multiple metabolic processes, as shown here. So we went ahead and took BEAT-ML data and TCGA data, and we segregated them in the patients who have high expression of SMARCA1 from the patients who have low expression of SMARCA1. Okay, and what we found here, you can see the patients with high expression of SMARCA1, they are having high expression of SMARCA1 has more association with the stem cell factors or leukemic stem cell factors, and the less expression is related to differentiation factor. And the similar thing we saw in BEAT-ML data also. The PCA shows very nice clustering, a very segregated cluster, again showing that the gene, its regulation is quite unique. So we went ahead and did some knockdown studies in cell lines, and what we saw that when you knock down the cells, just by the knocking down SMARCA1, you will end up in having some level of self-differentiation into myeloid lineage. And if you induce the cells with vitamin D, say for example, which differentiate into monocyte macrophage, you can see compared to control, all the cells are shifted and they are differentiated. We have done multiple studies, different cell lines, all this shows the same result. Did RNA-seq again, and what we got, multiple upregulation of differentiation genes, cytokines, chemokines, and so on. And the cell radar, you can see, is showing very nice that all the upregulated genes are associated with the monocytic lineage. So the final nail in the coffin, basically we did when we took all the patient samples and we just did the knockdown of this SMARCA1. And you can see here that just the knockdown without any induction, you get a significant differentiation of the cells. The cells are automatically differentiated, and you can see here that the morphology of the cells are becoming more monocyte macrophage-like, again suggesting that this factor was actually involved in maintaining the stemness of the cell, and loss of this somehow doing. So this we kind of published, and this is a snapshot of so much of data. Basically, what we found that when you lose SMARCA1, you land up in upregulating the differentiation genes and the immune genes by having higher H3K4-Me3 activation mark and concomitantly reducing the 27 mark, which is a repression mark. Now we are actually now focusing and looking at different cofactors, co-repressors, which could be identifying together. We have some lead, but yes. So this is basically the mechanism what we are seeing. In the normal myeloid differentiation, you have a Swiss-Smith complex which associates with SMARCA1, and when there is a stimulation for differentiation, both SMARCA1 and there is another factor ACTL6A gets removed from the complex, and SMARCA2 or SMARCA3 can get associated for neutrophil or granulocyte or monocyte differentiation. Now in leukemia, what happens, these are highly overexpressed, not mutated, but highly overexpressed and very strongly associated with this, such that this differentiation pattern is kind of blocked and so on. So going back to the big data, we thought that let's see if we have some, what is its phenotype in leukemic stem cells. So you can see here in this panel that leukemic stem cells, the expression of SMARCA1 is very high, and it goes down in leukemic LSC negative, basically CD34 positive, CD38 positive and CD34 positive. So we knock down in those AML blast cells, and what you can see here in the figure, that when you lose SMARCA1, you lose LSC signatures, you'll land up in increasing progenitor signature, and differentiated blast is also enhanced. So finally, The association of SMARCA1, we also looked at with the data that we have, that in initial diagnosis, when you see the expression is high, when the patients are treated with drugs, the expression goes down. And when there is a relapse or there is a residual disease, it comes up. So again, suggesting that, this SMARCA1 may have a very strong prognostic effect, which we are now looking into. So I hope I am in time. Try to simplify my talk. We can discuss. This is my lab, and thanks to everyone. And there's two posters. One talking about the Swice-Nif auxiliary subunit ACTL6A, which I did not discuss on purpose, so that you can go to and talk to Smruti. And Shyam Smruti is also here. She will be discussing more in the different integration of omics method, like chromatin accessibility and gene expression, to understand leukemic stem cell signature in a better way, which we can take it further. With this, thank you very much for patiently.
Thank you, Dr. Prasad. It's very elaborated talk, and it's open for the discussion and the question from the audience.
There are inhibitors for SMARCA1, right? What did you find?
BRG1 inhibitors are there.
Okay.
But SMARCA1 inhibitor is not there.
It is not there. Okay.
No. The interesting thing is Swice-Nif complex, as it's known, that as a tumor suppressor in solid tumors, but in leukemia, it is overexpressed and it promotes leukemogenesis.
Okay.
It's the PRM that is involved in more differentiating stages.
Did you see other chromatin remodeling groups being regulated, like ISY and other groups?
Yes. So there is CHDs and ISYs are also there. Imitation switch 1 especially. So those are also in pipeline that we are actually looking into. So there's like now we have good number of epigenetic factors to screen. So from this whole thing, we have now 49 factors in our hand, where there is no study in leukemia or AML or whatsoever. And if I goes into those genes and look specifically for this survival curve or their differential, they are all dysregulated in leukemia. So I know that those will be a good pick for us to go ahead for screening that we have established in the lab.
The continuation of these questions, actually, Smart CD, whatever you were showing, you are checking the expression based on the CD4, CD34 positive populations, right? So any normal conventional therapy for the AML, what is the status of this Smart CD upon that idea?
Right. So so I have already shown a lot of patient data from both mine as well as published or Beat ML or TCGA. But apart from that, what we are also now doing, we are collecting the patient samples, and we are in Indian patient cohort. We are seeing how SMARCA1 expression is segregated. Okay. So I must clarify here that 100% of the patients are not showing SMARCA1 positive. It is a subset which shows. So one can say that if the SMARCA1 is high, they will go into this kind of prognostic factor. So it is not a universal factor, but yes, there is a significant number of AML patients with high SMARCA1.
Yes, thank you. Any more questions? Only one we can have. Okay. If there is no question, we can go for the next session. Thank you, Dr. Prasad. And we are inviting the Dr. Shilpi Datta. Now, currently, she is a professor at School of Life Science, Jawaharlal Nehru University, and she did her PhD from University of the Zurich. Personally, I am also from JNU Life Science, so I know you before. So, her research area is the advancement of the molecular basis of therapy resistance in cancer. So definitely you learn a lot from her. She was awarded Janki Amol National Women Bioscientist Award in 2019. So Dr. Shilpi, please.
Thank you, and good morning, everybody. I was supposed to speak yesterday in another session, but I solved with Amit, so here I am. The work that I'm going to present is mostly done at F3. So I was working at F3 before, Hence, last year only in March, my lab has moved to JNU SLS. So that's why I have both the logos here. So today I'll talk about one of the stories that we actually completed and published recently, where we had looked at the senescence in the cancer cells, and we have shown how you can exploit this senescence for therapeutics. So the two major scientific areas of work in the lab is glioma and leukemia. Today I'll talk about, like I said, the story of senescence in glioblastoma. So GBM, I don't have to give introduction. The introduction was already there. So, but just briefly, so that I am still in the flow of what I had to say. It's great for brain tumor. It's very, very challenging, and the recurrence is due to mainly the resistance that we see in the glioblastoma, despite having surgery and chemo and radiotherapy in the cancer. So, like I said, when the patient comes with GBM, you can see that after resection, post treatments, there is no tumor seen. However, inevitably, all the cases, the tumor comes back with recurrence, and what we were interested in these mechanisms. And as you would also appreciate from this slide, is that it is very difficult to understand anything from this scenario from the patient samples, because there are no samples post treatment. So then to fill in this lacunae and to try to still understand how this resistance could happen, we have developed models, the cellular models and the mouse model in the lab. So Ekjot, my first PhD student in the lab, she took up this challenge, and what she did is that she wanted to make an ex vivo cellular model so that she could study, we could study the mechanisms. So she took GBM samples, which are naive, so not treated, and then she would make primary cultures from these cells, and these cells would then be treated with radiation and chemotherapy. And then we waited and see what happened to these cells. And like it's an old story now. Now we all know that when we give radiation and chemotherapy, more than 90% of the cells die, but we did find some cells that were surviving. And interestingly, when we followed these cells, it was just serendipity. We thought that because these are, I'll show you that these are multinucleated giant cells, probably would die of apoptosis eventually or mitotic catastrophe. They did not, and they did come back with the relapse, which was a highly aggressive population of cells. So, like I said, when we looked at these cells, the residual cells that we call, they had a very strange phenotype, which is like multinucleated and giant cell phenotype, and they also had a lot of DNA damage in these cells. We did identify a mechanism by which these MNBCs are formed, which was the cell cell fusion, which is also a very rare and very interesting phenomenon that we are studying in the lab right now. Then another very interesting phenomenon and was the presence of these senescent cells. So all these MNBCs, when we looked at them carefully, they were all senescent. They were not dividing, so we just wanted to see if they are senescent, and indeed they were senescent. But eventually, like I said, when we looked at the division, we did find that these cells start to divide, and it was strange. And I'll tell you like how these cells are able to probably divide, because senescence is supposed to be the end of the replication per cell. So the first thing before getting into any mechanistics was to see if any of these phenotypes also hold true in vivo, because if they don't, then the whole exercise of looking at the mechanisms and looking at the inhibitors would go waste. So we wanted to first see if this would hold true in vivo as well. So Jyoti, another PhD student in the lab, she generated a preclinical orthotopic mouse model where you would label the GBM cells, and now she's also done afterwards the primary cells, which was a little tricky because these supposed to be primary. But then you label these cells and then you put them in the brain of the mice. And once the tumor start to grow and it reaches a certain stage, then you give radiation and chemotherapy to the mice, fractionated doses, like you would do to a patient, and then you monitor and see what happens to the tumor. And because this is mice and these cells are labeled, we also label them with GFP. Then we have a handle that, wherever at what stage we want to study these cells, we can sacrifice mice and take the cells out and then study these cells. We've also generated PDOX model directly from the patients. This was a little tricky because we did not label these cells, as we wanted them to not see the culture at all and just be in the state that they were taken out of the patients. So it was a little tricky because we didn't know when to sacrifice the mouse to look at the tumor. But now we have kind of standardized and we have an idea when to look at these tumors. And this to just show that, once we get the tumors and we take them out, all these cells are labeled here, and these cells then can be used for any kind of downstream analysis. So when in the in vivo model, when we looked at these mice, we did see like what we would see in a patient, that the mouse shows there's no there is a tumor, and then you give radiation and you see that the tumor is gone. Like I showed you the image before from the patient, and you wait for a while and you see that the tumor starts to come back. So this is the stage which is so-called the residual stage, and we were interested in this stage. We wanted to see what is happening to this stage, and because after this the tumor is coming back. So this first shows that in vivo as well, this what do you call, say, the residual disease stage is there in the mice, and these cells, because the tumor that comes back has all the labeled cells, we know that the tumor is coming back from the original tumor. So there must be some cells that were left that they kindly found in the in vitro system that are now giving rise to the recurrent tumor. We looked at these cells, and indeed these cells were also MNGCs. We found a lot of MNGCs, and they were non-mitotic, non-apoptotic. There was no necrosis, and very interestingly, they also had a senescent phenotype. So everything that we saw in the ex vivo cellular system was also true in the in vivo. And when we looked at the patient samples, we did primary cultures with the patient samples, and we found that these giant cells also correlated with the poor survival. So clinically also this would be relevant. So now it makes sense to actually look at the mechanism and see how we can prevent this recurrence or target these cells. So what I've shown you right now is that this stage that was missing, and we didn't know what was happening here. We could see that there are certain small cells that were left in our cellular system as well as in the brain of the mice. These cells we call as residual cells. They undergo cell-cell fusion and they form MNGCs. And these MNGCs have a lot of interesting and strange things. They have a high amount of DNA damage. They have senescence phenotype. And we have looked into all the aspects of these cells. But today I'm going to talk about the one that I said, the the reversal of the senescence. So senescence was something that kind of drew the interest of one of my students, because senescence is supposed to be the cell should be arrested, or cell is metabolically active but they cannot divide. It's the end of the life kind of a thing. But still, in our case, we did see that these cells were were able to come out of senescence, and then not only just able to come out of senescence, but when they started to divide and give rise to a very aggressive phenotype. So there are two possibilities that if we want to use it for therapeutics, is to somehow either if we try to understand how these cells are reversing, we make them stay senescent forever, so they don't grow, or we induce them and push them towards apoptosis, so they don't come out and they actually go into apoptosis. So reversal of senescence was something that Madhura was interested in. So what she did is that she took these, in our system, she took the cells at the residual senescence stage, then she monitored these cells, and then she identified a stage where the cells are not showing any senescent phenotype, but they're not dividing as such. So that's like an end of residual stage, because after a couple of days, these cells starts to divide very fast. So this is like the parent population, the residual population, then the end of so-called senescent population, and then the recurrent population. And this is like months of work where every cell and every primary culture she has stained, and she has identified a time point where she would take these cells at different stages. So a lot of work gone over there, and she did see that all the cells in the residual senescent stage were senescent, and the ones that were coming out, so-called the end stage, were not positive for senescence, and then obviously the recurrent cells are not mentioned at all. This is to she just wanted to make sure that the senescent cell that she's seeing, this was with the beta-gal staining, that they at the molecular level also they show all the features of a senescent cell. So she looked at the some of the marker genes like P21, P16, and the protein levels of these genes, and they found that in the residual population, all these markers were high, and in the end of senescent population, these markers were down, and obviously they were not there in the recurrent population. So then the most important thing, which I'll tell you, and then we the reviewers also came back to us with this, was that if you would carefully see this graph here, there are 90 to 95% of the cells that would show senescence. So what about the 5% cells that were not senescent? Is it possible that these senescent cells are not the ones actually that are coming out and start to divide? It's this small 5% population that is coming out and dividing. So we labeled these cells with C12FTG, and we sorted the so-called positive cells, which are really senescent, and the very small population, which was not positive for this drug. And then we grew them and tried to see what happens to these cells. And it was only the senescent cells that were able to come back and give rise to a newer population of cells and give rise to recurrence. And all the cells that were not senescent eventually stayed for a while, but then eventually all of them died. So we are sure that it is only the senescent population cells are coming back and they are able to give rise to the recurrent population. So again, because we had no idea where to start with, we didn't get into an unbiased approach, and we did an RNA-seq to find the differential gene at different time points. Mostly we were interested in two stages, that is the residual stage and the end of the senescent stage and the end of senescent stage. And in the genes that would be upregulated in the residual state and downregulated in the end of residual state, because these would be the genes that we can inhibit and we can have the cells stay as senescent. So data analysis, and we found that a lot of senescent associated genes were high in the, as you can also see from the GSEA, they were high in the residual, the senescent population, and they were low in the end of the senescent population. But one of the very, interesting, I'm not getting into the, I'll try to just keep it to the part where it is most relevant and interesting, because in the paper we have done a lot of mechanistics, but I think for this, I'll not be able to finish in time then. So of all these, this ATF4, which is endoplasmic reticulum stress pathway, was also upregulated in the residual cells, but then it gets resolved in the end of the residual cells, end of the senescent. So we looked at the ER in the parent population, looks fine, normal, like how it should look. We looked at the ER in the residual, the senescent cells, and it was huge. I mean, you can see over here, there was huge human, and it was like really very big and stressed. And the end of this, when these cells go into the end of senescent stage, where they are not senescent but not dividing yet, you can see that it comes back to the normal. So the stress is kind of resolved. And there is then this is the percentage quantitation of the same. We looked at the stress marker, and indeed the stress markers were high in the cells which were senescent cells, and they were low in the parent and the end of the senescent cells. Then we try to look if the protein content, what is it in this ER which is making it like really huge. So we isolated ER, and then we looked at the protein content in the ER, and indeed the protein content in the ER was very high, much higher than the parent population, but then it goes back to the same level as the parent when the cells come out of the senescence. So this protein content and the SASPs are the secretory proteins from the senescent cells. We did make a lot of connection, and we try to, in the paper we have shown what could be the mechanism why this is like that. But here I will just restrict to something else. Okay, so ER stress, when there is stress, there are three pathways that get activated. Either all of them can work in parallel, or one of them will work. There are three pathways, PERK, ATF6, and IRE1, and we looked at all of them, and we found that it is the PERK pathway that was activated in the senescent cells, and this was the same pathway that we also found in the from our transcriptome data. So it was a kind of obvious choice that we would go for it. So we looked at all the markers of the ER stress that were high in RS, and also the PERK QPR pathway, which was high in the senescent cells, and then it comes down in the end of the senescent cells. Which means that if we want to keep the cells, so the theory was that hypothesis was that we would want to keep the cells under stress. So if the cells are able to resolve this ER stress, they're able to come out of senescence. So either they should stay in stress, and if they stay in stress, ER stress for a prolonged period of time, it might also kill them. So lead them to the apoptosis. This is then again we wanted to see how this PERK pathway could be actually resolving this, or how is it that the PERK pathway downstream would be working. So the downstream transcription factor that is activated in this pathway is CHOP. So we looked at CHOP, and then we found that CHOP was high. But interestingly, when we looked at the Western for CHOP, we did find high expression. But CHOP is mostly in the literature shown to be binding to the apoptotic genes and inducing apoptosis. Now, in our system, we did not find any kind of apoptosis. So we were intrigued, like, why is there no apoptosis when CHOP is so high? There could be two reasons. Maybe there is mislocalization of CHOP. It's not going to the nucleus. We did IX, and it is indeed going to the nucleus. Then we looked at the promoter occupancy of CHOP, and cutting a long story short, what we have found that in this certain case where these cells are senescent, CHOP not only binds to the apoptotic genes, which is few, but it also binds to the promoters of the anti-apoptotic gene and the senescent gene, which was not known before. So somehow the occupancy of CHOP changes from apoptotic to anti-apoptotic and the senescent genes, and that's why there is a skewed binding of this, and it is from the apoptosis to the survival that the balance is taken. Now the most important part where we wanted to see, so if we inhibit this pathway, which is giving them a survival advantage, what is going to happen? So we found that there is an inhibitor of PERK, and very importantly, this inhibitor of PERK is also able to cross blood-brain barrier, so which was very good for us. So we first we looked at the cells, we gave the inhibitor, and indeed we found that when the inhibitor is given to the parent population, then nothing happens to the cells grow nicely. If we give an inhibitor along with the radiation, as you can see over here, the cells stop dividing, but then eventually they start to come back. So it is acting as a xenostatic. So they stay in senescent for long, but they start to come back. So that was not right. Interestingly, what we found is that when you give the inhibitor, when the cells have reached this maximum number of cells are in senescent stage, if the inhibitor is given at that stage and not with the radiation before, then the cells die of apoptosis. That is, it is acting as a senolytic. The same with the primary cultures, when we looked at primary cultures. And then, of course, after we give the inhibitor, we saw that, yes, there is, after PERK inhibitor, there is ER stress that is there. And this ER, because we wanted to see that the inhibitor should induce ER stress enough so that the cells eventually die of apoptosis. Then the most important experiment, I'll quickly go to it. The in vivo effect. So there are three regimes that we followed, parent and the inhibitor, where as expected, we did not see much effect, either with the cell line or with the primary cells that we had put in the mice. And then the second was when we give radiation and the inhibitor together, and we did see that there was a dip initially, but as in the in vitro system, it did not last long, and these cells then start to, it was kind of a stable disease for a while, but then they start to come back. Also can be seen over here, there was a dip, but then it's slowly starting to come back when we give the inhibitor along with the irradiation. However, if we give the inhibitor when the cells have reached a state where they are senescent, then we do not see that these cells come back and there is no tumor seen in the mice. So that means that the timing of giving an inhibitor is equal, not even equally, it's more important also to understand, like in this case, that these cells that are vulnerable to this inhibitor are the senescent population. And once this inhibitor is given to the senescent population, then these cells are pushed towards apoptosis, and you don't see any tumor coming back. It acts as a senolytic. This is to show that in the mice, when we give the inhibitor, indeed the cells were in the senescent stage. and which is beta gal positive, and they also had ER stress. These are the different IFCs from the mouse brain. And of course, the last and the most important slide where we show that the IR plus inhibitor also gives a little survival advantage, but not that much. So overall survival is better and statistically significant as well. However, when we give the inhibitor, when the cells have reached the senescent stage, then the survival was much better and much more significant than the other one. So in this studies, what we have demonstrated is that PERT-mediated UPR acts as a switch for senescence reversal, and its perturbation leads to senolysis and prevention of the recurrence in vitro and in vivo, with the overall survival being very significant. The summary is very straightforward and simple. When we give radiation, there are senescent cells. These cells have increased ER stress, and there is a lot of SASP response also there, which is resolved by the PERT-UPR pathway, which leads to the reversal of the senescence and recurrence. If we inhibit this pathway with PERT inhibitor, then the cells would die of apoptosis. So it's like a two-hit senolytic therapy where you give radiation to induce senescence, and then once the senescence is induced, you give a senolytic drug where the cells would die of apoptosis. These results were recently published in Neuro-Oncology, Madhura's work, like last year, 2024. So with this, I would like to thank everybody for coming here, and I hope that you are not shocked that you were here to listen to Amit, and it's Shilpi actually talking, and I could justify my presence here. These are members of the lab. Some of them have moved to JNU with me, and the former members, and the funding agencies, of course, without whom, and the clinical collaborations, which are very, very. Thank you.
Thank you. That's very informative talk. Please open for the questions.
Shilpi, it's a good talk. I just have one question. The therapy-induced senescence and senescence reversal responsible recurrence. So I see a lot of similarity between cancer stem cells and the senescent cells. Stem cells also resistant to therapy, and they also undergo senescence. So at any stage, the cell population you isolate, cells which are undergoing senescence on therapy and then reversal, do they have similarities to cancer stem cell in terms of markers, expression? Did you check on that?
Not check in this study, but we had checked before, and we do see that there is enrichment of stem cells in this population. However, not all it was like really small population fraction that was enriched. So the rest of the cells were not did not at least show stem cell markers. I mean, we did not, to be honest, we did not do functional assays to see if they had a stem cell properties. But I can imagine they might have, but we have not systematically looked at it.
I'm just wondering whether there are subpopulation of stem cells. Suppose if you purify those therapy-induced senescent cells, you may be able to initiate a tumor independently inject them out, and meaning that they are kind of population of stem cells rather than they are another type of population of cells.
They might be, but like I said, because we have not looked at it, what we know that these cells are able to induce tumors in the cells, in the mice, these residual cells, if we take them, and even if we put them in the mice, they are able to produce tumors. Now, if it is because of the stemness enrichment, or it is because, these cells have that property, is what we have not looked at.
Yeah.
Thank you. Excellent work, Professor Shilpi. Welcome again. We are neighbors.
Yeah.
So now. So exhaustive work. So one or two questions. One regarding the SASP. The morphology is changing. The ERs are becoming big. Is there changes in the SASP quantity and the content? Because is it like some kind of a signal which it is telling the others?
Yes. So actually, I skipped that whole part because it was becoming very.
Okay.
Once we saw that our hypothesis was that a lot of these SASP proteins are there in ER, and we looked at so there is a change in the SASP, and in fact we found one of the we have done the cytokine arrays to find which of them would be, and we actually in this same paper that we have taken when we take this the media from these cultures and puts them in another new media, those cells undergo senescence.
So that was my next question, actually. Yeah.
That is something that would maintain the senescence, and with a lot of analysis, we had narrowed down to IL beta because it's upstream to a lot of these SASPs, and it was the one that was like highly expressed in one of these in our samples. So we got the neutralizing antibody for IL beta, and we did all these experiments. Unfortunately, it wasn't. It did nothing.
Oh, okay.
The cells were still coming back. There is something else. There is something else which is helping these cells to maybe maintain senescence, but we are not able to figure out right now.
Okay, another quick question. Yeah. So you told about ER stress, the whole pathway, right? Have you also looked at the autophagy or other this ones which are also parallelly being signaled because it's
Yes, this is something that a new student is doing.
okay.
And we don't have that many data.
Yeah, continuation
right now,
of his
yeah.
questions I have also in mind because he have shown the PARP inhibitor and it is actually inducing senescence as well as inducing the ER stress, but survivability is low. But on the other hand, ER stress can give the reactive oxygen species more. So in that case, like in our research in case of the hepatic stellate cells, it's just opposite way it works. So
ER stress is doing everything that the ER stress should do.
Right.
The thing is that these cells are robust enough to somehow overcome that stress and then come out. And what we actually have not really identified what is making them overcome, but what we have shown is that PERT pathway is really important for this ER stress.
Of course.
So it needs to be the stress relieving pathway. If we inhibit this pathway, then the UPR will be, the whole pathway will be there. So there will be more and more stress. So once there's more and more stress, the cells will be pushed at some point of time.
Yeah, but in EPR pathway also there is a several pathways because there is a three blocks.
That we ruled out. We did the screening and then we narrowed down to PERT pathway. This is the one that works.
Okay. Anyway, short time that is we have to go move on. Hello, कहाँ जा रहे? Hello, students. Please. Session अभी है or there is another talk. Okay, sure. Okay, so thank you, Dr. Shilpi. The next speaker is Sanjeevan Gautam, actually from AstraZeneca UK. He is the associate director, and Dr. Sanjeevan Gautam is accomplished immunologist, and he received doctoral degree from the Freiburg University, Germany. He's here.
Yep, I'm here. Yeah, thank you.
So his postdoctoral training at National Cancer Institute, United States. His area of expertise include T-cell biology, cancer immunology, cell therapy, metabolism, infection biology, therapeutic cell manufacturing, etc. And currently he is serving as a group leader at AstraZeneca and especially developing the CAR T cells and the solid tumor. Very interesting talk. In addition, his research is dedicated enhancing the function of the T cell therapeutic applications. So we'll listen to you, Dr. Sanjeevan. This for you.
I'm not sure if you are able to see my desktop. Okay.
So topic of Dr. Sanjeevan Gautam's talk is modulating T cells for adaptive cell therapy. Very interesting.
Can you see my slides? Sorry for the delay.
Yeah, just skim.
Awesome. Yeah.
Yeah, put it into the slide mode.
Okay. Does it look good?
We can see.
Okay, perfect. Thank you so much for the invitation. So it's really wonderful to be speaking virtually, although in Delhi, because I'm also a JNU alumni. So today I'll be talking about a little bit on T cells and how we can modify these T cells for the enhancement of adoptive T cell therapy. So when we say adoptive T cell therapy, that includes, TCR T cell therapy, CAR T cell therapy, tumor infiltrating lymphocyte therapy, and all. So this is the current adoptive T cell based immunotherapy. So what we do here basically is we collect the T cells from the patient And then we genetically reprogram those T cells utilizing, lentiviral technology, retroviruses, CRISPR-based technology and all. And we expand these T cells in billions and billions of number in the laboratory. And then finally, we infuse back these cells into the patient that have the cancer. So this therapy can be autologous or allogenic. So when we take out the cells from the patient and then engineer them and then infuse back into the patient, it's called autologous therapy. And if we take the cells from the healthy donor, then we call it allogeneic therapy. But basically, this process involves collection of T cells either from the patient or the healthy donor and reprogramming them in the laboratory, growing them in large number and infusing back into the patient. So this is called adoptive T cell therapies. And it can be CAR T cell therapy, as said before, or TCR T cell therapies, or tumor infiltrating lymphocyte therapies. And here is a quick example of one of the successful tumor infiltrating lymphocyte therapy. So what you can see here is the patient, he comes with a metastatic melanoma. So there is a growth of tumor on his head. And this patient has gone through all sort of existing therapies, but he did not recover. So he came to the National Cancer Institute where Dr. Steve Rosenberg and his team took out the T cells from these tumors and they grew these T cells in the laboratory and they infused back into the patient. And what you can see here is the dramatic effects. So on day 76, if you look at this picture, this patient is free of the tumor. And there is another example, so very popular example, and recently India has also built their own, indigenous CD19 CAR T cell therapy in IIT Mumbai. So this is the example of one of the successful cell therapy. So which is again, as mentioned, CD19 CAR T cell therapy. So this therapy was first done in UPenn and also at National Cancer Institute. So this data comes from National Cancer Institute. So basically, this patient with non-Hodgkin lymphoma comes to the National Cancer Institute and his T cells are taken out and they are programmed in the laboratory using retroviral technology. So with that technology, his T cells were modified with CD19 CAR. And then they were infused back into the patient. And what you can see here after 25 months is all these, the lymphoid organs like spleen and the lymph nodes, they go back to their normal sapien size. So this indicates that how potent these therapies are. But the main question exists here, how many patients they are able to receive benefit out of this therapy. So this is the data published back in 2014 by Steve Rosenberg and James Kuckendorf at National Cancer Institute. So what it reports is in the patient with acute lymphoblastic leukemia, out of 100 patients that have received the CD19 CAR T cell therapy, only 80 were able to receive the benefit out of it. So what about other 20? Why don't they get benefit out of this therapy? So that was the major question asked. And then in the another indication, which is non-Hodgkin lymphoma and CLL, the complete response rate was 30%. So what about other 70% of the population? Why don't get benefit out of this, CD19 CAR T cell therapy? So that was the basic question asked. And then Jim Kuckendorf, he went back to the patient. So that's called reverse translational study. So he took out the samples from the patient that have received this CD19 CAR T cells. And he determined the total number of CAR T cells circulating in the blood obtained from this patient. And he segregated the patient based on non-responders and the responders. And what he found was in the patient that are responding towards the therapy, the frequency of the CAR T cells was much higher than in the non-responders. And then he also evaluated the quality of the T cells in the product that was infused into the patient. So basically, there is a cell called memory stem cells. We call it TSCM. So this is one of the phenotype of the cells. So this particular phenotype was enriched in the product that was infused in the responder. So what you can see here is the correlation between this population and the total number of CAR T cells in the blood of the patient that have received the CD19 CAR T cell therapy. And on the right-hand side, you can see the frequency of the memory stem cells is higher in the patient that have received, CAR T cell therapy and that have responded well to this particular therapy. So basically, the conclusion was, the patient that have received the CD19 CAR T cell therapy, if they are responder, they show the increased frequency and the number of CAR T cell in their blood. And if you look at the quality of the infused product, they see that the frequency of memory stem cell is improved. So that was the basic learning out of this reverse translational study. Now, what are these cells? What are memory stem cells? So that's the basic question we have here. So when naive T cells, they encounter antigen presenting cells, so they start to differentiate. So they form memory stem cells, central memory cells, and the effector memory cells. And finally, they undergo terminal differentiation and they form a cell called EMRA population. So what we have found out from the reverse translational study is if you have the higher frequency of memory stem cell, the patient are likely to respond towards the therapy. So what are these memory stem cells? They can be distinguished based on the expression of the various markers. So if you look at these markers, these are memory associated markers. So they are highly expressed in this population. And if you look at the markers which are in the lower section of this table, so they are expressed in lower level in the memory stem cell. So these are a specific type of cells that have the property of stem cell, property, and they also form as a memory cells. They are a part of memory cells. So now this was the experiment done by Luca Gattinoni, I think, who was the keynote speaker yesterday and who is also my postdoc mentor at National Cancer Institute. So basically what he has done on the left-hand side is he harvested the memory stem cells or central memory and effector memory population out of the mice, and he adoptively transferred these cells into syngeneic animals, and he gave them the vaccine support, and he evaluated the expansion of these cells in vivo. So this data comes from the spleen, and what he found out was if you infuse the memory cells into the animals and give them the vaccine support, they expand the most, followed by the central memory population and the effector memory population. Right? And then on the right-hand side, you can see the therapeutic benefit of this particular cell, particular memory stem cell. So what he has done here is he harvested the memory stem cells, central memory, effector memory, and he adoptively transferred into B16 melanoma-bearing mice. And what he found out was the mice that have received the memory stem cells, they were able to eliminate the tumor in more efficient way compared to the other cell population. So this indicates that memory stem cells, they are indeed, very highly proliferative, and they have the better ability to clear the tumor in the mouse model, and which correlates with the clinical data as well. And then there is another example. So what we have done here back in 2016 is, we generated a protocol to develop the memory stem cells. And then we transfuse these cells with CD19 CAR, and we adoptively transfer these CD19 CAR T cells into animals bearing human acute lymphoblastic leukemia cells. So what you can see here on the left-hand side is these are the animals that have not received any treatment. So these mice, they die by day 12 or day 15. And this is the group of animals that have received the standard CD19 CAR T cells. So they are able to live much longer. And their survival is extended for a significant amount of time, which you can see here in the survival curve in the black line. And the final group here is the group of animals that have received the CD19 CAR T cells with memory stem cell phenotype. So these are the group of animals that are able to respond very well. And these mice, they are able to clear the tumor in much more efficient way compared to the standard CD19 CAR T cells. And on the right-hand side, what you can see here is the clear benefit of memory stem cells, CD19 CAR T cells. So meaning like these memory stem cells, they are not only, kind of efficient in the syngeneic model, but also in the humanized mouse model of acute lymphoblastic leukemia. Then we asked the question, is it possible to generate this kind of cells in the laboratory for the therapy, for the treatment of the cancer? And to answer that question, we do a lot of things in our laboratory, and we, play with the transcription factor of the T cells. We either overexpress them or, knock out some of the transcription efficient against the cancer. Or sometimes we play with the metabolic pathway of these T cells as well. For example, if you inhibit the glycolytic pathway, your T cells, they become more efficient. If you enhance the fatty acid oxidation of these T cells, they become more efficient. And then sometimes we use the synthetic biology tools and techniques to enhance the function of our T cells as well. For example, you can express IL-15 receptor on these T cells. You can express IL-12 receptor on the surface. You can, express, the flip molecule, which can neutralize the function of TGF-beta in the tumor microenvironment. And sometimes we also play with epigenetic modulators of the T cells so that it enhances the function of the CAR T cells or the T cells for the adoptive T cell therapy. But today we will be focusing just on the transcriptomic mechanism of the T cell differentiation and the generation of memory stem cells. So now what we have seen so far is, these are the different phenotype of the T cells, naive cells, memory stem cells, central memory cells, and effector memory cells. So we sorted these cells out using the flow cytometer technology, and we did, RNA sequencing experiment. And out of, several genes that were differentially expressed, one of the gene was MIB. And why MIB? So what we found out was MIB was highly expressed in memory stem cells, and the expression of this particular gene was, relatively less in naive population, central memory population, and effector memory population. Then we asked the question, so what's the role of this molecule in the T cells, right? So before understanding or before answering that question, let's know a little bit about this molecule called MIB, which is also called c-MIB. So it is a transcription factor. So what is known about this molecule is it is required for a stem and progenitor cell homeostasis in several tissue. And it is also required for B cell differentiation and the survival of the developing lymphocyte. And the knockout mice has been generated, but they did not survive because of hypoxia. So this is what we know about the MIB, but until 2019, we had no idea about what's the function of MIB in CD8 T cell compartment. So we asked this question, does MIB have a role in CD8 T cell immune response? And in order to answer this question, we adopted a model called PML1 GP100 specific TCR model. So basically what happens in this model is we have a T cells that express a transgene called PML1, which is a TCR transgene. And then this TCR can specifically recognize glycoprotein 100 that is expressed by the B16 melanoma. So we generated these mice, which is a black 6 mice that were transgenic for PML1, Taiwan1, which is a congenic marker. And this is the inducible CRE ERT2, and MIB homozygous flux. So what happens here is if you inject tamoxifen, which is inducible agent, in these mice for several days, CRE is induced, and this CRE deletes the MIB out of the T cells. So we adopted this model in order to understand whether MIB has any function in the T cell immune response, or the response towards cancer or not. So here is the initial piece of data. So basically, in the black, you can see the wild type mice and wild type cells, and in the red, you can see a MIB knockout cells. And the wild type and the MIB knockout cells are validated by the Western blot here. So we harvested the naive cells out of wild type and the MIB knockout mice, and we adoptively transferred these cells into the syngenic animals, and we gave them the vaccine support. And then we monitored the immune response given by or elicited by this wild type and MIB knockout CD8 positive T cells. So what you can see here is the frequency of the CD8 T cell upon vaccination of this animal that have received either wild type or the MIB knockout cells. In the upper panel, we have the wild type cells. In the lower panel, we have the MIB knockout cells. So early on, you don't see a huge difference. But as you move ahead, on day 5, day 10, or even on day 30, what you see here is in absence of MIB, CD8 T cells, they lose their frequency as well as number, as seen here in this particular graph. So this is the data from the spleen, where we, harvested spleen from the animals that have received either wild type or the knockout animals, and we counted the CD8 T cells there. So this tells us that c-MIB is very important for the generation of, enough number of CD8 T cells towards the vaccination, in response towards the vaccination. And it also tells us that it is important for the maintenance of CD8 memory T cells. Then we asked another question. Okay, what is its role in differentiation of the T cells? So if you focus on the flow cytometer plot on the left hand side, what you can see here, this is the wild type kind of a data from the wild type animals or the cells. So what you can see here is about 7% of the cells, they are still positive for CD62L. So CD62L is a marker of stemness. So it's a memory marker. So in absence of MIP, this memory marker goes down. That means we are losing the memory cells. So it's just 2% of, roughly 2% of the cells are positive for CD62L. And if you look at the senescence marker, which is KLRG1, so it is about 15% in your wild type cells. But if you knock out MIP out of your T cells, so it goes all the way up to 60%. So now this tells us that MIP is not only important for the maintenance of your memory or the stemness, but also for maintaining the good health of your T cells. You need to have MIP in order to have the CD62L positive cells and in order to decrease the frequency of aged KLRG1 positive T cells. Then we asked another very important question. So now, if MIB is important for the maintenance of memory cells and maintenance of the CD62L positive cells, so is it important for the stemness of the T cells? So that was the question we asked, and this is the experiment we did. So we adaptively transferred either wild type cells or the knockout cells into the black 6 animals, and after day 45, we harvested these cells again. These are the wild type cells, about 5% are wild type, and about 2.5% cells, they are knockout cells. And then we sorted.
Even if you move little faster because for the time. Yeah.
And then we adaptively transferred into the syngenic animal. And after day 30, we performed the analysis. So what we found was in absence of MIB, you lose the frequency of your T cells. And not only the frequency of T cells, but you are losing also the quality of your cells. That means you are losing the stemness of your T cells. So this tells us that MIB is important also for the stemness of the T cells. Then we asked the question, why it is so? What does MIB do in order to maintain the stemness of the T cells? And we sorted out the cells, wild type cells and the MIB knockout cells, and we performed the RNA sequencing analysis. So what we found in the RNA sequencing analysis is in the absence of the MIB, you see a decrease in TCF7, which is a transcription factor that is important for stemness and memory marker, and BCL2, which is an important molecule for the survival of the T cells. So we found out that if there is no MIB, there is decrease in the expression of these two molecules, and there is increase in the expression of the JEB2, which is a senescence transcription factor. That means it is important for the generation of effector cells and less important for the generation of memory cells. Then we asked the question, okay, if you knock out the MIB, what happens? You lose the memory cells, you lose the stemness, you lose the frequency of the cells. Then we asked the question, what happens if you overexpress the MIB? So we did this experiment. So basically we transfuse our wild type cells with either MIB or just the control vector, and we mix these cells in 1-to-1 ratio and adaptively transfer them into the syngenic animal and perform the analysis after vaccinating these animals. So what you can see here in the green box, it's the MIB overexpressing cells, and the black box is the wild type cells. So early on, you don't see a major difference in the frequencies of the cells, but at the peak of expansion, what you see here is the MIB overexpressing cells, they expand almost 4-fold more than the wild type cells, indicating that if you have the overexpression of MIB, your T cells, they tend to expand better. And then what you can see here is the total number of T cells in the spleen obtained from this animal. So it not only increases the frequency, but also the total number of CD8 T cells in the animals. And we also looked at the phenotype of these cells. If you focus on the upper panel, these are the wild type cells. And in the lower panel, we have the MIB overexpressing cells. And CD62.
Sorry, Sanjeevan, actually, it's already time. Hello? Yeah. Can you wrap up fast? Yeah.
Okay, so basically the conclusion of this experiment is, MIB is important for the maintenance of the memory stem cells. So if you see the more frequency of the CD62L, that means you have the higher frequency of the memory stem cells. And this is the recall response, meaning like, if you have the second hit of the antigen, then you still see the ability of the MIB overexpressing cell is more to expand as well as to generate the memory cells here, which is 18 versus 42. So, based on this finding, we asked the question, can we enhance the efficacy? Can we modulate the T cells by CMYB overexpression so that we can generate more powerful cells for the adoptive T cell therapy against the cancer? So we asked that question, and what we found here in the laboratory is indeed, if you have the overexpression of MYB, you enhance the proliferative potential of these T cells, and you also maintain the stemness of these cells. And then we did the final experiment where we adoptively transfer either wild type cells or the MYB overexpressing cells into the animals that have received the B16 melanoma cell line. So they have these tumor cells, melanoma tumor cells, and they receive either wild type or the overexpressing cells, and we perform the tumor monitoring analysis. And what you can see here on the left-hand side is the mice that have received the MYB overexpressing cells in green. They are able to eliminate tumor more successfully than the mice that have received just the control cells or the untreated animals. And on the right-hand side, you see the benefit of the MYB overexpressing cells in clearing the tumor and also in the survival. And then also we asked the question, so why MYB overexpressing cells are so potent in these animals? And we did a bunch of different analysis, single cell sequencing analysis, flow analysis, and things like that. And the major take-home message here is if you have the MYB overexpressing cells, they tend to bring more T cells to the tumor. So that means it increases the therapeutic T cells in your cancer tissues, leading to the better therapeutic benefit. And also it decreases the exhaustion markers like LAG3, increases the expression of stemness markers CD62L, and also it helps to produce interleukin-2, which is important for T cell survival and function in the tumor microenvironment. And then we also asked the question, does this hold true in the human system? So we took out the human T cells, we sorted them out using flow technology, and we overexpressed CMYB on human cells. And what we found out is, in human cells as well, this MYB is able to overexpress the molecules like CD62L, CCR7, CD28, TCF7, and JAK2, indicating that whatever we have found out in mouse was valid in human cells as well. So what we are doing at the moment, we are infusing cells into the animals that have different tumor types, and we are seeing a positive outcome out of that. So this tells us that we have the ability to modulate T cells in the laboratory to generate stem cell-like phenotype. And this is one of the promising future direction in order to generate more efficacious, T cells or CAR T cells for the cancer therapy. So having said that, I would like to conclude my talk, and these are the people who have helped me to, accomplish on these different projects. And last but not the least, thank you all for your attention. And if there are any questions, happy to take those.
Thank you. Yeah, for a short questions. Yeah, please.
Yeah, can you hear me? The speaker, can you hear me?
Yes, sure, sir. Yeah.
The interesting talk. The MIP and MIC are the same family. They in fact work together as a heterodimer. How unique the function of inducing stemness to MIP alone? Can MIC also do that, or MIP heterodimer also can do the same function?
Well, MYC, that's a good question. Actually, we are thinking to test the function of MYC in the CD8 T cell context. So we have not done that yet. But, I can speak to my data, but MYB, it certainly is able to induce the stem cell-like phenotype. And MYC, we are doing that work, and hopefully we'll be able to answer that question soon. Thank you.
Thank you, Dr. Shinjiban. It's good talk. Thank you very much for the information talk. Thank you.
As we wrap up this engaging session, I would like to take a moment to extend our heartfelt gratitude to all our esteemed speakers and our session chairs. So I'll first invite our session chairs, Dr. Shubhrojit Bushras and Dr. Shamasundaran sir, to please come up on stage to felicitate our eminent speakers. So let me first start with Dr. Kumar Saran sir. Guys, please give him a round of applause. Thank you, sir. Next, I'll be calling upon stage Dr. Puneet Prasad. Now coming on stage, Dr. Shilpi Datta.
You have to give it to the chair also, right? Chair.
Yeah. So now I'll take a few moments to call upon stage Dr. Archana and Dr. Pallavi to give a token of appreciation to both of our session chairs. No, no, sir. So presently you guys are chair for us. Give them a big round of applause. Thank you so much all to be present here in this engaging session. And I request you all to have a small tea break. You guys can go to I2 Mood Court and there you can have your tea break. And please positively come back here in the same venue, F3-Seminar hall within 10 to 15 minutes. Thank you all.
The applause. Dr. Deepika Verma. We are looking for
Good morning, everyone. We now move on to the next session, which is the short talks. I'm pleased to introduce our session chairs, Gauri Shankar Shah from the Bose Institute and Shaukat Hussain from ICPR. ICMR and ICPR.
Okay. So, welcome back to the session of short talk. So, there are a couple of reminders. Like I would request the speakers to maintain a timeline of 10 minutes. And to introduce our first speaker, we have with us Dr. Swati Kulkarni. She is a distinguished medical professional, clinician scientist from LA. And Dr. Kulkarni has earned her MBBS degree from prestigious Father Miller Medical College in Bangalore, India. She is looking forward to matching into internal medicine residency, so I welcome her. So, Dr. Kulkarni, over to you.
Thank you so much for that warm welcome. I'll try to keep it in 10 minutes for sure. Hello, everyone. My topic today is the usefulness of curcumin for cancer immunotherapy. As we know, curcumin are yellow coloring compounds that we see from the turmeric spice that we obtain. You can see here in the image that this is the oral curcumin, which is just 3 to 4% from the spice. The second is the D-methoxycurcumin and the bis D-methoxycurcumin structures, which are not that useful because it's not that soluble in the body. This is my abstract. Curcumins are yellow-colored compounds. Usually, this is taken from the spice itself. And Dr. Nagbhushan Mulki is one of the first scientists to prove that these exhibit the anti-cancer properties and reverses cancer drug resistance in 1984 and 87 and enhances the retinoic acid-induced differentiation and reverse resistance in 2005. Curcumins may be combined with other immunotherapies to improve the treatment efficiency by modulating tumor microenvironment and restoring the T cell stimulatory activities of the dendritic cells, which can help induce the tumor antigen specific T cells. Additionally, properties of curcumins such as the anti-proliferating during inducing the apoptosis as well as suppression of expression of the anti-apoptotic proteins, inhibiting drug and immune resistance and enhancing cancer cell differentiation may boost the conventional immunotherapy modulations that have been used. Results from several NIH and other clinical trials on the various diseases, including cancer, showing promising results of oral administration of curcumins have failed to achieve the pharmacological efficiency in blood levels, and there is no injectable curcumins formulation that is currently present. Recently, Dr. Mulki has shown that curcumins in capsules are insoluble at the boiling point of water at 0, 1, and 2%. And in fat and whole milk, there is the powder formulation that is soluble. And similarly, different cooking methods, there are different solubilities that are seen in curcumin. I will try to explain this abstract in detail as we move forward in our presentation. This is Dr. Nagbhushan Mulki, one of the first scientists to show that the the curcumins exhibit the anti-cancer activity. In the year 1987, he proved the anti-genotoxic, anti-mutagenic, and anti-carcinogenic activity in various carcinomas like the skin, stomach, breast, mouth, and also blood cancers like leukemia and lymphoma. Curcumins in diet inhibit the viral-induced mammary tumors in the mice models and may be useful in preventing viral as well as bacterial pandemics like the human pathogenic flu, influenza, COVID-19 as well. There have been some studies that are done. Reverses anti-cancer drugs like adriamycin resistance in leukemic cells, prevents Maillard reaction, is basically a genotoxic chemical that is generated when you're cooking, frying, or roasting and keeps cells healthy. It's important for prevention of diabetes and many neurodegenerative diseases. In 1988, the experimental model showed that curcumins help in preventing genetic damage caused by BD and cigarette smoking and in humans as well. This is Dr. Nagbhushan Mulki. I'm taking time to explain that he's a phenomenal scientist based in University of Illinois, Chicago, with whom I collaborated in this project, and he's also a research director there. He's been awarded as an ICMR gold medalist, and he has obtained his PhD from the Tata Memorial Research here in India. In 1994, he proved that radiation-induced genetic damage in mice was used in clinics, and the Epstein-Barr virus, which is notoriously known for causing B-cell lymphoma and NHL proliferation, that's non-Hodgkin lymphoma proliferation, especially in post-organ transplantation patients, curcumins inhibited this Epstein-Barr virus-induced human B lymphocyte proliferation, hence showing its antiviral properties. Curcumins prevent multiplication of blood cells by environmental pathogens like bacteria, viruses, and allergens. Based on clinical trial data, it can be positively concluded that curcumins also prevent immunosuppressive drugs like cyclosporin A resistant pathway of human T cell activation. Its potential use can be seen in future, which will help prevent human transplantation, organ rejection, and the graft versus host disease as well. In 2004, Dr. Mulki again proved the curcumin plays a role in human immunity, especially in immunomodulation by enhancing vitamin A-induced human promyelocytic leukemic cell differentiation, and third, reduces the vitamin A toxicity in patients as well. Vitamin A is clinically used to treat leukemia, and curcumins are known to reverse its vitamin A-induced resistance, which is also found in the HPL cells cases. The mechanism of curcumin in cancer prevention, there are various modes in which it acts, especially the signaling pathway, the cell differentiation and the anti-cell differentiation resistance pathway, mutagenesis, oncogenic expression, cell cycle regulation, apoptosis, angiogenesis, and metastasis, prevention of secondary cancer development as well, and modulation of GI oral microbiota as well as the anti-inflammatory properties can be seen. How it helps in modulating the immune cells is by downregulation of the proliferation of T cell induced by the concovalent A, PHA, as well as TMA cells. In the dendritic cells, it reduces, the curcumins reduces the expression of the CD80 and 86 class 2 antigens and suppresses the release of inflammatory cytokines like IL-1B, IL-6, and TNF-alpha. Inhibits the proliferation of B cell lymphomas by downregulating the c-MYC, BCL-XL, and NFK-beta activities. In macrophages, curcumins prevent the regeneration of reactive oxygen species and enhances the stimulation of phagocytosis of peritoneal macrophages in mice models. The natural killer cells, it induces apoptosis by controlling the NFK-beta pathway and suppresses the BCL-XL as well as cyclin B1. So as you can see in this image, the tumor cells evasion strategies that is used by curcumins is, okay, thank you. So it prevents the secretion of immune suppressive molecules, production of M2 macrophage and abnormal antigen presenting cells, and negative stimulatory effect of the TCR, as well as low immunogenicity that is seen in the TAA, T cell apoptosis, and it helps prevent the induction of TREG cells and NK cell depletion and the generation of the IDC and PDC as well. This is the classification. I won't go through the details of how cancer immunotherapy is usually used. I just want to point out towards the combinational approaches, that is the checkpoint inhibitors, which are currently used along with vaccines as well as oncolytic viruses as well as monoclonal antibodies, which is used as a treatment pathway. Curcumins restore the T cell dysfunction via multiple level of immune checkpoint axis suppression. As we can see, it inhibits the receptors like galectin-9, programmed cell death, L1, and TIM3 as well. Curcumin as an immunomodulator, as discussed, we can use it as a nanoparticle, as a monoclonal antibody, as well as a myceliate, always in conjugation with the routine treatment modalities that are used, like TREG cell mediated treatment that prevents the conversion of T helper 1 cells and downregulates the other signaling pathways of FOXP3 and upregulates the INF gamma and CD8 T cells. It helps conjugation along with monoclonal antibodies. When co-associated with the cancer vaccination, it reduces the immunosuppressive factors like IL-6 and induces TNF-alpha. It also enhances the adoptive T cell therapy and activates CD8 T cells. Immune checkpoints are blocked, like I discussed in the previous slide. And dendritic cell-based immunotherapy, it inhibits the STAT3 signaling, and this acts as a dual mechanism wherein it's not only inhibiting the signaling pathway but also is depleting the IL-6 and IL-8 alongside the dendritic cell-based immunotherapy. The limitations that we see using curcumins is solubility and stability. In the GI, acid degradation and microbial metabolism, poor absorption can be seen, and in the bile, it's excreted in the form of hexahydrocurcumin and tetrahydrocurcumin. And there's no pharmacokinetics data that is available online at the moment, and there's no oral administration or IV administration formulation. Okay. One of the solutions that we have found is that if we mix the curcumin powder in diet with the viral-induced mammary tumors or oral administration of curcumin, coconut oil can inhibit stomach cancers. It's seen in mice models, which is proved by Dr. Mulkey in 1987. But we are yet to find out if curcumin nanoparticles can be used in future to enhance the bioavailability of this treatment. These are the references. In conclusion, there are more than 81K articles, 27.5K scientific publications, US patents, and so many grants that have been given for curcumins to check. Currently, it's a #1 alternative plant-based nutrition supplements with a lot of billion-dollar revenue, and it has tripled the Indian export as well. But the research is ongoing. There are a lot of things to be discovered yet. So that's what I'd like to conclude with. Hopefully, we get to know more research and scientific studies in which we can see how we can effectively use it and increase its bioavailability.
Thank you, Dr. Kulkarni, for an insightful talk on the role of curcumin in carcinogenesis. So we will take a couple of questions, quick questions for a minute or so. Do we have any questions from audience? If not, then okay.
Yes, sorry.
It is a very nice. It's called the curcumin is called the golden spice.
Yellow bread. Actually,
Gold
it is
spice.
one of the very 2 to 4% ingredient out of the turmeric spice itself. It's very less in quantity from the actual spice.
The problem, the advantage of curcumin that
Yes.
is a redox dependent molecule.
Right.
Yeah, in the low pH, it acts as a
Definitely.
enolic form,
Yes,
and alkaline pH it acts as a diketoform.
Yes.
So that's the advantage of this molecule because it acts as a multikinase inhibitor. It competes with the ATP. But the problem with the curcumin is the bioavailability
Yes.
and stability.
Definitely.
So in addition to the this nanoparticle,
Yes.
you need to modify this molecule
Yes.
so that it doesn't get degraded. So
Yes.
do you have any approach to modify
I think
this
the
molecule?
mycelial formation of it, if we can reduce the structure of the curcumin into a mycelial formation, it can help better absorption, because I know that micelles along with lipids help in better absorption. As in body pharmacokinetics, we learn that anything that is lipid soluble is more absorptive in the body. So that would be one of the approaches that I would share. And the other one being the nanoparticle.
No, you can use the glucuronic acid mediated degradation inhibitor like piperidine.
Right.
Piperidine.
Piperidine, yes. Like piptaz.
Yeah.
Okay. Well, here in this image, you can see that there are some formulations and theories that are there on the paper out there that we can use it as the nanoparticles, but we have no scientific study that proves that it works to increase bioavailability. There needs to be more study done for it. But to answer your question, I would think is reduction of natural curcumin into curcumin nanoparticles might have an increased bioavailability for future. Yes.
Okay. Thank you.
Thank you.
Good.
Okay, I would now request our session chairs to felicitate Dr. Swati.
Dr. Dil Afroz is here. Dr. Deepika Verma. Dr. Deepika Verma. She's also not there.
So in this session, our next speaker is Dr. Pallavi Verma from the Pallavi Agarwal, Deepika Verma. Yeah, sir. She is from AIMMRCR, New Delhi, and the title of her talk is Epigenetic Targeting of Malform Deficiency Dependency in Ovarian Cancer. Dr. Pallavi is an associate professor and Ramalingam Swami Fellow. She received her PhD from the University of Cologne, Germany, and did the postdoctoral work in University of Cambridge, UK. Her work on demonstration of potential G9A methyltransferase inhibitor as cancer therapeutics. She is a very good. She has a lot of collaboration, and her recent work focused on the epigenetic pathway, including the histone methylation and chromatin regulation. Yeah, you can start. Yeah.
Hello, am I audible?
Yeah.
Yeah. Good afternoon, and thank you for the organizers for giving this opportunity to me. So, one interest of our lab is epigenetics, and this work is on looking at epigenetic targeting of MECOM dependency in ovarian cancer. Now, first, let me discuss with you what is MECOM. So, MECOM is a complex. MECOM is a complex transcriptional locus. It comprises of two promoters that can drive NTS1 and EPI1 gene expression. So multiple transcripts can be generated through this locus. So two major transcripts that are generated are NTSI1, EVI1 gene fused gene, as well as there is EVI1. And this fused gene has been shown to be overexpressed in nearly 10% of acute AML, and it has also been shown that it correlates with aggressive disease and poor survival in myeloid leukemia. Now, our interest in solid cancers asked us to look its value in solid cancers, and we found that it has been reported as an oncogene in various tumor cancers, such as in breast cancer, renal cancer, and also in lung carcinoma. Now, if you look at its mechanism, it is a zinc transcription factor, and it can regulate the transcription of various genes, such as AP1, P10, PML, and thus it can regulate the survival pathway as well as the immune response pathway. Now, what is interesting that this regulation can be both in a positive and negative manner. Now, having this background, our major interest was to look at what is the function of MECOM and it's in ovarian cancer, because not much reports were available in the literature. So we looked at the cancer databases, and we found that 25% of epithelial ovarian cancer tumor samples, they show copy number amplification of MECOM. In addition, we find that there is significantly higher expression of MECOM in ovarian cancer patient samples when we compare it with the normal epithelial surface epithelial samples. Now, this was quite intriguing for us, and we asked whether the ovarian cancers show dependency on MECOM amplification. And if yes, what are the pathways that are being regulated by MECOM, and can we target these? Can MECOM act as a therapeutic target, and can we target it epigenetically, because epigenetic is another aspect in our lab. So to do this, we selected cell lines that show MECOM amplification. So what you see here is the copy number amplification profile of 52 ovarian cancer cell lines, and SCOPE3 and OXAHO showed very high copy number amplification compared to A2780 cells that do not possess this amplification. So these were our model system. Now, by Western blotting, here as you see on the right, we confirm that indeed A2780 do not express MECOM, while we see very strong expression in OXAHO as well as in SCOPE3 cells. To understand the function of MECOM, we did a silencing assay with transfecting cells with siRNA-mediated silencing, and by performing colony forming assays and transcell migration assay, we see that MECOM depletion showed proliferation and migration effect. We also observed that there is a defective activation of phospho-ERK, that means phospho-ERK is reduced upon MECOM silencing. Also, the epithelial mesenchymal transition markers such as Z1, vimentin, and cathepsin, these were downregulated upon MECOM silencing. So that suggested that there is a therapeutic targeting potential for MECOM, and research in the literature, there is no known inhibitors for MECOM till date. And currently, people are trying to target amplified oncogenes by using epigenetics. So we thought that maybe if we can target MECOM epigenetically. For this, we actually performed a screening of 7 different epigenetic inhibitors, and this is the result of that screening. So these are inhibitors that you see here from ZIP04, GSKJ4 and others. These all target histone modifications, that is histone methylation. Some are demethylases and some are histone methyl transferases. Interestingly, so here as you see, we did a cell viability assay treating the cells with increasing concentration of these inhibitors from 1.1 micromolar to 30 micromolar. And we find that ZIP04 can very potently show a cell viability defect. To see that there is no toxicity to the normal cell, we also treated the cells with these inhibitors, PJ-TERT cells, which are normal fibroblasts, human fibroblasts. So this suggested that ZIP04 can be a potent epigenetic inhibitor which targets the histone demethylase family. So we treated the cells with ZIP04 and we found that it is phenocopying the MECOM silencing phenotype. So what does that mean? That means that actually the treatment leads to proliferation defect as same as what we saw with MECOM silencing. It also showed migration defect and also it was showing an effect on the OFF signaling pathway, downstream signaling pathway, as well as inhibiting the EMT pathway. So this was quite intriguing for us. But what exactly is the relation of ZIP04 with MECOM? And to understand that, we actually found that when we treat the cells with ZIP04, MECOM is downregulated. And that was quite a dramatic effect that we saw. So here, due to time constraint, I'm not talking about that we did all these experiments with the two isoforms, which is the E and the Z isoform. Further studies were done in with E isoform because that is much potent compared to the Z isoform. So that's why you would see the treatment with E ZIP04 and Z ZIP04. Right. So as we treated, we see a dramatic downregulation of MECOM expression both at the transcriptional level as well as at the protein level. And further on, so this is basically the quantification of at the protein level. And further on, our idea was that ZIP04 is actually targeting histone modification. It can target three different histone modifications, which is H3K4 methylation, H3K9 methylation, and also H3K27 methylation, and which we indeed confirmed by other experiments. I'm not showing it here. So, having said that, our question was, does ZIP04 modulates MECOM expression via histone modification? And for that, we perform a chip qPCR where on the MECOM promoter using the three antibodies H3K9 methylation, H3K4 methylation, and H3K27 methylation. And what we found was that the ZIP04 treatment modulated the MECOM expression by altering H3K27 trimethylation by altering its enrichment at the MECOM promoter. So, mechanistically, we could identify that ZIP can downregulate MECOM, and this downregulation is because of alteration of H3K27 trimethylation. Now, the next question for us was how exactly MECOM regulating the proliferation pathway. So, I'm not shown, but we have seen that ERK is affected, GAP45AB are affected, and we have also seen that ERG1 is affected, which are all downstream of KRAS ERK pathway. So here in this whole figure, I've really compiled a lot of data. So first thing what we did, we injected mice with the tumor cells and generated the tumors and then injected with ZIP04. Yes, yes. So we did find that there is tumor slowing down of tumor generation, and we also see that MECOM is downregulated in the mice which are treated with ZIP04. In addition, interestingly, we also find that KRAS is also inhibited upon ZIP04 treatment in the mice. Now, the new story that came up was that what is the direct relationship between MECOM and KRAS? What we found in the patient samples that there is a positive correlation of expression of MECOM with KRAS, and we hypothesized that maybe MECOM being the transcription factor can regulate KRAS. So for that, we did again a chip qPCR, and we found that indeed it is regulated in a ZIP04 responsive manner. So just a bit shift to understand a bit more on the translational aspect of this molecule. For ovarian cancer, cisplatin is the key therapeutic, which is the first standard regime for treating the ovarian cancer patients. So here, what we found that the cells which were having MECOM amplification, they were quite resistant, 7 to 10-fold resistant to cisplatin. And when we treated such cells with ZIP-04 and then treated with cisplatin, we can see the resensitization of the cells to cisplatin. So I think this is a quite remarkable study. Taking this into the preclinical and clinicals could have real advantages in the field. Now, our last question was that at the molecular level, what exactly is causing this cisplatin resistance, and can we identify certain genes that are relevant in the cisplatin causing the cisplatin resistance? So again, a compiled figure here. So what we did RNA sequencing for the drug-treated cells, and also we did RNA sequencing for the siMECOM or the MECOM knockdown cells. And then we identified the differentially expressed genes, and these were overlapped. And finally, we could see 34 genes that were differentially regulated both by treating with ZIP-04 and by MECOM. So actually, these are the genes that are regulated by MECOM. So I just retarget ZIP-04 regulating MECOM, and these are the genes regulated by MECOM itself as well as upon treatment with ZIP-04. So when we look at all these genes and try to correlate them with cisplatin resistance, what we could find is a gene called SUB1. Yeah, I think so. So this is basically a transcriptional regulator, and what we found that its expression is high in non-responders to cisplatin, and also we could see that when we treat ourselves with the drug or when we do the knockdown, we increase the expression of SUB1. So this is just a comparison that what we show. So due to time limit, I will not go through it. And this whole work is currently under revision in Cell Death and Discovery. And eventually, I'll thank all the team members. So this all work that is presented is majorly done by Eva Singh, who is sitting here looking for a postdoc, and will be submitting her thesis. Amar Anita Ujjwal supported the whole story very well, and Meenakshi Nal, Sakti Vedini, our other lab members, our collaborators. We thank the funding agencies who have generously funded us, and I thank you all for listening to me. Thank you.
Thank you, Pallavi, for your very concise and very informative talk. The podium is open for questions and answers.
Either they understood everything or they did not understand.
Yeah. Is there any question?
It's not a question. Thank you for an illustrious talk. So just wondering, like you mentioned that, KRAS. So I was wondering that, but they mutant or wild type?
This is wild type.
Wild type. And in that case, like if we have a scenario, we have a mutant, so what would be the role of MECOM and in terms of what happens to P53?
Right, I think that's a great question. But actually, what we see is that MECOM is regulating the transcription. So, because KRAS mutation are point mutations, right? So it's basically just at a single point mutation. So even though if MECOM can bind to the promoter, whether it is a mutated KRAS or if it is a wild type KRAS, it should not affect. So it would be interesting to see that if we can use this understanding in cases where the cells are KRAS mutant as well. That's what I think. It should, because it is a transcriptional regulator. It is not directly binding anywhere onto the protein. It is binding at the promoter region of the gene.
Any other question?
So this MECOM is very interesting because it's a methyl transferase inhibitor.
04.
So did you find that any other target genes are affected by this inhibitor? Because this transferase is very important for some other cellular functions.
Yeah, absolutely. So I didn't have much time, but you see, we did all the RNA-seq data we have, and we are looking into it, that what are other targets that are affected, and we can take it forward. So we do understand that MECOM is one target, and that we confirmed with our RNA sequencing data. But You do
You do see, yeah.
So actually, PIK3CA is another target, let me tell you, which we have seen that MECOM can also transcriptionally regulate PIK3CA, PI3 kinase.
No, I'm not talking about the make one. I'm talking about the inhibitor.
Yes.
Because that could be toxic to the cellular system.
Yes. So for that, we do see that it does not have a toxic effect, at least with the fibroblast cells. We have seen that at the concentrations that we are using, it's much lower and there is no toxicity. And what is interesting is the pre-treatment. So when you pre-treat and then you remove it and then you treat with cisplatin, it shows a very dramatic change of 7-fold reduction in the the concentration that we use for cisplatin. So that combination is worthwhile to look into the mice models, and more studies are required. But unfortunately, we do not have that strong facility now. I can collaborate.
Did he do the molecular modeling to see where it is binding in a reversible manner, or it's a tight binding?
For the ZIP04, you mean? Okay, so functionally, actually, it can bind with 4-5 targets, which are all Romanji histone demethylases, and because of which the resultant effect is on histone modification. So we find that H3K4 trimethylation goes up, K9 goes up, K27 goes up. So, but all these are global. So that is the challenge with epigenetics, that the effects are global. But we have seen many a times that it can be, tissue specific, cell specific, and we see that different conditions show different epigenetic modification, and that's a huge field yet to explore, I would say. Yeah.
Okay.
Thank you so much. We can discuss more. Thank you.
Thank you, Dr. Pallavi, for the wonderful presentation. I would now request our session chairs to felicitate Dr. Pallavi, please.
Speaker online or she's on? Okay. So we have next speaker, Dr. Gunjan Mandal. So she's faculty at ILS Bhubaneswar. So Dr. Gunjan has done her doctorates from Calcutta University, and then later she did her postdoctoral from Moffitt Cancer Center. She has an illustrious record, and she has published papers like Nature, Science, Cancer Cell. So let's listen to her, and she's basically working on gynecological cancers. So over to you, Dr. Gunjan.
So a very good afternoon to everybody. Thank you. A big thanks to the organizers to give me this opportunity to present a piece of work. So the topic that I'm going to discuss today is the B cell responses, how the B cell responses in TLS, tertiary lymphoid structures, antagonize human ovarian cancer progression. So as you may appreciate, our findings in human high-grade serous ovarian cancer, endometrial cancer, and non-small cell lung cancer, that the malignant epithelial tumor tissues are highly coated with class-switched IgA and IgG antibodies, and many others have also found the same result in some other epithelial tumor tissues. So we try to look at TLS structures. Now, what are these TLS? These are tertiary lymphoid structures. These tertiary lymphoid structures are known to because their structure is very similar to the structure of lymphoid tissues. Now, these TLS are very organized conglomerates of B cells, which are surrounded by T cells and interdigitating follicular helper T cells, follicular dendritic cells, like that. So, in approximately 22% of high-grade serous ovarian cancer patients, we have found the presence of these TLS structures with presence of follicular dendritic cells. As you can see, the presence of CD23, presence of follicular helper T cells by BCL6, and also the presence of these TLS has shown us a better survival of these high-grade serous ovarian cancer patients. Together, we also found that TLS positive tumors have a higher density of B cells, CD4 and CD8 T cells compared to the TLS negative tumors. So now we tried to find out that within these TLS, the B cell populations are producing the IgA, IgG, or IgM antibodies, and very surprisingly, we are amazed by the results that these B cell population within the TLS produce class switched IgA antibodies and IgG antibodies more or less in equal fashion, but no IgM. So there is a chance of class switched and more or less more towards affinity maturation. So we further progress to these B cell zones to find out the oligoclonality of this B cell zone within the TLS structures. For that, we stained the tumor tissues with CD19. We cut out those CD19 areas with the use of laser LCA method, laser microdissection method, and we analyzed the DNA's area by immunosequencing, and we are very much surprised and very elated to find that all the 4 tumors of the TLS region, within this TLS region, the B cell clone was highly oligoclonal. The B cell population was highly oligoclonal. As you can see, this first CDR2 sequence is highly dominant in the first TLS, and also you can find the same repetitive CDR2 sequence in the second TLS also. While the respective total tumor area of the same TLS has been shown polyclonal repertoire compared to the its respective TLS. So we aim to produce antibodies by recombinant method, and from the TLS, first TLS, we chose a CDR sequence, and from multiplex PCR results, we recombinantly chose the variable light region for kappa and lambda and produced the antibodies. We produced 3 antibodies, but the TLS lambda and the second TLS lambda antibodies did not produce promising results. So we proceeded with the first TLS kappa antibody. We intratumorally injected that antibody within to a rag knockout mouse, and we found a significant decrease in the tumor volume with respect to relevant IG. Now, what we found previously in our Nature paper is that this relevant IgA in the dimeric form binds to TIGR. That's a quasi-universal receptor expressed on the surface of humans' ovarian cancer, epithelial cancer tissues. So, relevant IgA binding to TIGR, it dampens RAS pathway by dephosphorylating ER4. It makes a call for the myeloid cells and thus facilitating ADCC or ADCP. So now our target was to target the antigen specificity of this antibody, and we did a IP precipitation and sent the readouts for masking. And out of all, we found GPR85 transmembrane G protein coupled receptor to be a unique target for the antibody TNS kappa. And further, from global data platform, we found two peptide sequences on the extracellular surface of GPR85 to be two putative target where the antibody will bind. And finally, peptide 1 found out to be the winner for TNS kappa antibody binding. And we also found relatable similar kind of efficacy for the antibody when we compare with the commercially available antibody against GPR85. Next, we wanted to also check out the efficacy of this or these TLS effect on antagonizing the tumor growth. So, by using the same LCM method, we cut out the tumor tissues which are located just adjacent to the TLS area and a far tumor tissue area from the TLS. We extracted the RNA and sent out for RNA sequencing, and we are amazed to find the result that in the proximal tumor tissues just adjacent to the TLS area has a high inflammation pressure compared to the far tumor tissues. So, as you can see, with a high influence of interferon signaling, TNFR1, TNFR2 signaling. So, in a nutshell, if I summarize, that these TLS has some B cell population, very highly oligoclonal B cell population, which produces class switched Ig and IgG anti-tumor antibodies, and which binds to specific targets and thus produces anti-tumor effect for or antagonizes the human cancer, ovarian cancer progression. In one way, the neutralization method, and in the other, this high inflammation pressure imparted by the TLS generated oligoclonal B cell population also antagonizes the human cancer, ovarian cancer progression. So, I want to acknowledge the core facilities of Moffitt Cancer Center, Florida, and my postdoctoral mentor, Dr. Conejo Garcia, for his NIH funding support. Presently, this work is under revision, so I want to. A portion of it is performed in my laboratory at Bhubaneswar IMS. The funding support is partial from SOLVE and ICMR, and I want to thank my current lab members, my students, and the collaborators. So, thank you.
Thank you. Thank you for finishing before time. So, can we have a quick question? Okay.
Hi, Gunjan. Very interesting talk and like really very interesting concept as well. So I was just wondering if these TILs, all these B cells and all, they also evolve. Like the tumor evolves, there will be new antigens that will be exposed on the tumor. It's always evolving. So is it that this pool of these cells also start to produce new antigen or whatever you have found, or have you like looked at different time points when the tumor is evolving? Is this a pool also changing?
No. So this is a TLS structure. So we have looked upon, like, we have taken a array of samples, but only out of all the samples we found 4 TLSs to have these highly oligoclonal B cell populations. So as you are asking me that if it's that like the tumor is evolving, so maybe we can look at that angle, like when the tumor is evolving, the TLS producing the oligoclonal is a populations are different, or those are same or not. Maybe we can have a look at that.
Yeah, thank you.
No, it's very nice and very updated work. I really like it. So I have a couple of questions. One of them is that since during tumor condition, you will find that the B cell number goes up, but the IgM by IgA ratio goes down. Yeah. So is that and for that T follicular helper cell plays very important role, as well as the B regulatory cells. So did you find the B regulatory cell in this tumor microenvironment along with the B cells?
So very good question. But no, we haven't checked for B regulatory cells. And the coming. Yeah, we haven't checked for the regulatory cells in this one because TLS are the structures where mainly the B cells are there with the surroundings of T cells. Oh, yes. But we haven't checked for the status of it. But coming to the first question, we have checked for both IgA, IgG, and IgM, but we found no IgM. Like none of them produce IgM. So typically, all these antibodies are already class switched within the TLS.
Thank you, Dr. Gunjan, for the insightful presentation. I would now request our session chairs to felicitate Dr. Gunjan, please.
Our next speaker is Aparna Sinha from National Institute of Biomedical Genomics, and she worked with the cRNA and the molecular signature of clinical relevance of preventive HPV positive cervical cancer. I guess she is doing her PhD and from Somila. Yeah, from Professor Somila Singh. Yeah, thank you.
So good afternoon, everyone. Today I am going to present about the distinct cRNA molecular signatures of clinical relevance prevalent in HPV-16 positive cervical cancers among Indian women. Cervical cancer is the second most common cancer among Indian women, and it mostly occurs due to persistent human papilloma virus infection. So HPV-16 is the most high-risk type of virus which can cause this cancer. One should keep in mind that this is most prevalent among the Indian population. So when the virus infects the host system, there is an intricate interaction between the viral genome and the host genome. And as a result of which, the host genome gets completely disrupted and cancer occurs. So this virus can interact with both the coding genes of the host as well as the non-coding genes. There are many reports which has already been published on the coding genes. So our aim was to look into the non-coding genes, whether it can be appropriate for therapeutic or biomarker usage and everything. So our aim was to identify signatures of non-coding RNAs, mostly microRNAs.
For students to be silent.
Which are associated with HPV-16 related cervical cancer pathogenesis, employing a global assay approach. So for this, we have taken human cervical tissue biopsies, and from HPV typing, we have separated these cervical tissue biopsies into two groups. One was the control group where HPV negative, which was HPV negative controls, and the other was the HPV-16 cervical cancer positive cancer group. We had compared these two groups through RNA sequencing and small RNA sequencing analysis in order to understand what are the deregulated non-coding genes present in these samples. So, from the RNA sequencing analysis, we had identified the differentially expressed long non-coding RNA and the mRNAs. And from the small RNA sequencing analysis, we have identified the microRNAs which were differentially expressed all at a cutoff of log 2 fold change of greater than or equal to 2 and p-adjusted less than 0.05. So in order to understand how these differentially expressed microRNAs work, we need to understand this competitive endogenous RNA network. So now how is this working? So microRNAs work in unison with the LNC RNAs, the long non-coding ones, and the coding ones. MicroRNAs have binding elements in which both the mRNAs and the long non-coding RNAs try to bind together. So there is a competition between the LNC RNAs and the mRNAs in order to bind to the binding elements of the microRNAs. So this ultimately leads to the competitive endogenous cRNA networks, which can ultimately lead to cancer. So we needed to understand the intricate networks to identify proper therapeutic targets. So previously, only the mRNAs were targeted. So our aim over here was to identify both the regulating LNC RNAs and the microRNAs behind the expression of mRNA, so that targeting all of these together will be much more effective. Now, how did we choose this cRNA networks? We had identified the DEGs first, that is the differentially expressed genes. From there, we had identified or calculated the correlation coefficients. First between the microRNAs and LNC RNAs, and then between the microRNAs and mRNAs. So why the correlation coefficient? Because it gives us an idea about whether there is an association between the two differentially expressed genes. And then we have selected only the significant negatively correlated samples, correlated gene pairs. So only those pairs which shared microRNA between the LNC and the mRNAs were chosen for this purpose. So after this analysis, we had identified these two particular cRNA networks. One is with downregulated microRNAs and the other is with upregulated ones. So targeting, so on the left side are the LNC RNAs, the middle ones are the microRNAs which are being shared by both the coding RNAs on the right side and the LNC RNAs on the left side. So if we can target the microRNAs and the LNC RNAs together, then it will be much more beneficial in regulating the coding RNAs. Now, these had biological relevance, and apart from the biological relevance, what was more interesting was that they had also prognostic relevance. We found that those genes which were associated with the cRNA networks were somehow significantly associated, most of them were somehow significantly associated with overall patient survival in the TCGA dataset, some of which we had also validated in our own dataset, which had a survival data of around 5 years. So this can be said that they had prognostic relevance in the cRNA networks as well. Now next, apart from the case control study, we wanted to introspect into the intratumor heterogeneity. So we wanted to see what kind of a difference was there among the cancer samples. So we divided the cancer samples based on invasion purpose. So for this, we had taken lymph node invasion. Why? Because in cervical cancer, distant metastasis is not that much common. Instead, local invasion is much more common. So we had taken the lymph node, which is the most local invasion in cervical cancer, and divided the cancer samples into two groups, like PN positive, the lymph node positive and negative. And so we had identified, after that, we had identified the differentially expressed genes between these two groups. Now, from the previous study, we had identified the differentially expressed genes between the case and controls. And from here, we had identified the differentially expressed genes from within the PN positive and negative group and compared these two groups. And from there, we had found the common genes. So these common genes can be considered as the cancer and metastasis associated genes which are responsible for the invasion purpose. So taking these genes, that is the mRNA, LNC RNA and microRNA, which are the common ones, we had formed the cRNA network. We can say that this particular cRNA network is involved with the invasion purpose. So targeting this will be beneficial for those cancer patients where the invasion has already happened. So it is beneficial, and it is also prognostically and biologically relevant. Similarly, we had grouped the samples into early and late stages as well. We have divided the samples, and in the same procedure as I had shown in the previous slide, where the invasion related cRNAs were found, here also we had identified the cancer and stage associated genes with the common genes between the two groups of DGs. And from there, we had identified the cRNA network, which can be said that if we can target the cRNA network, the late stage patients will be benefited if we can target these. So we also found that there is prognostic relevance because it was associated with patient overall survival and biological relevance as well. Now, for the viral part, the most important oncoprotein of the virus is E7, which is responsible for the cancer pathogenesis. So we wanted to see how this E7 gene was actually regulating the expression of the host genes. So in order to understand that, we had taken HPV-16 positive CHA samples and knocked down E7 from there. After knocking down E7, we had perform the RNA sequencing and small RNA sequencing analysis on the untransfected and the transfected samples. Now, after that, between these two samples, we had compared and identified the disease with the cutoff which we had taken. Now, we had integrated this cell line data along with the previous tissue data which we had found. Now, if you can think properly, that in this cell line, when we had knocked down the E7 targets, then those genes which will be getting upregulated are upregulated in absence of E7. That means in the tissue samples, when E7 is almost normally present in the tissue samples, the genes will be absolutely in the opposite direction. So upregulated genes under the effect of sure, sir. So upregulated genes under the effect of E7 knockdown represents the downregulation in presence of E7 and vice versa, just the opposite trend. So we had compared it and we have identified the E7 targeted and cancer associated genes from the tissues and cells together. From there, we had identified these two cRNA networks, which we can say that they are E7 targeted cRNA networks. They also have a immense prognostic relevance because most of the microRNAs over here were found to be significant with overall patient survival, and biological relevance for importance was also associated with it. So in conclusion, we can say that we had identified the cRNA networks for the first time in Indian population with deregulated microRNA and long non-coding RNAs in focus over here, because in the normal and the cancer samples we were able to identify those, and also we were able to introspect within the cancer samples, keeping the invasion property and the stage related properties in mind, and identify those cRNA networks associated with it. So we can also say that those cRNA networks are associated with important biological pathways, prognostically relevant genes which were identified, are harboring immense therapeutic properties. We can target them, and if we can show the functional validation, then it will be much more beneficial for the Indian patients. I would like to thank everyone associated with this study, especially my ma'am, Professor Shomila Sengupta, without whom this wouldn't have been possible. And thank you everyone for listening to me. January is the Cervical Cancer Awareness Month, so please be aware of it and make everyone aware about it. Thank you so much.
Thank you, Banda. Now the session is open for question and answer. Yep.
Yes, please.
Yeah, I had a question that when we are trying to see the 3 different RNAs, you're just trying to take that set which is common in the 2 cohort which you are taking, right? So
yeah, so this one, are you talking about this one?
Yeah. So in all the comparisons, we are taking the common part and then you're claiming them to be the cRNA. But
so the common microRNAs were considered, because if we don't consider the common microRNA between the correlation coefficients, then one will be showing correlation with only one long non-coding RNA. It may not show the correlation with the coding RNA, or the vice versa.
Yeah, that's true. I got your point. Yes. But you because only looking at the that is the common one, you're looking at the expression level and trying to say they are common. But binding property for a cRNA needs several other binding, that at which region it is binding, how is the loop forming, what is the base pair. Did you look into all that aspects for the cRNA?
No, actually we have been considering the already available databases where we can find the targets for the microRNA so that it can be said, but for those we had to functionally validate it, and which we are looking forward to it, that whether we can functionally validate these cRNA networks.
Yeah, because the databases just say that they are just sponging to that sequence. They do not claim them to be cRNA. So claiming them to be cRNA, you need to check further more properties.
So these are all based on our own RNA sequence data, and further we'll validate it in cell lines to see that whether they are actually working in unison.
Yeah, that would be done as a validation. Yes. But before going into that, if you looked at the exact binding, that would be helpful.
Yes, thank you, ma'am, for your
time. Welcome.
Next.
Thank you for a nice talk. I have a question related to this, that you have selected some common down-regulated miRNA. Yeah, exactly. And then you see some upregulated mRNA.
Yes.
And then you correlate these downregulated miRNA with that upregulated.
Yes, ma'am.
Then how you know these downregulated miRNAs is just the target of these targeted genes? How you correlate this?
So after finding the correlation, which we had set a stringent cutoff criteria, if they are meeting that criteria, we had taken that. And also, as I just mentioned now, that from the databases, we will be checking, we had already checked that whether those mRNAs are having targets with those microRNAs. So 3-4 databases we had taken into account, and from there we are selecting the proper targets as well. So if it matches the databases and also with our own data, then only we'll further validate it in the cell line.
Yes, this is very important. Otherwise, it's very create a confusion.
Yes.
Any other question? Yeah.
So, nice. So I was just having technical questions. So you mentioned that you have normal, non-malignant tumors. So what do you mean by normal? So that from you selected number one. So the second question is, you said we did typing only 16. So what was the method you selected that you did HPV typing? So, and did you find any of the and also you mentioned that were there any precancerous lesions involved? Like did you, because there was no clarity. Yeah. So,
for your first question, we take the control samples from only those subjects who come to the clinic with any other problem other than the cancer or malignancy. So those subjects who are there for hysterectomy, then only we take the cervical biopsies, tissues from them, and those are the cancer. Those are the normal samples which we take. After that, we again histopathologically test it and HPV type it to see and check whether they are really non-malignant or not. So in that sense, it is HPV negative normal samples, a separate group of samples which has been taken from completely normal person who has come to the clinic with any other disease other than cancer.
so in that case, like, there may be, chances you have, like if I say, because they're coming to the clinic with some gynecological problems, so it is likely possible that they may have their miRNA degrade because inflammation, those also can, upregulate or upregulate in a sense that there could be a deregulation of microRNAs. So how did you control those during analysis?
So mostly we have a very detailed questionnaire which we usually fill up when we take the samples. So in that questionnaire we have plenty of such questions, whether there is any other kind of infection or any other kind of disease related to it. So only if we can see that there is no other diseases which will be hampering our study, then only we further proceed with those samples. Otherwise, we don't proceed with them. And for the other, sorry, I forgot about. Yes. So there were precancerous samples as well. We had not included it in this study. So we have actually separated it out.
Any specific reason?
So
because I'll tell you why I'm saying so. Once there is a cancer, cervical cancer, so HPV does not have a role to play because it has already done its job. So could be, for your future research. So see, maybe CN1 plus or CN up to CN3. That could be because we need an early detection. So to manage those, so they have a chances. So if it is a cancer, HPV has a least role to play.
Yes, sir. We have those samples in hand, but since we were performing the sequencing study, those were not actually meeting the criteria of the number of samples which we were going forward with. And so for that reason also we had excluded it because the main aim was to separate the control and the case samples. But in our future also we had planned that we'll do a progressive study where we can see the SIN 1, 2 and 3 properly.
Thank you.
Thank you, sir. Thank you, sir. Thank you.
Thank you, Dr. Sinha. I would now request our session chairs to please felicitate Dr. Sinha, please.
So because of only one cancer, I would say, if it is detected early, it can be cured 100%. Thank you. We are one of our students. You haven't been paid, right?
They're very proud.
So we have a last speaker of the session. So Dr. Pratibha Shrivastava, she is scientist C at Agarkar Research Institute, Pune. So she has done her PhD from Pune University, and then later on she has done her postdoctoral fellowship from NIH. So I welcome her. So she is a chemist. So we'd be happy to listen to you. 10 minutes.
I may have a different perspective, whatever I have in the presentation here on the gene expression or upregulation. I will give you a different overview. And yesterday in the dinner, we were cordial friends. Now we are healthy competitors. So, since in the introduction, I did not do my PhD from Pune University, I did it from CDRI Lucknow, and I have my batchmate here. So, let me present, and I will take 10 minutes only. So, all that time, whenever we, let us skip this one, we talk about the breast cancer or any kind of the cancer. There are so many types of treatments, stuff, research is going on, and people are making new drugs. So many drugs are synthesized, they go into the clinical trials. They sometimes they vanish in the clinical trials. And if you see that in the breast cancer, it is basically women's are target in the different stage of their life. Maybe male are also target, but women are more target due to their lot of hormonal imbalance during their life stages. And the premenopausal or menopausal women are more vulnerable for the breast cancers. So I have the point of view that when we are making drugs or something like that, as I have shown here, if we will see that natural products, that paclitaxel and all that, they came into the market as a single entity. So when they are in the single entity, there is nobody to counteract their toxicity profiling. So what happens that these are the single, then we have to do toxicity studies, annual studies and everything. So and even the hormone therapy, when we go for that, hormone therapy also has its pros and cons. And targeted therapy, that is a very new area, and there are so many target therapies. But the negative effect is what? That drugs are so costly because it is not affordable for every sector people. Because suppose, of course, if you will ask me that take 2.5 lakh injection for 1 month, I cannot afford it. So the thing is like that medicines or treatment should be in that way that everybody can afford, those who are people, because the disease do not differentiate between the weaker people and the richer people. Celebrities can afford it, but not the weaker. Okay, so this is what I have given. So I have taken a purview of the power of the Ayurveda. Ayurveda is what? That natural product based medicines where many compounds work together synergistically. And recently we have seen that this lady from the Prime Minister of Kenya, she got tumor in her brain. When she got treated, she lost her eye. So the Prime Minister asked that Kenya Prime Minister that come to the India, we have the Ayurveda to try to treat for her eyes, and she had got her eye back. So that is the power of the Ayurveda, or we can say that natural products, where so many compounds are working together, and they counteract the toxicity also, and they help us each other to bring very positive effect for the tree. So I will say not natural product, I will say food waste. So food waste, that if you see that orange, we eat the orange and we throw the peel away. Okay, so that peel has too much potential than the fruit. So that I will show you how it has too much potential. So I will correlate with the theme of this conference. If you say immunity, okay, that immunity will be boosted. If you say the cancer treatment, that cancer will be also cured with this one. It will help in improve the digestion also. So there are so many positive effects of this peel which we are throwing. So why not convert these food waste into the potential that it will be treated? And this is not, we will say this is economical also, that anybody can afford, even the weaker people can afford. And always we say that prevention is better than cure. So if you are taking these kind of things in your life, maybe you will not go to that way that you are getting cancer, although cancer can be entered in your phase of life. So before doing that, what we have done the extraction protocols. We tried to extract it in different solvents and wanted to know that what are the ingredients inside it. Some of them are on the tips of the people, okay, that orange peel, it has hesperidine. Some of them it will say tangerine. So when we have done with the LCMS, and LCMS is the instrument which is highly sophisticated. It can identify even if it is in trace in the carbon. So very traceable materials all can be treated. And we know that all plant metabolites, they are in the ppm level. But in that ppm level, they have the potential to treat you. So in that way, we have some method development. So you have seen in that chromatogram, so many peaks are there. And each individual peak belongs to the different compound. So that peaks I have, when I have tried to find out, I found almost more than 90 compounds in that particular orange peel. And that orange peel has so much tremendous potential. So if you see that how we can identify, this may be boring for you people because all of you are from the biotechnology. But I will say one thing, how we identify, that is the top in the blank, there is nothing is coming out. When we are doing it, that single piece it is showing with the aspirin. So it is a three-way technology. Three-way, it will give you TIC, it will give you HPLC chromatogram, and it will give you mass fragmentation. So that it will confirm which compound is it. Okay, and it has the tendency to, even these two peaks are overlapping. So my friend sent me the sample. There was only one peak in the HPLC. But when I did the LCMS, I sent her that there are a number of compounds in your sample. So only you should not believe on the HPLC for the Yeah, I know. So these are the compounds we have identified. Now I don't want to go into the detail that these are the compounds identified in the different extracts. I will go further. And this is the thing which we have done. Why it is important? Compound is there, but what is their quantity? So for that quantity, we have done MRM method and find out that these are the major compounds which are present in this much quantity in this particular extract. So in that way, we have done the quantification. Now I'm coming to the point where the theme is of this conference is here. So we have done heat map analysis to correlate the different extracts and with the respect to the compound. We have done molecular docking also for the individual component. how it can correlate with the activity with the estrogen receptors, which are related with the breast cancer. We have also done the toxicity profiling in the animals and everything that I have done. So this is the cell viability also. So not only the oral, I try to make that edible formulation where you don't need to put any kind of toxic material. That edible which eat every day. So there should not be toxicity in the formulation to be given to the patient. So people will remain happy if you give them the molecule or the formulation which is eatable, which will not give them side effects and everything. So they will happily take your formulation instead of this one. So this is the results where I showed you more than 90 compounds I have identified in that. And future perspective, we want to do detailed animal toxicity on the breast tumor, even for the ovarian cancer and all that. I want to correlate how much it is useful to treat the cancer of different types. Okay. So I would like to thank the conference organizing committee for giving me an opportunity to be here, and RGSTC who have sanctioned me 7.6 crore project for identifying the potential of the medicinal plant. At the same time, I am thankful to the CCRAS and of course my director for providing me the infrastructure to generate such kind of data. Thank you very much.
Thank you, ma'am, for an thought-provoking cancer prevention research that you are doing. And the take-home message, what I could summarize is that, I'll come to that. So maybe we can have a couple of quick questions.
Initial part is good that everyone understands it's costly, but how you want to take it forward, that you have so many chemicals, what you're going to do with it?
Yes.
As far as the patient, because you started with the patients are suffering, so how you're going to take it to the patient?
That's why I said I will make that oral formulation, which will not be in the tablet form or this liquid.
That you will make it, but then what?
Then if you eat it like fruit you have eaten in.
No, so how do you prove it is working? Just because it works in cell line, out of hundreds of chemical which works on cell line, hardly anyone works in patient. So how you are going to prove your chemical will work?
I have not said a single chemical, sir. There are n number of chemicals which work in the synergistic, as you see in the iron.
How are you going to prove?
Proof is that when we do the animal testing and all that, we will see that breast tumor is reducing or not. I will see that ovarian cancer is reducing or not.
I'll add to it, and this is for because there are younger guys here. This is called nutraceuticals. Please be careful.
Yeah.
You have to prove or proof. You need to give it to the patient. Show it works in the patient.
Yes, sir.
Otherwise, there are n number of these kind of nutraceuticals with so-called efficacy in the market, and we need to be very careful.
Sir, I can answer you in a different way. COVID, you have seen that, okay? So many patients who have been hospitalized, some of them are not survived. And those who went for the naturopathy and eating the natural products or the nutraceuticals, they felt better.
So, hold on, sir. You have younger guys here. Where is the proof, madam? Where is that paper? Peer-reviewed paper, please quote one.
Okay. Sir, that people have to write on the basis of the feedback.
Not feedback. You are a scientist. Peer-reviewed paper which you quote, published in this place, this where, peer-reviewed by your group of people, and say, hey, patient benefited.
Thank you, sir, for your any other during the lunch. So the message is also we should take fruits and vegetables to boost the immunity. So that is one of the takeaways from this talk. Thank you.
Strong immunity.
Yes. So thank you all the speakers. Yes.
And for keeping their team time.
Yes.
Okay, Dr. Pratibha, thank you for your presentation. I would now like to request the session chair to please felicitate Dr. Pratibha. Students, you please need to wait for a minute, please. You can settle down for a minute. I would now request Dr. Priyanka and Dr. Sukhdeep Bose to felicitate our session chairs, Dr. Gauri and Dr. Shaukat. Students, please settle down for a minute.
So you did the testimony of your research. So we are getting Ayurveda.
Lunch, participants can go to I2 for the poster session and the lunch. We assemble here back dot 2 p.m.
And it's my honor to introduce to you our session chairs, Professor Gauri Shankar Sir and Dr. Sonia Kapoor ma'am. So Professor Gauri Shankar Sir is a fellow of National Academy of Science and is a leading expert in cancer biology and tumor immunology. His research has significantly contributed to understanding cell cycle regulation in cancer, immune dysfunction in the tumor microenvironment, and microRNA based gene therapy for overcoming immunotherapy resistance. His work is highly recognized with numerous high impact repeated publications, awards, and patents. So please come on the dais. And next, I'll be calling upon stage Dr. Sonia Kapoor, Associate Professor at MSR Amity University, Uttar Pradesh, Noida, specializes in tumor metastasis and drug resistance, integrating nanotechnology, biomaterials, and modern cell biology tools with a PhD from IIT Bombay. She has authored several books, completed UGC, DST, DBT funded research projects, and teaches courses in immunology, cytology, and genome engineering. So over to you, sir and ma'am, to call our next speakers for their enlightening talk.
Hello, everyone. Yeah, it's my I'm really honored to, as I have been asked to chair this session, this prestigious session, in a session which is tumor microenvironment, which is in the core of my heart. So the first speaker is Dr. Rajendra Kumar Motiani, and the title of the talk is Saga of Oncochannel Oral ORI 3 in pancreatic cancer progression from discovery to delineating the role of metastasis and chemoresistance. Dr. Dr. Rajendra Motiani is an associated professor and Wellcome Trust DBT Alliance Intermediate Fellow and a distinguished career. He has a distinguished career in biomedical research. He earned his DMB degree from Nagpur University, Nagpur Veterinary College, and then completed his PhD from the Albany Medical College in New York, and then followed for postdoctoral work. Then he joined Center for Biotechnology, and Dr. Matiani also served as an Inspire Faculty Fellow. So, welcome, Dr. Matiani. So, I request all the speakers to quickly adhere to the time so that we can have some time for interaction.
So, while the slides are coming up, I would like to thank the organizers and thank you, Professor Shah, for kind introduction. So, okay. So I am coming from Regional Center for Biotechnology. Those of you who have not come there yet, we are located in the scenic Aravali Hills, some 30 kilometers from here. And our research program focuses around pancreatic cancer. So obviously, first question that comes in our mind is, why do we care about pancreatic cancer and why should we study pancreatic cancer? And the original motivation comes from this 5-year cancer survival rates, and this data is coming from US. So if we look at it, 1970s, 1 out of 2 patients that are diagnosed with cancer could not survive more than 5 years. That has increased to now more than 2 patients out of 3 survive. In some cancers, we have made significant progress. Almost all patients survive 100% of them survive 5 years. But if we go through this graph, right at the bottom is pancreas. Pancreatic cancers in 1970s, only 2% patient used to survive, and today also it is less than 10%. And if we talk about India-specific data, that is also very alarming. And this is the data coming from last 2 decades between 2000 and 2019. The cancer type which is showing highest rate of increase in mortality is actually pancreatic cancer. So this is majorly because changes in our lifestyle and the dietary changes that we are going through as a society, and that has led to increased incidence and mortality rate in Indian population as well. So our lab focuses on ion channels, basically calcium channels, and obviously the next question comes, why do we care about calcium in this cancer sort of scenario? And to do that, I mean, if we look at, there are good amount of literature and good reviews out there that clearly show that calcium plays a very important role in different hallmarks that are involved in driving cancer, like proliferation, tumor cell growth, tumor cell death, metastasis, angiogenesis, and lot of tumor microenvironmental changes that are pro-tumorigenic in nature. So, based on this, the working hypothesis in our lab is that when a normal cells, they transform to cancerous cells, that is concomitant with the changes in the expression pattern of calcium channels transporters present either on the plasma membrane or intercellular organelles like mitochondria and ER. And what it leads to different sort of calcium signals. And fortunately, there are some FDA approved drugs that can be used or repurposed for targeting or manipulating these signals so that downstream signaling can be controlled in a way so that we can stall cancer progression. So with this, some 6 years ago when I started my own group at Regional Center for Biotechnology, we did a massive exercise based on this hypothesis, looking at what are the ion channels that are differentially expressed in pancreatic cancer. And one such ion channel, I'm only talking about one ion channel today in the interest of time, which is ORAI3. What is ORAI3? I will come to it in a minute. So what we observed that in pancreatic tumor cells, so this is the data coming from TCGA, Tumor Cancer Genome Atlas. And what we see that in pancreatic tumor cells, there is overexpression of this channel ORAI3, and the patients which have higher ORAI3 levels could not survive beyond 5.5 years, whereas a proportion of patients that have low ORI3 levels, they can survive beyond 8 years, suggesting that it is somehow associated with poor prognosis and it is overexpressed in pancreatic cancer. So with this, we started investigating the role of ORI3 in detail. First thing that we did, we looked at, is there any relationship between ORI3 expression and the metastatic nature of these cancer cells? And what we found that the pancreatic cancer cells which have been able to metastasize to different sites like spleen, liver, they have higher expression of ORI3 compared to non-pancreatic epithelial cells or non-metastatic cells. So, what is ORI3? Actually, it is a calcium influx channel, and the way it functions that when growth factors or hormones bind to GQ receptors, GPCR receptors or tyrosine kinase receptors, then it leads to generation of IP3. IP3 goes and binds to IP3 receptor present on ER, resulting in calcium coming out of the ER. And what happens at this time point, typically under resting condition, a sensor protein STIM1 senses ER calcium levels. When the calcium levels within the ER go down, this sensor then oligomerizes and transfers the message to the plasma membrane channel so that they open up and the calcium from outside can come into the cells to drive their various cellular processes. And this process itself is known as store operated calcium entry because intracellular stores are driving the opening of plasma membrane channels. So we started looking at the role of ORI3 in pancreatic cancer. First thing that we did to study this, we generated a good, cell lines, stable cell lines. We used both normal pancreatic epithelial cells and cancerous cells, made stable cell lines, and continuously using these stable cell lines, which show a decrease in ORI3 for our functional assays. First thing that we did, we looked at if ORI3 is playing any role in pancreatic cancer proliferation. And as you would appreciate from this, in a cancer cell type, when we knock down ORI3, we see a significant decrease in the proliferation. Whereas in a normal pancreatic epithelial cells, we do see some effect, but it is marginal compared to cancer cells. So to cut this long story short, we did multiple in vitro assays, but I will just come down directly to the in vivo experiments. What we did using these stable lines, which were expressing GFP, we performed xenograft studies, and as we can clearly appreciate from this, that the tumor volume or the tumor growth rate was significantly decreased when we knocked down ORI3, and so as the tumor weight, and one can clearly appreciate from the tumor pictures that were harvested after 3 months of initial injections. Moreover, since these cells were tagged with GFP, we could perform biofluorescence studies in live animals, and what we are looking at is GFP signal from a mice, and as you will appreciate, in a control condition, we see bigger higher signal coming at the initial site of tumor injection, as well as lot of metastasis that is going on. Whereas when we knock down ORI3, there is lot less of metastasis, and one can even appreciate it in this picture, wherein we are looking at liver and lung area, there is higher biofluorescence in case of control condition compared to ORI3 knockdown. So this work is already published, so I will just summarize this. What we have reported that ORI3 is overexpressed in pancreatic cancer through several signaling cascades. It regulates both initial tumor formation as well as secondary metastasis. And when we knock it down, we see smaller size tumors, and as well as there is a substantial decrease in metastasis. Now I would like to switch gear and talk about some unpublished data, and I will talk about chemoresistance now. Chemoresistance, when we talk about chemotherapy in case of pancreatic cancer, the first line of therapy is gemcitabine, which is well established. But another point that is well appreciated in the clinics is that gemcitabine resistance is a major cause of treatment failure in pancreatic cancer. So again, we looked at is some role of calcium reported in gemcitabine resistance. Indeed, there are some studies that have come out in last 3-4 years that have reported that STIM1 plays an important role in reprogramming in pancreatic cancer cells upon treatment with gemcitabine. And indeed, in an independent studies wherein people were looking for the potential markers, biomarkers for gemcitabine-induced pancreatic chemoresistance, people have reported STIM1. And again, I would like to remind here that STIM1 is the sensor that activates the channel on the plasma membrane. And to drive the function, eventually we will need calcium coming from the outside, and the identity of that channel remains to be discovered. So we asked a very simple question: is ORI3 that channel? Is it ORI3 that plays a role in chemoresistance? To do so, what we did, we generated three pancreatic cancer cell lines with gemcitabine resistance. MIAPACA2, PANC1, and CFPAC1. It took us 3 to 4 months to get these, gemcitabine resistant cell lines. As you will appreciate that with the resistance, CFPAC1 shows the highest amount of resistance compared to all three lines. Further, we molecularly validated this resistance by looking at the expression of the transporter that is involved in gemcitabine uptake and enzyme kinase which is involved in its metabolism. And when we did this analysis, again we found that CFPAC1 shows highest level of gemcitabine resistance. So going forward, we have used CFPAC1 for our chemoresistance studies. First thing that we evaluated, what happens to ORI3 in gemcitabine resistant cells. And we use STEAM1 as a positive control, which has been already reported in literature. STEAM1 was going up, and so as ORI3 was going up. Now we started asking, is this increase in ORI3 actually contributing to gemcitabine resistance? And for that, what we have done, in the gemcitabine resistant background, we generated ORI3 knockdown. So this is a control line, SHNT, and then SHORI3, wherein ORI3 levels are knocked down. And what we can appreciate from here, that in comparison to this brown line, which is parental, the yellow line, gemcitabine resistant line, has a higher IC50 values. And SHNT, which is a control, looks similar to the gemcitabine resistance. But when we knock down ORI3 in this resistant background, this IC50 decreases significantly, even less than the parental cell line, suggesting that ORI3 is actually contributing to this gemcitabine resistance. We validated that by performing FACS studies and apoptosis. Then we went on to look at is ORI3 also contributing to gemcitabine induced hyperproliferation, and that we did by using BRDO assays. Again, all those 4 cell lines, if we compare blue versus this red, you can clearly appreciate that ORI3 is playing an important role in gemcitabine resistance. We performed cell cycle analysis and found that ORI3 actually regulates G0, G1 phase transition, and thereby it regulates gemcitabine resistance. We did lot of functional assays. One of them was again migration. As we know that resistant cells have higher migration rates, but if we knock down ORI3 in this background, we see a significant decrease in the migration upon knockdown of ORI3. Then, important characteristics of any chemo-resistant cell type is a stemness property. They develop stemness property. So what we did, we looked at stemness properties by performing spheroid assays. These are hanging drop spheroid assays, and what we can again appreciate that in comparison to parental cell line, gemcitabine resistant cell lines have higher stemness properties. And if we knock down ORI3 in this background, we see a considerable decrease in this stemness. We have validated all this at molecular level as well. We have looked at stemness markers, and they also go down when we knock down ORI3 in the chemo-resistant cell lines. Now, the important thing is it having any relevance in vivo as well? So to understand that, we have used zebrafish as a model system. What we have done injections in the 2 DPF zebrafish, that is 2 days post fertilization, 48 hours post fertilization, and we have followed the metastasis in the zebrafish. So typically the zebrafish will form a xenograft at the site of injection, and then the migratory cells, they move towards the tail. And when we perform these assays with the 4 different conditions that we have developed, what we could clearly observe that in comparison to control condition, the gemcitabine resistant cells show higher metastatic potential, which is already reported in the literature. Importantly, what we observed that when we knock down ORI3 in this gemcitabine resistant background, this metastatic potential is significantly decreased, and data from tens of fishes is quantitated over here, suggesting that indeed in vivo also this holds true. Now the question is how this is happening at the molecular level, how ORI3 is driving gemcitabine resistance. To understand that, we took advantage of bulk RNA sequencing. We performed RNA-seq between these 4 different cell types that we have formed. Again, I would like to remind that we have 4 conditions: parental cell type, gemcitabine resistant cell type, and in the resistant cell type, we have either control or we have knocked down ORI3. So essentially, we have 4 different conditions. And what we looked at very carefully the differential regulation, meaning the genes that are increasing when cells are acquiring gemcitabine resistance, and the genes that at the same time decrease when we knock down ORI3 in that condition. And when we did that sort of analysis and looked at the Venn diagram, essentially looking at the genes that increase with the chemo resistance and go down when we knock down ORI3. And at that, when we did that analysis, we found 42 genes coming up. But we wanted to narrow it down to 1 or 2 potential players to start the work with. So we applied one more factor, and over there we used this database, which is calcium genes database. And when we included that, we found only one gene that qualifies all 3 criteria, and that was SLIT3. So we started, reading about SLIT3, what it is, and can it be a potential player involved in the ORI3 mediated gemcitabine resistance. So SLIT3 is a extracellular matrix protein which is secreted out. It interacts with its receptor ROBO, and downstream it is known to play an important role in cytoskeletal reorganization and thereby contributing to cell migration. So this was very interesting, so we followed it up. We looked at literature if there is anything known about SLIT3 in cancer. Interestingly, what we found that SLIT3 is reported as a negative regulator of tumor progression in thyroid and hepatocellular carcinomas. When we started looking at SLIT3's role in gemcitabine resistance in pancreatic cancer, what we found that when we knock down SLIT3, the sensitivity actually decreases, meaning that somehow it is contributing to gemcitabine resistance, which can be also appreciated from the spheroid assays. Knockdown of SLIT3 decreases the stemness properties of gemcitabine resistant cell types. The migratory properties as well go down when we knock down SLIT3. But important question was, is SLIT3 working downstream of ORI3? Because our goal was, because we identified ORI3 as a potential regulator of chemo resistance, is SLIT3 working downstream of ORI3? So to do that, what we have done, we have overexpressed SLIT3 in ORI3 knockdown background. We could see a very good overexpression of SLIT3 in this condition. And when we overexpress SLIT3 in ORI3 gemcitabine resistant background, as you will appreciate, that the cell viability considerably increases, essentially suggesting that indeed SLIT3 could be working downstream of ORI3. We have validated this by performing several assays like spheroid assays, migration and invasion assays, and all of them collectively suggest that downstream of ORI3, SLIT3 is the player that drives chemo resistance in case of pancreatic cancer. So if I summarize this chemo resistance studies, what we have observed that when cells acquire, pancreatic cancer cells acquire gemcitabine resistance, there is increase in ORI3 expression that brings in calcium, which leads to increase in SLIT3, and this SLIT3 increase provides more stemness and self-renewal properties and enhanced migration and metastatic properties. When we knock down ORI3, we see decrease in the levels of SLIT3, and again the chemo resistance goes down, and the other properties associated with chemo resistance like stemness and migratory properties also go down. So collectively, taking an unbiased approach, we have identified a novel targetable pathway that can be used along with the first line therapy of gemcitabine. So as the title suggests, the saga of this onco-channel in pancreatic cancer, what we have shown collectively that in human pancreatic cancer samples, the expression of this ion channel is increased, and that in turn drives cancer progression and metastasis. I just showed you that it plays a very important role in pancreatic cancer chemo resistance. What I could not discuss today in the interest of time is that this channel also plays a very important role in partial EMT, which is epithelial to mesenchymal transition in pancreatic cancer. And this property of regulation of partial EMT actually feeds forward to both higher metastasis and chemoresistance. So collectively, ORI3 is emerging as a key player driving several aspects of pancreatic cancer progression. And with this, I would like to thank my team, very dedicated set of students. The work that I presented is almost completely done by senior graduate student who is about to graduate, Samriddhi Arora, collaborators, and funding from several funding agencies. And with this, I would like to thank you all for your attention and will be happy to take any questions.
Thank you, Dr. Natami, for finishing your work well ahead of time. So we have some couple of questions. Switch on the microphone, please. Hello.
Yeah, it's very interesting talk. So when you saw the ORI3, have you seen this in the gene expression databases, any TCGA or any other pancreatic cancer database? And also the another question is that the SLIT3, the other gene which you have seen an anti-correlation. Okay, have you also seen these two genes in the database, whether they anti-correlated?
Yeah, it's a very important question, and actually, ORI3, I started my talk with that. So first, the initiation of this work was that we looked at TCGA data sets, also GPR data sets, and essentially looking for ion channels that are differentially regulated between normal pancreas and pancreatic tumors. And from there, we picked ORI3. So the ORI3 initiated from that big data set. Right. SLIT3, again, we have seen it is what I have not included again in the interest of time that data. SLIT3 also follows the same pattern in case of pancreatic cancer. But in hepatocellular carcinoma and thyroid cancer, it has been reported the other way around. But there is a difference. I mean, hepatocellular carcinomas are usually very solid tumors. And whereas pancreatic tumors, they are relatively not as solid as those tumors are. And they have lot of fibrous tissues also contributing to that their tumor volume. So that those sort of differences that are there in the tumor microenvironment, that might be because this SLIT3 is a extracellular protein, which is secreted out. So what my thought process is at this time point is that because the tumor type and the tumor microenvironment is different across, say, hepatocellular carcinoma versus a pancreatic tumor, and that could be the reason that we are seeing differential role of SLIT3. But we are very confident because, if we go through those details of volcano plots, one gene that pops out very far away from that volcano is SLIT3, and showing very strong regulation in all three different ways of, the gating ways that we have used.
Thank you.
Excellent. Excellent work, Rajendra. So my question is out of curiosity. You told that in pancreatic cancer, there is overexpression of this ORI3 channel, which is a calcium channel basically. So does it also affect the other side neighboring channels present in the plasma membrane, such as sodium channel or potassium channel? Have you ever looked into that?
Yeah, so we have not looked at sodium or potassium channels in this context, but there are calcium sensitive sodium and potassium channels. So there is a possibility that changes in the cytosolic calcium levels may activate them as well, but we have not looked at them.
Yeah. So I have some questions. So did you check that dermatitis resistant cells, they possess the stemness marker like OFF, NANOG, and the CY3P450 knockdown?
Yeah, so we have checked all of them, but again, in the interest of time, I have not shown the molecular data.
My second question is, when you are developing this resistant cell line, you have to constantly give the germicidin at low dose. If we withdraw the germicidin at that point, did you find any differentiation, de-differentiation of this stem cell into cancer cell? Because either they differentiate or they make the uneven division to make their own population and the cancer cell.
Yeah, so we have not done exactly the experiment where you are asking, because as you pointed out, to maintain this resistance, we have to keep a low dose of gemcitabine all throughout. So we have not done any experiment where we have taken away the gemcitabine resistance 6 months down the road and looked at how they are now going back to the.
No. As soon as you will withdraw, the stress, germicidin stress withdraw will again the cells will be differentiated.
Yeah, we have not done that experiment.
That will be interesting. Then you can reuse another drug to make them sensitive.
Right.
Yeah,
it's a good suggestion. Thank you.
Thank you.
Thank you, Dr. Mutani. I would request our session chairs and Dr. Manoj to do the felicitation, please.
It is with pleasure that I now invite Dr. Nandini Verma. Dr. Nandini Verma is a distinguished principal investigator and assistant professor at Homi Bhabha National Institute, Mumbai. She has several awards to her in her kitty, which include the Koshland Fellowship, Senior Post-Doctoral Fellowship at the Weizmann Institute of Science, and the TAC PBC Indo-Israeli Fellowship. She has also been a Dean of Faculty Post-Doctoral Fellow and was honored with an ISCA Young Scientist Award, National Nominee. And she will be speaking about hyperphosphorylation state of the tyrosine kinases, which create a targetable vulnerability in the drug-tolerant triple-negative breast cancer.
Thank you. First of all, I would like to thank the organizing team for inviting me here, and thank you for the introduction. Cheers. So today I'm going to talk about not really related to the microenvironment, but something which is related to the receptors and its so the talk is related to breast cancer, and as we know that breast cancer is a very heterogeneous disease, and you can have it from the ductal region that it is originated, and also it could be from the luminal region, and as it is progressed, it can infiltrate into the ductal lumen, and this is how it actually progresses for metastasis. So apart from that, the origin of cells which can form breast tumor are also different. That could be a luminal region that originates from the lumen that secretes milk, and they are ER/PR positive, estrogen and estrogen percent positive sometimes, and also from the myoepithelial layer which actually surrounds the ductal lumen for a purpose of contractile region. And then also people try to classify these breast cancer into clinico-pathologically, and most of the clinical pathological classification relies on the presence of certain receptors, which are estrogen receptor, progesterone receptor, Ki67 scoring for the proliferation, and HER2 positivity. And then they classify them into the luminal A for type of disease, luminal B, which is HER2 negative, HER2 positive luminal B, HER2 disease, which is negative for ER and PR, and triple negative breast cancers which do not express either ER, PR or HER3, but they are actually very aggressive tumors and hard to manage in the clinic. So if we look at the prognosis of the disease, the survivorship of luminal tumors are actually quite high. As we go along, HER2 is the disease which is now better managed because of the presence of targeted therapy. And triple negative is a group of tumor which only has chemotherapy as a treatment, and these tumors they are very aggressive and they are very drug resistant, and most of the time they are metastasized to important organs like brain, lung, and liver. So this is how there are the heterogeneity in the disease. And the second thing is the triple negative breast cancers mostly frequent in South Asian countries nowadays, and overall in world that you can see that up to 20% tumors are present as a triple negative in the rest of the world, but in South Asian countries the prevalence could be up to 31%, which is high. So people try to classify this disease, triple negative breast cancer, into multiple ways, and somehow they could not fix this problem, because if you try to classify them with molecular parameters for the gene expression, they could classify into 6 to 4 classes, like basal, like mesenchymal, and androgen positive, and others, and then they classified into certain 4 subtypes, basal, like 1, 2, mesenchymal, and large. There are certain other groups which also try to do this with the help of some markers, immunohistochemical markers, and some immune system expression of the genes. But somehow, and so epigenome and metabolome also. But somehow people try to see that most of these classification actually overlap, and they could not actually make consensus out of that how to classify this disease in a better way, and how to find out the correlation between the pathological parameters and also outcome of the therapy. So the problem is there is no targeted therapy available, and when people go for chemotherapy for this triple negative breast cancer, the outcome can be either most of the people, more than 50% of the patients do not respond well to the chemotherapy, and the difference between the survival of the people that are treated with chemotherapy and cannot respond good is very huge. So people who survive better, they are the only ones who are getting better outcome with the chemotherapeutic. But if they are resistant to chemotherapy, the survivorship of the triple negative patients are really bad. So there are multiple challenges, and the most important challenges to manage the disease is that we lack molecular targets to really, first of all, that clinical functional molecular targets are not there for treatment, and if the people are resistant for chemotherapy, we do not have better options for them. So there are some class of molecules which are highly expressed in TNBCs, especially the receptor tyrosine kinases, and EGFR is one of the most expressed tyrosine kinase here, receptor tyrosine kinase, and up to 70% tumors actually express it high. And there is a genetic amplification in up to 24% of the tumors where this really high expression is there, and the prognosis of this disease is also poor because of this expression. But however, in clinic, if you try to target EGFR as a monotherapy, people are not successful, and because all the time it can give rise to resistance of the EGFR targeted therapy, and still there are combination therapy which are actually explored in this disease. So what we actually were looking at that why this is happening, and how we can find a better approach to target EGFR. And what we looked at, that whenever you have a stimulation of this receptor, we found that there's a relay of signal from not just the receptor to that, but to the cytosolic kinases also, like pocket focal adhesion kinases, and as well as there are other receptors like c-Met which are also overexpressed during this type of relay. And these are also implicated that these receptors actually crosstalk or not only with another cytosolic tyrosine kinase, but also the receptor tyrosine kinases. And this is how the signals are actually amplified in this disease. So then we looked in the expression of EGFR, and we could see that only looking at the EGFR without any tyrosine kinase which are expressed inside the cell, like focal adhesion kinases, the prognosis of the disease cannot be defined just by looking at the EGFR, and EGFR high and low cannot actually bifurcate the disease into bad or good survival. However, if we can re-look at the same thing with the co-expression of EGFR and other cytosolic kinases like PYK2, you can really divide the disease into bad and good prognosis. That means EGFR is not the only one who can determine the prognosis of the disease and the outcome of the patients. So then, because of this, we took a mathematical approach to find out how we can target this, signaling pathways and this complex of network. And we could see that if you target EGFR with gefitinib with other, tyrosine kinase of focal adhesion kinases, the outcome in the cell line is also good. And then we simulated this experiment with multiple tyrosine kinases, and we could see that dual focal adhesion kinases, PARC and PIK2, can really suppress together with gefitinib, and they behave like a switch-like behavior, where if you combine these two drugs, gefitinib and PF3-96, which is a dual inhibitor for focal adhesion kinases, the signaling of important signaling like phospho, ERK and phosphostat goes down like a switch-like behavior. So this actually can be used, this approach can be used to classify TCGA patient just as a group of disease that you can classify them into TNBC patients, and then you can find out certain signatures that can classify which patients can actually be benefited by this combination approach. By you can prioritize the patient and also find out which patient will do good. So here with this approach, with this pipeline, we found that basal like 1 and basal like 2 may be the best patient cohort that will be benefited by co-targeting of EGFR with population kinases. And then we tested this hypothesis, what we found in the mathematical modeling. And the expression of EGFR was also very high in the basal like 1 and 2 cell lines. And the combination therapy with focal adhesion kinase inhibitors was also working very well. Also, it worked very nice in mice. It could regress the tumor. And then we also found out that HER3 mediated resistance, which all the time comes up when you target EGFR, was also going down if we target EGFR. And this happened because there's a new protein expression coming up, which is NMIC, the downstream regulated gene, which is NDRG1, which actually recruits the E3 ligases to degrade this receptor, HER3. So we could also see that this is happening because of the protein-protein interactions, and this is mediated by ubiquitin ligase, and these are targeted. So this is just one part of the study where we could see that the sustained signaling of the EGFR is actually important to maintain any kind of result, that is phenotypic effect. Co-expression of two kinases together should be targeted in order to break this nexus. And mathematical modeling can help to predict and prioritize the patients for any combination therapies, at least in triple negative cancer that we have done. And combination therapies of EGFR and dual FAK inhibitors can overcome drug resistance and can provide better efficacy for the EGFR targeted drugs. So then the second part is that the bigger challenge is overcoming the drug resistance when it comes to chemotherapy. So what we can do there, so we derive some models for chemotherapy resistant triple negative breast cancers. So these models are basically drug tolerant models where the cells are actually reprogrammed. And why we are doing that? Because in triple negative breast cancers, new mutations are not found when they are resistant to the chemotherapy. So they are basically reprogrammed, and this is a heterogeneous population. So we derive subtype specific models for that, and we try to look what is happening in those cells when they are reprogrammed. So for that, we took cell lines of 4 different subtypes, and this is just a representation of one cell line. So we could see that the cells are behaving in certain phases of cytotoxicity, and then they could overcome this as a drug-tolerant persistent cells, and then they could also proliferate after some time. So we are looking at multiple time points here and trying to see what is the reprogramming happening here in terms of many other things, and how this drug-tolerant population, when they are dormant as well as proliferated, they behave in a different way, and how we can target those populations in triple negative breast cancer. So with a lot of things, we are also looking mostly on also the tyrosine kinases and their partners in these cell lines. So what we found that all these cell lines that we derived as persistent cell line, they are very aggressive and they can even metastasize, and they have in vivo growth very strong, and they are resistant to the primary chemotherapies, and they proliferate very well. Also in vivo, their proliferation rate is very high once they are resistant to the chemotherapy. So then we started to have a very simple approach that we took the slices of this resistant cell, tolerant cell line, and we tried to do just a profiling of the tyrosine signal in there. So we plotted with the phosphotyrosine, and we could see that there's a high signal in the phosphotyrosine in the lysate, and we also probed this lysate with the phosphotyrosine array, and there we found certain receptors which were actually having very high phosphotyrosine signals here, and one of the top was EGFR, phospho-EGFR signal. So, and then we also started to look at if the phosphotyrosine signal is so high, what about the downstream signaling, which actually regulates the growth in the metastasis. So we also looked there, and we found that many of the signaling like phospho-C3, phospho-ERK, and P38 was very high, and same was suggested by the RNA sequencing data that there are certain pathway of the RTKs which are highly enriched in these cells. Then we looked another way that we do the mass spec analysis of this lysates from persistent cells and their parental cells, and we could immediately see the opposite thing, which was surprising, that EGFR was one of the receptors highly suppressed in terms of total protein. And there are certain proteins like vimentin, which is EMT protein, which is highly expressed here. And then we blotted this thing, and we found out that EGFR definitely suppressed in multiple cell lines that we have derived. However, the phosphorylation of this EGFR receptor is very, very high. which is totally opposite. And we then validated in multiple cell lines and multiple chemotherapeutic agents. And then we try to look where is this receptor, why it is degrading, and we could see that parental cell has this receptor is actually on the membrane, but in the cells which are resistant, this is not on the membrane, however, in the cytosol. Then we try to look what is the relation of EGFR expression with people who had underwent treatment and the total patients in the breast cancer. So if you just look total patients in the breast cancer bifurcated by EGFR expression, you do not see something which is really significant that you can have a survival benefit of the expression, low expression of this receptor. However, high expression of EGFR actually is beneficial when people are treated with chemotherapy, which is also correlated to what we are finding. So we also check that as the tumors progress, metastatic tumors actually has less expression of EGFR. Then we also try to look in the TCGA data that if the EGFR expression is correlated to the chemotherapeutic outcome. And yes, when we just segregated the patient in terms of treated versus non-treated patients, so the recurrence-free survival in the lymph node positive patients as well as distant metastatic survival was definitely very different in these two cohorts. When the EGFR expression is low, they are not doing good. And also in some cohort of TCGA, when the people were treated with the tumors were treated with the chemotherapeutic agent like platinum agents, and they are the non-PCR people who are not getting better with the chemotherapy actually has a lower expression of EGFR. And EGFR can actually predict the outcome of the patient. And here is the ROC plotter. So then we try to see what is happening to the EGFR receptor in these resistant cell, and we could see that localization is completely altered. And here we have a perinuclear localization, which is actually with the lysosome. So here the receptor is actually degrading very fast. But now the signaling is sustaining, how it is happening. So then we try to see also the degradation pattern was there, and we could overcome the receptor, we can rescue the receptor while we are treating with the inhibitor of the lysosome, chloroquine. So we verified there in other cell lines also, and we again found the same protein NDRG1, which is associated with the degradation of the receptor. It is actually high, and NDRG1 expression is also correlated with the TNBC patient which are treated with chemotherapy, that they are doing poor if you have a high expression of this receptor. So then we look why the signaling is high, and we could see there's a switching of the RTKs outside versus inside. So now SAR kinase and lymphine kinase, which are the cytosolic kinases, actually are very high in these cells. So that means although EGFR is degrading, the phosphorylation is very high, but there's a hub which is maintained inside the cell, and you have very high SARC phosphorylation lynfin expression, which are actually behaving as a hub, and we also could co-immunoprecipitate all these together as a complex. Then we also verified all these lymphine kinases into multiple cell line, and we could see yes, they are all expressed very high in TNBC cells. So there was one paper in the PDX model that in chemotherapeutic, treated PDX model was created which was resistant to the therapy, and they also found actually proteomically the same thing that we have found. And this paper was also published in 22. So that means there is a validation of our work already. So then we also try to grow the cells and try to see what is the susceptibility of these cells with the agents like gefitinib, which are classically available and they are cheap therapies. So we did the colony formation assay as well as organoid assay in the cell lines, and we could see that if the resistant cells are actually highly susceptible to gefitinib as compared to the parental, although these are resistant to chemotherapy. And we also try to do combination therapy, asking a question whether the treatment with gefitinib can really resensitize the cells to the primary therapy, cisplatin in this case, or paclitaxel in this case, and we could see that there is a good combination if you combine back the therapies which were resistant, and now with the gefitinib they are sensitizing. Same thing we also observe in the organoid system, that we could actually see a very good response in the spheroid system in the triple negative cell lines, and the response was quite high, and this is very specific to the resistant cells as compared to parental cells. So we also checked the signaling in the combination, and we could see that whenever you combine paclitaxel with gefitinib in the drug tolerant cells, you could get rid of the signaling very nicely, that even targeting is causing low STAT3, low phosphorylation of AKT and P38. So this is how the cells are actually dying and they could not sustain. And also we checked multiple pathways, and again lymphine kinases were also suppressed in this combination therapy. Also SARC phosphorylation was suppressed. So we could really target the hub of the signaling by doing this. And the conclusion is of the study that you have this perceptor cell which having this kind of hub complex by supporting EGFR signaling in the cytosol, and if you target gipitene with paclitaxel, the cells can be desensitized to the same therapy again. So we are testing this therapy in also in the mice models, and we will be waiting. We are waiting for the results. So I will like to now acknowledge my team, who is all contributor of this work, and my collaborators, and funding by multiple agencies like DBC, SERV, and the system. And I think, thank you.
Thank you, Dr. Verma, for sticking to the time. Now we may have questions.
So your triple negative drug is sense. Did you find the sameness property of this? Because you said that they are they form organoid. So organoid and spheroids, they are different. So does they form organoids or spheroids?
These are spheroids. We also did mammosphere assays there, and they do retain certain sameness properties because there is an increase in c-MYC and other basically they become more mesenchymal in nature. Although this is not a pure stem population, but they are enrichment of the stemness vectors. At least c-MYC and others we could see that they are having.
So did you correlate this finding with the clinical data?
So we are trying to do so that we would first take it in the preclinical model that if it is successful, because it's now very easy because the drugs are available, right? So and then we'll test it into the organoid, and also to validate what is this. Already a PDX model was done in some other group. They already validated link and sarcopenia are high in the PDX model. So we might also repeat the same model soon, and because it is of a clinical interest that if we can combine this therapy post chemotherapy. Because EGFR was something that was always tried to connect with the mostly targeted therapy in combination, not with the chemotherapy.
Yeah, the problem with the PDX model is that you don't have the good immune system, but immune system plays very important role in both chemotherapy and drug therapy. So maybe organoid culture will be better prognostic. I mean, for the prognostic purpose. Yeah. Thank you.
Thank you, Dr. Verma. I think it was indeed an insightful presentation. I would like to request Dr. Tandon, Dr. BC Das, along with our session chair, to felicitate Dr. Verma, please.
Our next speaker is Dr. Sarath Chandra Arankar from the ACTEC Mumbai. He is a principal investigator and district recipient of DVT Ramalinga Swami Re-Entry Fellowship. Dr. Ankar did his PhD from the Indian Institute of Science, and then he moved to University of Paris, France, for his postdoctoral work. Doctor.
Right. So thanks for the kind introduction. Let me also thank the organizers to giving me the opportunity. Okay, I'll switch the gears and I will talk more about the tumor and stromal interactions in the tumor microenvironment. So far we have heard a lot about the cancer cells and their mutations and the drug resistance. So what I'm talking about the how the tumor stromal interactions also plays a very important role in the cancer biology. So my title says that IGF-BP is a shape the distant caps subpopulation in the tumor microenvironment. So what I'm going to talk the cancer fibrosis, which is also another important aspect you see in the many cancers. Okay, so in several cancers you see the cancer fibrosis, including the lung cancer, pancreatic cancer, and other cancers. So today I'm going to focus mostly on the lung cancer. So if you see, this is a cartoon, and then here is the histology. So here you see that these are the fibrosis part. So if you see the cartoon, that these are the tumor cells, and that the tumor cells are also surrounded by the various types of the stromal cells. So the stromal cells are a very important role in the cancer biology, in addition to the tumor cell genetic mutations. So our lab is mainly focuses on how the tumor and stromal cells, how do they influence each other, and what is the outcome. So this is our main focus. So if you know that, this is in a deconvolution analysis from the published RNA sequencing data from the TCGA. When we did the deconvolution analysis in the lung cancer, we could see that the fibrosis is very high. All right. So also there are multiple publications says that whenever you see the fibrosis, the outcome is poor. So that means the fibrosis, that cancer-associated fibroblasts are playing a very important role in the tumor biology. So see, the caps are one of the most abundant stromal cells in addition to the other cells. So if you see that in the tumor microenvironment, you see the tumor cells and the immune cells, macrophages, endothelial cells, and the cancer-associated fibroblasts. Among all these, the cancer-associated fibroblasts are one of the abundant cell type. So we try to understand the biology behind how these cancer-associated fibroblasts are forming and what is the impact on the tumor biology. All right. So one of my students joined. She's Dharapia, and she started this project, an interesting project. That when we did when we looked at the RNA sequencing data from the normal fibroblasts and cancer-associated fibroblasts derived from the lung cancer patient, and we looked at the several proteins are differentially expressed. So among them, the IGF-BP proteins are very interesting to us, and they are one of the most abundant proteins expressed in the cancer-associated fibroblasts compared to the normal fibroblasts. So with this, she started the project. She wanted to ask the question that what is the importance of these proteins expressing in these cells? What they are doing in these cells? So what is known about these proteins? The IGF-BPs, as I mentioned, they are insulin-like growth factor binding proteins. They bind to the IGF molecule and they sequester the molecule from the binding to the IGF-1 receptor. So that way they can control the IGF signaling pathway in the most of the cells. So this is what already known in the literature, that the IGBPs usually they are acting as a tumor suppressor. But when we looked at the caps are pro-tumorigenic. So what the interesting part is that why they are expressed very high in these cells. So but which is already it's an interesting that if they are expressing high, they should sequester the IGF molecule and they should reduce the tumor, but which is not happening as such. So when we looked into the published publicly available RNA sequencing, single cell RNA sequencing data, so so if you see here, these are the multiple subtype of the cells in the tumor, and if you see that everywhere the fibroblasts are expressing high amount of the BP5, IGF-BP6, and BP7. So that's already an interesting fact that which is also independently showing from the other publication, these proteins are expressed in the stromal compartment which is a cancer-associated fibroblast. So, and we validated this data by doing the real-time PCR in the NFs and CAFs, which we have in the lab. That is, in this pair, we can see that BP5, BP6, BP7, all three mRNA expressions are high. And also, we have done the Western blot analysis to see the total protein levels. And if you see here, the caps are expressing BP5, 6, 7 again, as you see in the mRNA. The interesting part, these proteins are all secret proteins. So when we took the soup, when we cultured this caps from the in cell culture, and we took the conditioned media, and when we looked at the expression levels, you can see here in the caps, the BP5, 6, 7, all three proteins are highly secreted out also, in addition to the total cellular protein. So that's an interesting aspect, that what they are doing actually. So mainly in the first part, I'm trying to focus on the cell autonomous functions. So if you look at what we did, that we assume that expression of these proteins may have a very important role in the caps. So we knocked down the BP5, 6, 7 individually in the caps, and we are trying to look at the different properties of the caps. So what the caps are known to show the different morphology in the tumor microenvironment. They are in a stellate shape. They also migrate more, and they have a contractile property, which is a myofibroblast phenotype. So this is just to show how we did the knockdown. And after the knockdown of these cells, we try to see the different phenotypes. Here, before that, we looked at the BP5, 6, 7 protein. When we knock down, and you can see the morphologically they are different. The cap morphology is changed here when we knocked down the BP5, BP7, and when we also did the full knockdown. So this gives an interesting phenotype. And also, as I mentioned previously, the caps have the more migratory properties. When we did the knockdown of these individual proteins, you can see that there is a less migration in the Boyden chamber experiment. Also, as I mentioned, that the caps, they have a myofibroblast phenotype. So when we mix with the collagen, and you can see the caps are contracting the collagen disc, and when we knock down these individual proteins, and you can see that the loss of the contraction property. So this indicates already that the caps are the BP5, 6, 7 protein expression is contributing to the cap properties. So further, what we have done, we did the RNA sequencing, and then we are trying to analyze these proteins, which pathways they are involved. So what we found interestingly, when we knocked down the IGF BP5 protein knockdown, the most of the pathways are deregulated or associated with the ECM pathways, and these ECM pathways are very important in the cap, maintaining the cap phenotype. So we have also looked at the vulcanoplat, and we also validated some of this the knockdown the deregulated mRNA expression, and these genes also are important in the ECM biology. So similarly, what we did further, we have done the series red staining after the knockdown of the BP5 protein in the caps. What we saw, the series red staining is reduced upon knockdown of the BP5 protein. So what it suggests that when we knock down the BP5 protein in the caps, the ECM biology has changed, then the less ECM production. So that's why we see the series red staining is less. So further, when we knock down the BP6, the interesting, most of these pathways are deregulated in the metabolic pathways. So it is interesting, very interesting that these are the same family protein, BP5, 6, 7, but yet they have a different functions. So the cell is very, finely regulating the cap behavior with expressing the different proteins. So when we knock down these proteins, some of these genes, we have validated that also downregulated, and we believe that the BP6 protein is controlling the metabolic phenotype of the cap. So similarly, we have looked at the BP7 protein, and this is also an interesting that the BP7 protein expression, high expression, they are regulating the interferon response or inflammatory gene signatures. So this indicates, it's also already known in the literature that the caps are highly heterogeneous, and we don't know that which proteins or which pathways are regulating this heterogeneity. So here, in short, we found that the BP5, 6, 7, these three proteins are the differential expressed in the caps, and they're controlling the different the phenotype of the caps. So later what we have checked, that so far I said that the caps express the highly these proteins, and they regulate the cap phenotype. And now what we have started looking at that, how these the expression of these proteins in the caps are controlling the tumor cell behavior. So what we did that we took the condition media from the caps, and we grown them with with the tumor cells. As you can see that when we supplied the condition media to the tumor cell from the caps, and they are growing better. But when we knock down the BP5, 6, 7 in the caps and then collected the condition media, you can see that the tumor cells are growing less. So this indicates that caps secreted proteins are also having a non-cell autonomous function. Similarly, they are also promoting the tumor cell migration and tumor cell invasion. So whenever there is a high expression of these proteins in the caps, they are regulating the non-cell autonomously the tumor cell migration, proliferation, invasion properties. So also here we have showed that they contribute to the cisplatin, the one of the first line therapies to the lung cancer treatment. So we show that these protein expression high in the caps also promoting the drug resistance in the tumor cells. So then further what we have seen that what we did the experiment, which is a very important thing, that we injected the tumor cells alone here in the mice, and tumor cells with a fibroblast control knockdown, BP5 knockdown, BP6 knockdown, BP7 knockdown, and pool knockdown. So when we did that, so these are all the co-injections here. So what we are trying to see when we knock down the proteins in the caps and how they are showing an important, how they are influencing the tumor cell proliferation or the tumor growth. And we have seen that the tumors are growing smaller when we knock down the individual proteins in the fibroblasts. So this indicates that the protein expression in the caps not only supporting the cap biology, in addition to that, they are also regulating the tumor cell growth. So further, we have done the with our collaborators' help, Dr. Kumar Prabhash, we procured this retrospective patient samples from the non-small cell lung cancer, and we looked at the staining, immunohistochemistry staining for these proteins. As you see here, the stroma. Zoom in. Okay, zoom in option is not. So never mind. The stroma here is expressing the BP5, 6, 7 proteins are high. So this indicates that the stromal expression of these proteins are regulating the stromal biology. Also, they are also controlling the tumor behavior. So now we are trying to get the more information for the multiple patients, and then we want to correlate what would be the outcome when there is a high expression of these proteins in the stroma in addition to the tumor cells. Okay, so with that, I would like to summarize that we have showed IGF-BP5, 6, 7 protein, these mRNA and proteins are highly expressed in the caps, and they are secreted, and they are trying to increase the tumor growth, migration, invasion, and proliferation properties. Also, interestingly, the chemoresistant property. And from the gene expression studies, what we understood that these three proteins, although they belong to the same family, but they have a different functions. So they are controlling the different subtypes. That is a myofibroblast like phenotype, and the metabolic caps and the inflammatory caps. So we propose that these three proteins are regulating the different subtype of the caps in the lung cancer patient subfibroblasts. So with that, I would like to thank all my students. They're tirelessly they're working in the lab, and just I'm presenting their data and taking all the credit. Also, I want to thank my collaborators, clinical collaborators, Dr. Kumar Prabhash and Dr. Subhash Yadav and Rajiv Kaushal for helping me and analyzing this the patient data. And finally, I would like to thank all my funding agencies. And with that, I want to conclude, and then I would like to take questions. Thank you. Yeah. Here.
Sir, I have a question regarding the TGF beta 1 expression. Have you checked, it has the role in CAF regulation as well?
Yes.
And have you checked any cross connection between this IGF BP6 and the TGF beta 1 expression?
So that's a very the interesting question, and we have done some experiment. With the lack of time, I did not show that. What we found a mechanistic aspect that when we treat our caps with the TGF beta, BP6-7 proteins, both of them are overexpressed. So they are induced. TGF beta is inducing these proteins. So definitely, one of the well-known, the protein which is controlling the cap biology is TGF beta. So we see that these proteins also regulated by the TGF beta.
Thank you.
If no one has any question, I will ask one question. So, we have spoken this all the time, but I first time saw the tumor, the photo of the tumor. So, in one of the tumor, I think, no, not this one, the image of the whole tumor, yes. So, in some of the tumor, it is more red. So, do you think that it has more of the inflammatory caps?
Could be that we haven't looked at this.
So this inflammatory cap and the other one, the antigen presenting cap, they're functionally also like that or only phenotypically?
So at the moment we have looked at the gene expression data only.
Gene expression.
See, no, there are no functional assays at the moment. Okay? Most of the studies showed that they did the single cell RNA sequencing from tumor, and based on the single cell RNA sequencing data, they have classified them as a different cap subtypes. But there are no as such any phenotypic assays or functional assays to differentiate these the subtypes. So that's the otherwise we could have done some functional assays. Yeah.
Thank you. Fantastic talk.
Excellent talk, Dr. Chandra. I was just out of curiosity, I was thinking. You have done the knockout studies in the CAFs and the overexpression also in CAFs. I was just wondering.
We haven't done the overexpression. They already express high.
They already express high. So, if we were to take a normal fibroblast cell line and we were to overexpress CAFs, the IGF-BP.
Yes.
So, do you think it will be more reverted to the CAF phenotype or the markers, especially the
we expect that
immunophenotypic markers?
We haven't done the overexpression because we try to do these fibroblasts are they do not undergo the transfection very easily. Only there is a way that lentivirus transductions. So that's the one challenge we face. It's not easily doable unless we have to construct a lentiviral construction. But we would expect that whenever we overexpress, this can happen. Yeah.
Okay.
Thank you.
Our next speaker is Jayanta. Okay. Yeah.
Thank you, Dr. Chandra. Yes, I would like to request the session chairs to felicitate, sir.
Our next speaker is Dr. Jayanta Bhattacharya. Dr. Bhattacharya is the assistant professor in the Center for Biomedical Engineering at IIT, Delhi. He earned his PhD degree from IICT in Hyderabad, and then he moved to US and joined in Duke University as a postdoctoral fellow. His initial interest is something called system biology because he want to connect the engineering with biology and chemistry. Is that right, Jayant? Yeah. Thank you.
Okay. Good afternoon, everyone. And first of all, I'd like to thank Dr. Prakash and especially my friend Dr. Pore for the invitation and for the kind introduction. So in the next 15-20 minutes, I'll try to convince you with the data which we have on the efficacy of an immunosomes which have developed in our lab, which can modulate the tumor microenvironment and how it can overcome the associated toxicities and how it provides the durable anti-tumor immunity. So I think we don't need to discuss this because in the past 2 days, I think you have heard lots of lectures on cancer. There are different therapeutic modalities. And I think now you are convinced that the immunotherapy is a better options to treat the cancer. So people generally use immune checkpoint inhibitors, and people have found better efficacy. But it is not true for all the cancers, because sometimes we will not get the results as expected. Like for example, in the case of this non-small cell lung cancer and melanoma, if you see the data which is mentioned here, that the efficacy is not that what we want. So it was observed that the efficacy of this immunotherapy depends on the status of the tumor infiltrating lymphocytes. What does it mean? That if you have more tumor infiltrating lymphocytes, you see a better response than if it is less. So that's why the question we have in our mind, that when Vidit has joined in our lab as a PhD scholar, so I asked him to find out a molecule which can help to activate most of the immune cells, and it can increase the infiltration of those immune cells in the tumor microenvironment. So he did the literature survey, and he found that CD40 is a molecule which is known to do that job, and he found that it also helps to reduce the tumor burden, especially for the pancreatic tumor, which is very difficult to treat, as a monotherapy as well as in combination with others. So then I asked that how did it work? So how the CD40 works in our body? So CD40 is basically a tumor necrosis factor superfamily co-stimulatory molecule, which is expressed on the cell surface of antigen presenting cells and many other cells, and sometimes in the cancer cells as well. And the ligand is CD40L, which is majorly expressed on the activated T cells, natural killer cells. And what happens when the ligand binds to its receptor? So there are several cascades of events which happens when the CD40 ligand binds to its receptor. You can see here in the slides that we'll start with the left. You can see that when the CD40 engage with the receptor on the macrophage, it releases the pro-inflammatory cytokines, which helps the proliferation of CD8 T cells. It increases the phagocytic activity of the macrophages. When in the case of neutrophil, it binds to the CD40 present on the neutrophil, it increases the cytotoxic effect and directly kill the cancer cells. In the case of dendritic cells, when it binds, like the CD40 agonist, the CD40 ligand binds to CD40 present on the dendritic cells, it releases cytokines, it increases the antigen cross-presentation and helps the proliferation of both CD8 T cells and CD4 T cells. It also helps the proliferation of B cells when the CD40 ligand engage with the CD40 present on the B cells, and increase the cytotoxic effect, kills the cancer cells by ADCC. So that means, if you see here that in this slide, that CD40 is a molecule which can activate both the innate and adaptive immunity. So my student is very happy once that he found a molecule which can take care of all the mostly different types of immune cells. It can activate, it can proliferate, it can help the infiltration. But he found that there are few problems associated with the CD40 administration. So it is summarized here. You can see that it induces hepatotoxicity. So within 24 hours of administration, it induces the hepatotoxicity, and which leads to severe liver damage. And also it is not very stable. And that's why frequent dosing of CD40 is required, which also increase the toxicity. Not only hepatotoxicity, but also increase the issue of cytokine release storm. So CRS is there. It also increases the PD-L1 expression in the cancer cells. It also has another problem, that CD40 cannot take care of the CD47 expression, which is present on the cancer cells, and which is another way it can escape the immune surveillance. So now, if you see that, because PD-L1 interaction, PD1, PD-L1 interaction, we already have heard in the yesterday. So if we see that how CD47 works, so when there is a receptor which is present on the macrophage, it binds, which is called SIRP alpha, it binds to the CD14 presence on the cancer cells, and once this binding takes place, it decreases the phagocytic activity of macrophages. This is one way to escape the immune surveillance. So now the question is how to lower the PD1 PD-L1 interaction. and also lowered the binding of CD47 with this ligand SIRP alpha. So we found the molecule which is called RRX1. You can see the structure here in this slide. So it actually binds to both SIRP alpha as well as CD47 and inhibit their binding. It also has few other roles. It also decreases the PDL1 expression by inhibiting the MYC. And also it helps the polarization of macrophage from M2 to M1. So that means RR has many roles. So which can take care of few problems associated with the CD40. So like for example, RRX can take care of these two problems, PDL1 expression as well as the immune escape by the CD47 expression on the cancer cells. But it cannot take care of the hepatotoxicity, and it cannot lower the dosing or the frequent dosing of the CD40. So now the target is to reduce the toxicity of both CD40 agonist antibody and as well as the toxicity of RRX1, because all molecule has some side effects. So that means we need to use some device, some formulation which can have the targeted delivery of both CD40 agonist antibody as well as RRX1. So I will not going through this result because you know the advantage of using the nanoparticle drug delivery system. So what we did that we use a lipid-based nanoparticle and we decorate the surface with alpha CD40, that means CD40 agonist antibody, and we encapsulated the RRX inside. And we checked the loading of both the drugs, and you can see here 1 millimolar of immunosome contains 15 microgram of the antibody and as well as 33 microgram of the small molecule drug. We did the physiochemical characterization. We found that they are spherical in nature, and we did the nanoparticle tracking analysis. Next, we did the release study. You can see the release of these two drugs at different pH, 7.4 and 6.5. And in both the pH, you can see there is very demotivating results. They don't release at all. So we are very demotivated with these results because only you can see that 20%, 30% release is there. However, when we did the uptake study, you can see here we have used two different cell lines. One is GL261, which is CD40 positive cancer cell line, and B16F10, which is a melanoma cell line, which is CD40 negative cell line. And you can see this one, there is a difference in uptake, that CD40 positive tumor has a better uptake than the CD40 negative cancer cell line. Next, we check the in vitro macrophage activation. We treated our immunosome with macrophage and we checked several cytokines, for example, IL-6, TNF alpha, and you can see in the graph that, and we compared with the alpha CD40 because alpha CD40 is, you have seen this one is known to stimulate the macrophage, it can activate the macrophage. And you can see here in these two graphs that that our immunosome is also capable to stimulate, to activate the macrophage because it also have released the IL-6 and TNF alpha into the solution. But we also check this one, that does it cause any toxicity to the macrophage, because it also interacts with the macrophage. We have found that immunosome does not impart any toxicity. However, it can impart toxicity to the GL261 cells. What we did here, we treated with the macrophage, we took the supernatant and treated with the GL261 cells. And we can see here that supernatant can kill the GL261 cells, which are treated with the immunosome. So with those results, in vitro results, we move to the animal studies, and we first check that one, how far it stays inside the body. We first leveled with the immunosome with our NIR dye, DIL, and you can see in the left side curve that the serum concentration of the DIL, and you can find that the DIL concentration is still detectable till 96 hours of administration. But interestingly, when we check the alpha CD40, which is when we have injected the immunosomes, which is not labeled with DII, but here we checked on the CD40 concentration. And you can see here, interestingly, that it increases over time. And in 96 hours, you can see the concentration, which is better than the other. Similarly, we have sacrificed the mice at different time points, and we check the distribution of the dyes at different time points. You can see here the data is given here for 24 versus 96 hours. In the tumor, 24 hours is a huge accumulation of the immunosome. Whereas generally when we inject nanoparticles, mostly you can see that they reach into the liver. And in our case, the immunosome has very low or insignificant, in some cases, that concentration in the liver. And it dies down over time. But you can still see in the tumor that there is still the concentration is detectable even after 96 hours. So next we move to the tumor regression. We established the GL261 tumor model and we injected twice with immunosome, and you can see the graph here. I will rather ask you to concentrate on the survival. You can see here that all the mice are survived which are treated with immunosome and which is not for the other monotherapy, and when we use the free drug combination. But when we use the same drug for the melanoma, which is CD40 negative tumor, the result is not that promising. So then we want to check this one, why the GL261, what is the mechanism or happened inside the tumor microenvironment? So here I want to point out that the green means those are good cells which can take care of the progression of the cancer, and red means which are helping the tumor cell proliferation. So you can see here that immunosome increase the CD8 in T cell infiltration, increase the DC infiltration, dendritic cell infiltration, increase the macrophage 1 population inside the tumor. It also increase the B cell infiltration in the tumor. And in the same time, it reduces the T regulatory cell concentration, M2 macrophage concentration, as well as the MDSC. We also check the cytokine level. We check both the pro-inflammatory and the anti-inflammatory cytokines. We check interferon gamma, IL-2. These are pro-inflammatory cytokines. It increases with the immunosome and also decreases the IL-4 and IL-10, which are anti-inflammatory cytokines. This means that immunosome can balance the Th1, Th2 cytokines ratio inside the tumor microenvironment. Next, we check the CD47 expression because I told you that the TARX is the molecule which can take care of the CD47 expression and PD-L1 expression. You can see in this data that the CD47 expression as well as the PD-L1 expression is lower in the case of immunosome. So next, the things when we were doing the tumor regression study, we saw that there is a drop in body weight for when you are treated with the CD40. As you have known this one, I already mentioned beginning that it can cause hepatotoxicity, and that's that causes the drop in their body weight. But when we use the immunosome, there is no drop in the body weight. So it means that this is an indication that it is not causing any hepatotoxicity or other toxicities which can cause the body weight drop. So generally hepatotoxicity we generally like to check. We use the ALT and AST marker, and you can see in this data that immunosome. So when we treat it with CD40 alone, it increases the expression or the concentration of ALT and AST, which is a marker of hepatotoxicity. And when we use immunosome, it drops, and which is basically the same like healthy mice. So it indicates that the immunosome can take care of the toxicity associated with the CD40 agonist antibody, which is also reflected in their H&E staining. You can see here that with the CD40, you can see the mark of this toxicity, which is reduced when we have injected the immunosome. So it not only CD40 not only increases hepatotoxicity but also increases the immunotoxicity, and that's why the IL-6 and TNF alpha concentration is increased with the CD40 administration. And we found that when we have injected immunosome, it lowered both the IL-6 concentration as well as the TNF alpha concentration. So with that, the summary of so far what we have done, that it increases the cells which are important to take care of the control the cancer cell progression, and lower those which are helping the cancer cells proliferation. And also it can take care of hepatotoxicity and immunotoxicity. So Vidit is very happy. He is asking me that, sir, can we write the paper? I asked him that what would you do with the mice? He is telling that they are still there in the animal house. I said that keep them there and just wait and watch. So we wait for 90 days, and then we found that there is no tumor, so that after 90 days. So I said that re-challenge them. We took some age-matched mice and then we injected again the tumor cells. And you can see here that the tumor with the control mice, they grew nicely. In our immunosome, which is not another treatment we are giving, these are all pre-treated mice. They have no tumor. So next we want to check why. We check the IgG titer, and we found that immunosome treated mice has better antibody. We also did the cytotoxic CTL assay, cytotoxic T lymphocyte activity. What does it mean? That we took the splenocyte from those mice which are pre-treated, and we isolated the splenocyte and treat with the cancer cells in vitro. And we found that those splenocyte can kill the cancer cells in vitro. We check the interferon gamma release, and you can see here with respect to control, the interferon gamma is more released by those splenocyte. It also has more CD8, CD4 T cells, and most importantly, I want to concentrate on these two cells, like plasma and memory B cells. Both are high in the immunosome treated mice. So it increases the plasma B cells, memory B cells, and here the effector T cells, central memory T cells. So that is why the immunosome pre-treated mice didn't grow tumor even after the re-challenge. We also check the concentration of IL-6 and IL-4 in the spleen, because these two cytokines are important for the formation of germinal center where the memory cells stays. And you can see this data that immunosome increases the concentration of IL-6 and IL-4. So next we checked the HLA staining of the splenocyte to check the spleen to find out that the germinal center. You can see that it is marked as circle. So few germinal centers are developed in the control, but whereas in the case of immunosome, there is a nice formation of germinal center. You can see on the right hand side graphs, we have calculated number of germinal centers for field of view. You can see immunosome has better or higher number of germinal centers which are developed when you have treated with the immunosome. So in summary, what we found that immunosome increase the proliferation of CD8 T cells, it increase the infiltration of B cells, dendritic cells, lower M2, TAMs, lower the expression of PDL1, FOXP3, and MDSCs. It lowered the hepatotoxicity, immunotoxicity. It increased the memory and plasma B cells, increase the T central memory, T effector memory, increase the concentration of IL-6, IL-4 in spleen, and also increase the plasma IgG concentration. With that, I'd like to thank all my lab members, my collaborators, one of the collaborators sitting here, my friend, and also the funding agency. Thank you for your time. I'd like to take any questions.
Thank you, Dr. Varia, for finishing in time. We have time for a couple of questions.
Very nice talk. I just wanted to know what is unique about that molecule that binds CD47 and receptor. Is it bivalent? The same molecule seems to bind both of them.
So it is kind of a bivalent. So when it binds, it's one side. Yeah.
How was it identified? By screening or by design?
I think by screening. But I'm not sure 100% by how it was developed. But
so that was developed in another lab? Or
yes. It was already known that. Yeah.
So very basic question. How did you differentiate between memory and plasma cells?
So by their markers.
Achha, by the marker. Okay, and CD, you told there was CD8 positive. There was CD4 positive as well?
Yes.
Thank you.
So these are the markers mentioned here.
So nice talk. I was wondering, I don't know if I missed it. You mentioned at one time point that the release was only 20-30%, but you are still able to see so many effects. So is there something else going on along with the?
Yeah. So basically, the immunosome release slowly. And in the in vitro, so we are only using like PBS, for example, to check the release. But when you are thinking about the tumor microenvironment, there are many other enzymes, right? So the release kinetics, dynamics will be different. And if it is slow, that is actually very important because that actually can reduce, because it is not accumulating or increasing the local concentration of CD40. Because as soon as if you inject, in the case of monotherapy, like it is increasing the concentration so that it can shoots up and helps the activation of all living cells, its CRS and toxicity, which can be controlled by this.
And these inflammasomes were injected IV or IP or?
We injected IP.
IP. Thank you.
I have given 2 doses of immunosome.
So thank you, Dr. Bhattacharya. Here. So I was also wondering, after the primary treatment was done, when the re-challenge assay had to be done, in that duration, you had given no dose.
No dose.
So that's
even not after the inoculation of tumor also.
Yeah. So that's a wonderful.
Yeah.
I have one suggestion, not the questions. So since you have a differential release at different pH, like in alkaline pH and acidic pH, can you modify in such a way that they will release more in acidic pH? Because in the tumor microenvironment, that due to the Warburg effect, the pH is around 4.7 to 5.6. So if you can increase the release of this drug in low pH, that will redox dependent, then the normal tissue, they will not be affected. Only the affected tissue will be affected.
True, but actually, if you see that endosomal pH.
Yeah, but
endosomal pH is also very low.
Yeah.
So the thing is, why you want this molecule? So how it works? So we don't want to use any chemo drugs.
Yeah.
We want to use only those drugs, even they will reach whatever in the body. It will interact with the immune cells, which have the expression of this receptor. When they will bind, they will do their job. So it will not impart any toxicity.
No, but in the tumor microenvironment, infiltrating lymphocytes will be more affected, and they need more.
True.
Yeah.
True. So you are right that if we can do this one, they will maybe release. But even if we don't do that, so we also you have seen this.
Maybe you will get better efficacy with this redox dependent delivery system.
Yeah, maybe. Yeah.
Okay. Thank you.
Thank you. Thanks for the suggestion.
Because the tumor microenvironment has 3 to 4 pH will be there.
So first of all, tumor microenvironment does not have 3 to 4 pH because the pH is almost close to neutral pH.
No. Yeah.
So it is. So generally, it is observed that close to 6.5. So it is not like that so much acidic to the tumor microenvironment. So it's very close to not that much acidic, like 4 or.
Okay.
Yeah.
Yes.
Any other question?
Thank you, Dr. Bhattacharya. I would like to request the session chairs to do the felicitation, please.
I would request Professor Dr. Pore to please join. So we come to the last talk of the session. I request Dr. Avinash Bajaj. Oh, I'm sorry. So So Dr. Avinash Bajaj, coming from RCB Faridabad, he is professor with extensive expertise in biomedical research and translational sciences. He earned his PhD from Indian Institute of Science in 2008 and has pursued postdoctoral research at the University of Massachusetts, USA. And Dr. Bajaj's work is focusing on cutting-edge advancements in nanotechnology, drug delivery systems, biomaterials for therapeutic applications. And today he will be talking on targeting of triangular neuron cancer cell immune cell crosstalk as a potential cancer immunotherapy strategy. The floor is yours, Dr. Avinash.
Thank you, madam, for your kind introduction. So today I will be talking about summary of the work that we have done in the last 10 years on engineering nanomycels, how they can target the tumor microenvironment and activate the T-cell immunity. So in my group at Regional Center for Biotechnology, Faridabad, we are doing is we are working on 3 different programs on advanced healthcare materials. We are making different kind of hydrogel implants, nanoparticles for drug delivery. And in the second program, we are exploring these biomaterials for the cancer immunochemotherapy applications. And in the third program, we are trying to understand the impact of the lipid metabolism on the tumor microenvironment. So as I mentioned, the pipeline, especially in the field of the cancer therapy, we start our journey from the chemistry, where we synthesize different kind of building blocks and use those building blocks to engineer the nanomycels or the implants, and that we explore these biomaterials for different our tumor models. 3 models what we work is on breast cancer, colon cancer, and the liver cancer. And we then try to understand how these biomaterials are altering the tumor microenvironment, how they are changing the lipid metabolism, and what is their clinical correlations in the clinical samples. So as already in the last few talks, we have already understood that what are the different kind of the hallmarks, and we have been keep on adding different hallmarks in every 10 years. And in the recent report, you can see there are more and more hallmarks are added in after our understanding about the cancer tumor microenvironment. So question what we address in our group is that how does the host immune system can be altered to target the complex tumor microenvironment? What are the different delivery approaches which can be used where we can limit the toxicity and enhance the efficacy of the drugs, and how the multiple cancer promoting processes in the tumor microenvironment can be targeted, and how can we find the new metabolic targets for the future cancer therapy. And in our group, we have been working for the last 15 years on this scaffold, which is the bile acid scaffold, and you know these molecules are already in our body. So we use these molecules, we derive these molecules to make different kind of micelles or hydrogel implants, and these molecules have the facial amphiphilic character. You have the hydrophobic backbone as well as the hydrophilic groups. So what we do, we use the hydrogel implants, we make the nanomycels, we make the phospholipid drug conjugates, and now doing the antibody drug conjugates to target the tumor microenvironment, and then finding the changes in the lipid metabolism. So what we have done is we synthesize a library of the molecules and screen those molecules, and one of the molecules which we found is, I have shown the structure here, it is based on the lithochromic acid backbone. And the good property of this molecule is that it's self-assembled to make the hydrogel. And when you put this hydrogel in the mice, you can see here that it can stay up to 20 days in the mice. And when you add a fluorescence dye, and you can see in the NIR gel dye that this gel can maintain a sustained release of the drug at the injector site. Whereas if you inject the dye without the hydrogel, it will go all over the body. So this hydrogel can be used for the localized therapy. So what we have done, this data was published years back, that we use a combination of the drugs, 3 combinations targeting the proliferation, angiogenesis, as well as the inflammation. So these 3 drugs we have shown. I'm just summarizing here that if you use all the 3 drugs in a gel, a tri-gel, this is much more effective than the combination of these 3 drugs, either you use through intravenous route or at the tumor site. And we went ahead and studied what are the changes happening in the lipid metabolism, and this data, if anybody interested, can go through this paper. And now if you see about the triple negative breast cancer, as mentioned in earlier presentation also, is very difficult to treat because we don't have any targeted therapy available. And only options we get is that combination of the chemotherapy drugs, and these are the combination which is used, called doxorubicin and docetaxel, or taxanes and the carboplatin. So what we did is that we used that same hydrogel system which I showed you, that, and we use the combination of the docetaxel and the carboplatin. And if you can see here in the triple negative breast cancer model, the combination of both the drugs in the gel is much more effective if you give both the drugs either through intravenous route or the tumor site. And even when these drugs are given individually, obviously your combination therapy will be much better than the individual drug treatments. So now, what we want to see that what happened when we give these drugs, whether this combination of these drugs are causing any change in the tumor microenvironment. And very interestingly, when we see that combination of both the drugs is causing the decrease in the MDSs, both MDSs, monocytic and granulocytic, it changes the macrophage polarization from M2 to M1, so M1 by M2 ratio is increasing. And when we see the CD8 cells, you can see the TH1 population is increasing, TH2 population is decreasing, T regulatory cells are going down, and there is an increase of the CD8 by T regulation. And to see whether this combination effect is only the apoptotic effect or there is a immunogenic cell death and there is a cytotoxic T cell also causes the effect. And when you give the anti-CD4 and anti-CD8 antibody, you can see that there is an increase of the tumor growth. So it means the combination effect is causing the apoptotic cell death as well as also doing the immunogenic cell death, where the CD8 cells are responsible for tumor aggression. So now how you will prove this? So what we did an interesting experiment where we did on the left side of the mice, we injected 41 cancer cells, which were the primary tumors, and on the right side also we injected the 41 tumors. But we have done only the localized treatment on the primary tumor on the left side. Now you can see here in the tumor growth kinetics, you are seeing the decrease in the tumor volume, but you are also seeing the decrease in the tumor volume on the right side, right, where the drug was only on the left side. So it clearly shows that it might be the T cell activation which is going to the secondary tumor and clearing that. Now you can say that it can be the drugs which are getting released from the hydrogel which are going to the secondary tumor. How you will prove that? So you can inject the 41 tumor on the left side and CD26, which is a different cell line, on the secondary tumor. So because those T cells which are getting activated will be very specific for the 41 tumor only, so you are not seeing any regression in the CD26 tumors, right? Because the T cell activation is there. So it clearly proves that your drugs are not getting released from the primary side and going to the secondary side, it is a T cell. So we quantified the T cell activation both in the primary tumor and the secondary tumors, and you can see that granzyme CD8 positive cells are also present in the secondary tumors as well as in the primary tumors. So then we see that, okay, we have been working as in the earlier paper we have shown that the combination of the chemotherapy drugs is altering the sphingolipid and the granulocyte metabolism. And these lipid metabolites are already known to have a role in proliferation, apoptosis, and inflammation, angiogenesis, and immune cell activation. So I am not going to go into the detail, but what we have shown that the combination of these therapies basically altering the sphingolipid metabolites in the cancer cells, and these sphingolipid metabolites are causing the ER stress and activating the UTR pathway, which allows the antigens and the DAM molecule to get released, and these basically activates your T cells, and that is responsible for the tumor aggression. And this was published last year in the Science Advance. So what we are doing now, we are exploring these hydrogen implants in combination with immunoadjuvants, also in combination with the diet restricted feeding, as well as with other metabolic inhibitors that can we reduce the chemotherapeutic dose, so that because they are toxic drugs, so can we go to a simple metabolites and target the same tumor microenvironment in a different way. So I will just briefly go to another story which was on the nanomicelles. So as we know about the drugs, right, they are very toxic. Most of the chemotherapy drugs are very toxic. So what we have done is a story which we have started 2010, right? We engineered a small molecules, right, where the docetaxel was conjugated to a phospholipid based bilateral backbone. And what this lipid does is that it can self-assemble to make the sub 100 nanometer nanomicelles. And you can see that when we did the tumor growth studies, right, you can see here the control where the untreated mice having the tumor, where the DTX-TS, which is a docetaxel drug formulation which is given to the patient, has shown some regression, whereas these nanomicelles are showing a very good tumor regression. So then we have done the toxicology studies and compared with the commercial formulations, and we have shown that our formulation is not toxic at all in the monkeys as well. We have done in the mice, rat, rabbits, and then in the monkeys. So this was also published in 2021. So then what we have done, as we know the tumor microenvironment, this is only, you need not to target only the proliferation, but whole tumor tissues, the inflammation, angiogenesis. So what we have done, we use the same system to engineer different kind of nanoparticles, where we have used a combination of the chemotherapy as well as the anti-cancer drugs as well as the anti-inflammatory drugs to engineer the chimeric nanomicelles and show that these chimeric nanomicelles targeting the docetaxel and dexamethasone is much more effective than the single drug formulations. Now we see that these formulations are also altering the tumor microenvironment, where we can see easily that there is this increase of the infiltration of the CD8 cells. So I will not go into the mechanism of this part. These results are also published. Now what I will do, this was published. So then what we have done here, we have earlier studies, we have done the inflammation and the proliferations. And then what we did, we used a similar approach to target the angiogenesis and the proliferations and to make the chimeric nanomicelles having the two drugs. So I will not go into this story as well because this is the published data. So what I will now show you is the final story that to target a tumor microenvironment, you need all the combination of drugs. So ideal combination is to target the proliferation, inflammation, as well as the angiogenesis. So can we engineer a nanoparticles having all the three drugs and which can release the drug in a controlled way? So we have synthesized these three molecules with the anti-cancer drug, anti-inflammatory drug, and the anti-angiogenic drug. So we make all the 7 different kind of nanomicelles having single drug, combination of 2 drugs, or combination of the 3 drugs. And then this is the whole nanoparticle characterization. And then you can see here, very interesting data, the combination of the 2 drugs. Here, any combination of 2 drugs, if you see, is much better than the single drug. And then finally, we compare also the 3 drug combination, chimeric nanomicelles, as compared to the 2 drug combination. And you can see that the chimeric 3 drug TDC nanomicelles are completely regressing. The tumor does not allow the tumors to grow, and they are more effective than the dual drug nanomicelles. So we then try to understand what happens to the tumor microenvironment, and you can see here clearly there is a M1 macrophages increase, there is an increase of the CD8 cells, there is an increase of the granzyme B positive CD8 cells. And then we also did the orthotopic model where we injected the cancer cells in the cecum of the mice to make the colon cancer model. And you can see here on the treatment also, these mice became normal. Their belly size has come to the normal, whereas it was increased in the untreated mice. And here you can see here, this was the untreated tumors where the cancer cells were injected in the cecum, and on the treatment you see that there is a clear tumor inhibition which you don't see on treatment with the TDC. And you can see also in the spleen also, the spleen size has become the normal, its weight has become normal, colon length has become normal. Then what we did, we injected the cancer cell in the spleen, which will allow the cancer cell to migrate to the liver to make the metastasis. So you can see here on the treatment, this is Mac and Windows, right? You can see here the spleen size has gone down, and the number of the metastatic nodules has drastically reduced on the lesion. So these nanomycels can target the orthotropic tumor as well as can inhibit the metastasis. So then we have gone through the whole mechanism part, which I am not showing. So in summary, we have engineered different kind of nanomycels which can target the tumor microenvironment by either by inflammation, angiogenesis, or the product. With that, I thank all my students as well as collaborators as well as the funding agencies who have been supporting our lab in the last 9 years. Thank you.
Thank you, Dr. Bajaj. Any questions from the audience?
Hello. Thank you, sir, for your talk. I'd like to ask this question. You talked about your hydrogels and them as a nano delivery system. So my question is, recently we've also learned about how IIT Guwahati has also come up with hydrogels in TNBC using the similar mechanisms. So how is your study going to stand out in comparison to that one?
I don't know IIT Guwahati story, sorry.
It was very famous, it was in the media, so that is why I put it.
Yeah, I didn't know that. Yeah. But I don't know, but what we are now trying to do in future is that our focus is to how we can target the metabolic pathways. I don't know what they have targeted. We have been doing on the chemotherapy. Now we are moving away to the metabolism, how we can target the metabolism in the cancer cell and the immune cells and that. So it's very difficult to compare two different, it's such a complex problem, right? So everybody can tackle it from different angles.
So from what I studied, their paper that they had published, they had used doxorubicin in the similar manner from what I understood. They also conjugated it with a hydrogel and then they intravenously injected it to the tumor sites and they saw good results, which I am not so sure about. So that is why I put it.
So what I will say is you read that paper and this paper and then you can compare.
All right, thanks.
Rather than me commenting.
So, Dr. Bajaj, I was just wondering. I mean, I had a very similar question. In the first story that you had showed, where a gel and 2 other drugs were combined, what do you think? What could be the reason that they are really changing the cell population in the tumor?
Yes.
Because monotherapy, I don't think if you use the gel alone or the drugs alone, yeah. So they are not going to do that. But just the combination of these 3. And what part the gel has to play? Is it just the delivery system?
That gel part is only to maintain the sustained
Yeah.
for 20 days. Then we are doing the experiment.
Absolutely. Yeah.
Without gel, right? Then we don't see that.
Exactly.
So that is very important. So another story which we are building up, there also we have found that when you treat in vitro, for example, the cancer cell for 48 hours, you will not see that effect. When we are seeing the same treatment, not with the cancer cell, the sum of the molecule that we are testing, you have to keep on doing for it to sustain the rule. For example, one of the receptors that we are targeting in the macrophage is a good amount of receptor. T cell very low expression. So to target the T cells, you need to have it sustain the release of the drug to inhibit that receptor. So that's why the gel is important that.
Yeah. So
gel is not doing anything itself.
Yeah, so the sustained release of the drug combination, you feel that that is triggering. Thank you.
Thank you, Dr. Avinash. I would request the session chairs to felicitate Dr. Avinash, please.
No.
I would also request Dr. Archana Sharma to felicitate our session chairs for the day, Dr. Gauri Shankar and Dr. Sonia. I'm a faculty. Thank you all. We are breaking for the tea and we meet here after 10 minutes. Thank you. Please be on time. 15 Good evening, everyone. We now begin with the last session for the day, which is cancer targeted therapies. The session will be chaired by Dr. Manoj Garg and Dr. Shinjini Das Gupta. I now hand over the session to the session chairs.
Good afternoon, everyone. I hope you all are refreshed after the tea. So, first speaker is Dr. Alok C. Bharti. He is from Delhi University. Alok C. Bharti is a professor at Molecular Oncology Laboratory with a strong academic background and expertise in cancer research. He received his bachelor degree in zoology from Delhi University, followed by MSc in zoology. Dr. Bharti has completed his PhD from BHU. He also has a huge research experience as a postdoc fellow at MD Anderson, Houston, USA. His focus is on molecular oncology, contributing understanding and treatment of cancer through innovative approaches. I welcome Dr. Alok Bharti for his talk. Thank you.
Okay. Thank you very much. Thanks for the chair. And it is always a pleasure to come to this group. And I'm extremely happy that Professor Hridesh Prakash has organized this wonderful event with all the stars of this global cancer conference coming from international arena, and presenting a talk on this platform makes me feel quite elated and very happy. And here, since it is talking more of immunology, I will be taking a little bit of deviation, because the work that we are presenting is, of course, entirely targeted to the immunity, but it is perceived as a very separate domain where we were working on phytochemicals, and we are trying to find out phytochemicals that can target cancer of uterine cervix. And I'll take you through the journey where how we have worked with these phytochemicals. They are very, if you actually look into the development of medicine, whether it's a modern medicine or alternate medicine, phytochemicals have been at the fulcrum of all these developments. And if you look into the modern medicine, they usually talk about pure compounds, the analogs, and the mechanism of action is pretty well known. When we talk about the alternate medicine, they are not bothered about what is the mechanism of action. That leads to efficacy-based tracking of the phytochemicals which are present in plants, whether it is in a crude extract or some formulations. So most of the time, these alternate therapies
are backbenchers because the mechanism of action is relatively unknown. But still, they are the treasure trove because they are the guiding force for developing new therapeutics. So in our lab for over 20 years, rather over 20 years now, I have been working with my group, first as with my mentors, and then my own group. We have been working on phytochemicals and anti-cancer therapeutics. There are different categories of phytochemicals, you can say, or herbal derivatives. There could be pure compounds like you can see curcumin, berberine, trans-chalcone, piperine, nimbolete. So these are all very well characterized structures known, and they show these are the outcomes. I'm not saying that how many we have seen, but these are some of the outcomes. We have also looked into these compounds as extract or the herbal derivatives as extracts like Baracalam pinnata, Phylum amblica, and sometimes we have also worked now with their preparations in alternate medicine like Berberis aquifolium, mother tincture, and Berberis vulgaris. These are preparations, exactly same extracts, but they are commercialized under homeopathic arm. And then there were a lot of mixtures also, or formulations like Praneem and Basant, which also went into the clinical trial. So these were some of the 4 areas our lab was working. But one of the biggest problem with these researching with these phytochemicals and herbal derivatives is the economics. So they are usually cheap, or I'll say they are cost effective, but they are less lucrative to the medicinal economy of the medicine. So that is why most of the time these are backbenchers. And we had actually suffered a lot because there were a lot of formulations that we developed which were patented, which were commercialized, but then there was no taker. So basically, whatever information that you generate in scientific world, that becomes sort of, the junk. So to mitigate that, I think my lab took another innovative approach that all these phytochemicals, they are in fact part of the medicines which are routinely prescribed under alternate medicine. And if you look into Ayurveda and Siddha, the kind of formulations nowadays, I think they are improving, but 10 years back, they were not that kind of, QC in these kind of drugs. On the other hand, homeopathy had quite very stringent QC available, and the commercial preparations are quite homogeneous and well characterized. So we looked into the phytochemicals that are actually very effective. We went back and checked that whether homeopathy uses those drugs which have those phytochemicals, and we ultimately came up that there are 2 drugs, Berberis aquifolium, of course, as homeopathy goes, there are mother tinctures, then there are potencies. So those things are there, and we have completed already preclinical phase 1 trial. It is not phase 1 clinical trial, so we actually found 2 of the drugs which are effective. And now the Council for Research in Homeopathy, they have already approved a phase 2 animal clinical trial for that. So that is another story. I'm not going to share about that story, but it's another very important thing that we are looking into the herbal derivatives, and we want to look into their mechanism of action, mediate or based screening of drugs and their efficacies. So here we have target-based approach, and we have used 3 different approaches: E6 primarily for the cervical cancer, and SOX2 and OCT4, which I am going to share the results today. We have in between also looked into the angiogenesis. So we had screened for VGF binders. So these are the 3 main domains which I'm going to discuss today. So there are a number of papers. It will be a sort of summary of those papers. I'm not going into one by one, but I'll share some data. So the first approach is we targeted E6. E6 has a lot of domains which are associated with apoptosis, transcription, and there are specific residues which are very well characterized. The molecule is already crystallized. So we have a crystal structure available for HPV F3-16E6. Okay, so these are the two structures if you see on the bottom. So these two structures are available on PDB, and we try to screen different molecules which are prepared or present in the plant preparations which we use for homeopathy. But again, I'll not go into detail. So we found that there are a number of compounds which actually have very direct binding with E6. And not only that, so these particular preparations of homeopathic origin, they are also showing effects on cervical cancer cell cycle, and then they are also working on different signaling molecules. PSTAT3 is one of the key molecule, JAK-STAT signaling, and then we have AP1 signaling is compromised and whatnot. I think if we look into that concern, so most of these signaling molecules which are associated with carcinogenesis were affected. So there was second approach, which was for the angiogenesis. We look for VGFR2 based targeting, and in this molecule there was a little difficulty because it's a very large molecule. You cannot work with a single molecule together. So we had actually performed molecular modeling for the intracellular domain as well as for the kinase domain. So there is a extracellular domain and kinase domain, both were separately analyzed, and there are over 100 phytochemicals have been already, we performed extensive literature search. We found that there are over 100 molecules which have all been published, which have some anti-angiogenic activities, but they are not known whether they are going to affect or target VGFR. So we actually performed a docking experiment of these phytochemicals with the extracellular domain as well as the other one, the kinase domain, and we have found that there are certain molecules like sanguinarin, cholesterol, vidafrin, piperine. So they all have very high binding energy, and this binding energy is much it is not crossing that the binding efficiency of VGF. So this VGF is a actual ligand with which VGFR is bind. So if the binding energy is is less for VGF, that means it is not these whatever inhibitors that we are going to use, they are not going to be competing with VGF. So these molecules, in fact, are not preventing the interaction of VGF with VGFR. So next we looked into the intracellular domain, which is a kinase domain, and we tried to dock, and certainly there was a lot of docking. But if you see it, the ATP is a major molecule because these VGFR undergoes phosphorylation. So the binding energy to bind with ATP is minus 6.4. But there are a lot of ligands we actually found that they were interacting with this kinase domain, and that shows that they can displace ATP. So that means they are very strong inhibitors for the intracellular signaling domain, and they are and the downstream signaling by VGFR. So there was a list of around 30 such molecules that we explored, and we performed a screening. There are a lot of other analysis that we performed. We performed Lipinski rule analysis, in which we have found that only limited number of them can actually cross cell membrane. So that means by in silico experiments, we can tell that they may, since even if they are actually targeting the intracellular domain, but they are not able to cross it. So we had a filtering, and then we found three important molecules, trans-chalcone, piperine, and nimbulide. So they actually can directly inhibit the neoangiogenesis, and this neoangiogenesis was studied in a very InGen model where we have performed a chorioallantoic membrane of the chick. So chicks were actually the fertilized eggs were placed in the incubator at say day 8, and you can apply these drugs on the developing vascular system, and you can actually measure how quickly this drug is actually affecting the formation of blood vessels, and you can capture it by the imaging of that particular egg. You can create a window over it. Not only that these drugs were effective directly on angiogenesis, we also developed a xenograft model right on the eggs, where we have placed one of the cell line U87, which is a neuroblastoma cell line. So if you place this, the neuroblastoma grows as a nodule, and with this nodule, there is a lot of angiogenic activity that takes place. So we can place the drugs, and you can see on this particular line how effectively the drugs are preventing the angiogenic process. And not only that, there is a direct effect on VGF, VGFR signaling, and VGF itself, transcript, because VGF transcripts are also under control of VGF, VGFR signaling. So that transcripts were also getting lost to some extent, although it was not a complete loss. And we can actually, because of the transcripts are coming from mouse or from chick and from the human origin, because human one is nothing else but the cell lines that we have placed. So we can actually, based on the real-time quantitative PCR or RT-PCR, we can find out which transcripts we are talking about. And we have found that if you look into this, the biprane and there is a nimbolide. So these are, if you see there are asterisks are there, so they can specifically inhibit the angiogenic process. So in the third, which I am going to talk in a little more detail, and this particular results are still unpublished. So the third very important and recent aspect that we are working using phytochemical is the OCT4 and SOX2 signaling, and these two transcription factors play a very important role in pluripotency as well as introducing the stemness in cancer stem cells. And these two transcription factors are essentially required for maintaining the, for tumor recurrence and chemoresistance also. So there are a lot of molecules which have been shown to target cancer stem cells in different systems, as well as there are two molecules which have also been, 2 or 3 molecules which have been shown to target cervical cancer stem cells. So we actually try to see whether these the anti-stem cell activity is entirely due to their action on OCT4 and SOX2, or this is maybe something unrelated. So we pull down these chemical structures, perform binding or binding studies. First of all, we actually looked into their cell membrane permeability, because this is very important. Even though if they are going to work on transcription factors, but if they are not able to cross the membrane, that is not going to help. So based on their character, chemical character, and the the bonds, you can actually see, and their hydrophobicity, how well they are going to cross. So this Lipinski's rule of 5 basically tells how effective a molecule could be to cross the cell membrane. So based on that, there are a number of molecules which were violating it, and there were molecules which were very effective in crossing the the cell membrane barrier. And second, another very important thing, if these phytochemicals have to be actual drugs, so then they should not be hepatotoxic. So this is one of the very important yardstick in which the phytochemicals, if they are hepatotoxic, so they are also not good. And we can actually, based on their chemical structure, we can find out whether they are hepatotoxic or not. So this is ADMET. There are 4 or 5 different kind of tools, and we have already worked with 4 different tools and found that ADMET tool is one of the very precise tool, because I'll not go into detail of that. Then PKSM web server we use, and we found that 3-4 very important deviators which are quite hepatotoxic. On the other hand, there are 4 important molecules which came up: Morrison, soralidin, sanguinarin, and phenylethyl isocyanate. So these are the 4 molecules which were showing no hepatotoxicity. At the same time, they were also cell permeable. So we took it forward and we looked into their IC50, which was already documented. Not only that, we also performed their docking on the SOX2 and OCT4. The biggest problem when we actually pull out this SOX2 and OCT4 from the database, so the crystal structures, there are around 20-25 crystal structures for each are available, but we don't have a human SOX2 and OCT4 still available and crystallized. So using the information of the peptide sequence, we have to generate a model of these 2 molecules, and this model and then it has to be compared with the available database. For that, we have used Swiss modeling, and this using this Swiss modeling, we have looked for and we have refined this galaxy observer. Tanya is with us, so she did all this analysis, and we got a model which has the least RMSD, and then there is another score called Roma favor score. So based on these two parameters, the most favored model number 4 was selected in both, and then we performed the docking. And using this docking scores, we have now found that all these 4 molecules are quite very well docked, and then they have a very nice binding energy. Particularly sanguinarin has a binding efficiency which is much greater than the standard drug, which is zoledronic acid, so which is -4. So you can see that all these molecules have binding efficiency or binding energy is much lesser than that. So that means these molecules form a very stable interaction with this OCT4 and SOX. So we performed, first of all, before we look the effect of these molecules on the tumor spheroids, which are indicators of stemness. So we performed the IC50 values in our experiments also, and we found that sanguinarin was one of the most effective molecules with the IC50 of 1.3, and then thereafter the Morrison and Soralidin, they had also IC50 of 3.5. Although PIETC that did not have very, in our experiments, particularly in our cell lines, that was not very effective. So we started with sanguinarin. It was quite interesting. We got tumor spheres, and it was very effective. That immediately, if you add even 1 micromolar of sanguinarin, so all the tumor spheres are gone. But at the same time, Tanya looked into a very important advisory that sanguinarin is prohibited and banned for clinical use. So we decided that even though it's a very potent molecule, since it is not going to be used clinically, so we abandoned it almost all the studies, and then we moved to another molecule called Morrison. And as I told you, Morrison has one report, and it is a very, of course, a very benchmark report which tells for the first time that Morrison is having anti-cancer stem cell activity in cervical cancer, but the data was very limited, in which they have seen or some of the assays where cell migration they have studied. They have also looked for some of the staining with crystal violet, and they have looked for the cell signaling markers or apoptotic markers. So it was quite not very strongly supporting, or not strong evidence was presented in which the Morrison is anti-cervical cancer stem cell activity. So we actually took more detailed investigation, because we have already shown that it has a very capability to cross the cell membrane, bind to OCT4 and SOX2. So in that case, we started working with different concentrations of Morrison, particularly 3 micromolar, which was the IC50. We have gone half of this IC50 and double of IC50. You can see that by if we increase by 3 micromolar to 6 micromolar, all the tumor spheres are gone. So not only the number, the size of tumor spheres goes down, and the Morrison also affects on stemness and associated transcription factor. This is a crystal violet staining or regenerative potential, where we take out the cells, replate them on agarose, and we look into the colony formation. So those cells which were treated with Morrison, when they're in 3D cultures, they are not capable of introducing or initiating the colonies or tumor spheres further. And we also looked into the effect on OCT4 and SOX2. So in all the 3 cell lines, this OCT4 and SOX2 were strongly inhibited. So this is a clear indication that the SOX2 and OCT4 are inhibited. It is not only that their transcripts are gone, so they are also inhibited. Their level in the protein is also low. So same way, we also continued further looking into this soralidin, and we actually made another interesting, co-culture system. We wanted to see exactly how these Morrison is affecting the polarization of the macrophages. These macrophages are one very important thing Tanya actually observed, that if you grow the cultures in 2D, and when you culture the cells in 3D, and start putting a co-culture with the, one of the monocytic cell line, which when you actually treat with PMA, it becomes macrophage. So it's an M naught. So if you co-culture them, so if you put it 2D, so most of the markers which are associated with M1, they are increased. But if you actually put them with 3D cells, means if you actually make tumor spheres and take out those cells and then replate them along with in this co-culture system, there is a very striking shift from M1 to M2 kind of behavior. So this most of the M2 behavior, M2 markers are high in those kind of macrophages which are developed in the co-culture. So that means the tumor spheres are directing the differentiation of monocytes to macrophages of M2 type. And if you treat these cells with Morrison, so again we actually start seeing that if you see here, this is IL-12, TNF alpha, and INOS. So most of these markers which are actually representing M1 phenotype, they come up. On the other hand, the markers which are associated with M2 type, they go down. So that means if you are treating them, these 3D cultures, their capability to convert or make M2 type macrophages go down. So Ralladin study is still under progress, and we have also found a similar kind of pattern. Of course, we have not gone further in this, but clonogenic assays, etc., it's kind of similar kind of effect is seen. So that means we have two very important phytochemicals which are which can cross the cell membrane and affect SOX2 and OX4. So although our lab is working in quite a different directions, but one of the major directions that we are actually tracking is the phytochemicals, and currently we are involved in targeting cancer stem cells, and certainly, as you have seen, it's a treasure trove for future anti-cancer therapies. So with this, I will acknowledge a lot of funding from ICMR, CCRH, DST, and University of Delhi, and there are a lot of clinical collaborators, may not be in exactly in this part, but certainly they have been contributing to the lab. And these are my current lab members. You can see Tanya, Divya, Apurva, everybody actually participates and contribute to this study. Thank you very much for your patience.
Thank you very much for this fantastic talk. Now the talk is open for the question. Question, please.
Thank you, sir. I think, sir, your future perspective is cancer stem cells you're targeting. So currently, did you check the stemness?
Yeah, stemness. So this clonogenic assay is for the stemness, the ability of the cells in. I think this is an assay in which we have to incubate the cells in agarose medium, and then to see whether they are forming colonies or not. Whereas if you see the treated cells, they are not able to form the colonies. On the other hand, the cells which are not treated, they have the potential to regenerate. So regeneration potential of these tumor sphere cells is an indication of stemness property.
Okay. And sir, you have also checked the SP effect of these on SP. I think
this was my great wish.
Yes, I love
and all my students tried. In fact, for doing SP, we need to have a UV or 407 laser. Our flow cytometer does not have it at the department.
I'll help.
We have got a great, very expensive system at USIC, but there is a big line. So you have
I
to
asked
stand.
Tanya, I will help.
Yeah. Right.
Definitely.
Thank you very much.
See, here you got the help also.
Any other question?
Yeah.
Hello, Dr. Bharti. Very nice talk. So I have some very basic questions. So first thing, in a tumor environment or a tumor, there are hypoxic regions, right? So do you think that if alters the efficacy of these phytocompounds? And second question is a little bit different. I'll ask it together. So I see all the stemness markers and other properties similar in both the HPV negative and positive cell lines. So does these chemicals work similarly for both these, or they are independent of any HPV infection as such?
Yeah, I think because we are talking about stemness. Although stemness is also regulated by HPV, that's what we had shown in our clinical cancer research paper.
where E6 is overexpressed and E6 somehow regulates the stemness. But here, when we are talking about, of course, there could be other regions where this molecule might be working. But if it is working on stemness or stemness associated transcription factor, that means we have some solution for tumor relapse and chemoresistance. It may be assisting as a general anti-cancer agent. I'm not saying that it will not inhibit other pro-carcinogenic signaling, but it is also going to curtail these stemness associated markers. And second, if we talk about the tumor heterogeneity, all molecules, whatever we try, they cannot work homogeneously. Certainly, there will be some molecules which, and we cannot generically say that they will not be effective on hypoxic. The only thing is that we have to screen and find out those molecules which differentially work better on the hypoxic cells. And perhaps we might have the answer by these molecules which are working on cancer stem cells, because these are the cells which really poorly respond to most of the standard chemotherapeutic drugs. So if we merge these therapies along with standard chemotherapeutic drugs, we might have an answer or a solution to tumor relapse or chemoresistance. Any other question? If not, I really thank you all for patiently listening. Have a great day.
So,
Thank you so much, sir. Now I would like to call upon stage Dr. Manoj, Dr. Shinchini, and Dr. Shilpi to felicitate sir. Please give them a big round of applause.
iPhone charger तो है लेकिन यहाँ नहीं मंगाना पड़ेगा आपको चार्ज करना है
So for the next talk, we now have Dr. Bharatwaj. Dr. Bharatwaj did her bachelor's in chemistry and master's in biochemistry, University of Calcutta. She has done PhD from CSIR Indian Institute of Chemical Biology, Calcutta. Dr. Maushmi Bharatwaj is now currently working in, she's a scientist G and expertise in molecular biology and pathology. She has completed her postdoctoral fellowship at Chicago Medical School at University of New Mexico, USA. She has served as a research assistant professor in the Department of Pathology at the University of New Mexico before joining ICMR and ICPR as a senior scientist. She has led multiple extramural projects, several publications. She is a fellow of National Academy of Science in Chabad and also a fellow of West Bengal Academy of Science and Technology. With these words, I welcome Dr. Maushmi Bharatwaj for her talk.
First of all, I'd like to thank the organizer, Professor Das and Professor Prakash, and for nice introduction from Dr. Sinjini and Dr. Manoj for having me here in this conference. So I'm going to talk about three of the work which just done under the ICMR task force project of the smokeless tobacco product related work.
which is a oral microbiome. As being in the ICMR Institute, we work in the three major cancers: cervical, oral, and breast cancer. And here, though I worked extensively in cervical cancer, but here I'm going to talk about the oral cancer, especially in the smokeless tobacco product usage is booming in India, and that is one of the reasons in ICMR made the thrust area for research aspect of that. Okay, so basically I am not going in detail because almost end of the day everybody got exhausted, and I'm not going in detail about the epidemiology of oral cancer. It's already aware of that. This is WHO data in 2020, where you can see, among the Asian countries, India is one of the topmost in the curve. So, as you know, there is a several risk factor for the oral cancer. Few is already modifiable, and other is non-modifiable. Modifiable risk factor among that is usage of smokeless tobacco product is one of the big issues in India now. So, if we go to the molecular chemical mechanism, where you know that smokeless tobacco product, the nicotine is a compound for addiction. Okay, so these are the chemical mechanism where smokeless tobacco product through carcinogen and ultimately it through different metabolic activation for the DNA adapt formation persistence, leading to the mutation of lot of RAS mutation and lot of tumor suppressor gene mutation. So that is a chemical mechanism for usage of smokeless tobacco product. Now I am coming to the next point, where carcinogen formation during this tobacco product cultivation or production, there is a no TSNA formation, right? TSNA is the key molecule for the cancer causing. But during the curation, nitrosation or nitrite formation and adapt formation with the alkylation and all this, ultimately these TSNA, the carcinogenic compound form. And here one point is that in addition to this chemical process, there is a lot of bacteria also, nitrate resistant bacteria also play a major role. So my talk is basically focusing on that microbacterium, where both bacterial and fungal microorganism is present. Okay. So next slide will be regarding this matter only. So for that, what we did it, we procured a very common smokeless tobacco product which are very common usage in North India, including NCR, and there is several brands are there. Okay, so K1 to K8 Khaini product means there is a lot of different brands. So we collected almost 36 of them, and we did extensive analysis, both chemical and molecular level. So this is basically the summarization of what the process we did it, procuring the SLT product. Physical characterization, we did it both pH and moisture, and chemical analysis, alkaloid, nicotine, TSNS and mycotoxin through GCFID, LCMSs, and the microbiome analysis, where the 16S rRNA sequencing both bacterial and fungal, and with the Illumina MySeq platform. And besides that, the molecular mechanism also will try to work in that is under process, very preliminary work in the oral cancer cell line, and there is some bioinformatic analysis to find it out whether we can find any kind of inhibitor for TSNA compound. Okay, so rest of the talk based on this result only. And one thing is that we did both SLT products which are already selling, but here I am going to talk about the microbiome where the patient sample where they are the user. Okay, so these patient sample we collected, that is the oral swab. I already mentioned that from our institutional clinic. Okay, as we know, the oral microbiota associated with the oral and systematic lot of disease, but in addition to the oral cancer and other oral diseases, it is also linked to the obesity, diabetes, liver diseases, pancreatic cancer and colon cancer also. So this is a study plan where the oral swab we collected DNA extraction, library preparation, sequencing analysis, diversity and functional prediction of the microbiome. So these are the 4 groups. Control, that is the no smoker, smokeless tobacco user, SLT, that means they are taking the SLT product but they are not having any kind of oral lesion, and another group that is a SLT user having the oral lesions, and another group which is a SLT user, not only they take the SLT, they also take the alcohol also. So when we did the bacterial and fungal analysis through the MySeq platform, that Illumina MySeq platform, and here you can see lot of bacteria is get prevalent. In a smokeless tobacco user having oral pre-malignant lesion, which is already written in the right, that is a Prevotella very low, and all these bacteria are there. So this is in compared to the control group. Besides the bacterial, there is a fungal analysis, fungal microorganism is also analyzed, and then here is the Pichia and Mortellia generi is also very high in the tobacco user having the oral pre-malignant lesion. So then the bunch of different statistical analysis is done. I am not going in detail to explain it now because of shortage of time and end of the day. Okay, if anyone is interested after that, please feel free to discuss with me or Dr. Sajid, basically Dr. Sajid, a postdoctoral student. His work basically I am presenting here, and he is a master of that. So that way we also found the bacterial alpha diversity analysis, and there is also we found that there is a smokeless tobacco user having the oral pre-malignant lesion. Similarly, we also did the alpha diversity analysis and among the fungal infection about the 4 groups. So when did the beta diversity analysis among the group, that is the alpha is a intragroup and that is a intergroup. Okay, bacterial analysis. Here is also you can see the bacterial beta diversity was significantly distinct for the smokeless tobacco user having the pre-malignant lesion. Similarly, for fungal group, fungal beta diversity were significantly different for the smokeless tobacco users having oral pre-malignant lesions. So similarly, when we analyzed the microbial biomarker for smokeless tobacco user, we see that there is a certain bacteria are very distinctly present in the oral malignant in case of smokeless tobacco user. Similarly, there is also distinct fungal group is present, like a Pichia fungus is very prominent in the SLT user having the oral pre-malignant lesions. So then he did, few of this interrelation of the oral microbial community, and then did this kind of networking, and their bacteriogeny involved in the oral pathology showed a significant correlation with each other. Similarly, the genus Fuchsia showed the highest number of positive correlation with the other genus. Similarly, when he did the association study of this smokeless tobacco user with the oral microbiome with the host disease, oral bacterial population of smokeless tobacco user can contribute to colorectal cancer and head and neck cancer, because it is already known, it is too obvious that oral microbe is associated with head and neck cancer or oral cancer. But from this kind of analysis, we found that colorectal cancer also having people, colorectal cancer is also very susceptible for the user. So this is kind of a novel finding out of this kind of data analysis. Similarly, functional potential of the microbiome associated with the smokeless tobacco user, we found lot of nitrogen metabolism genes and amino sugar and all so on for that, and pathotroph and saprotroph, that is for the fungal, and also was significantly abundant in smokeless tobacco user. So therefore, if we summarize that, we can see that is the people using all the smokeless tobacco products which are easily available in the local market, and from there so many introduction of the microorganism in the SLT user. Ultimately, that mechanism, that is a DNA damage and in the cellular level, that interfere for ultimate creation of the oral carcinogenesis. So that in the future direction, we can see the composition of SLT mediated alteration of the oral microbiome is studied by several work. Still it is in infancy. Okay. Still more work required to identify the impact of the different types of and duration intake frequency for the SLT products of the oral microbiome. And what function these pathogen microbes performing is still not fully understood, and future studies are required for exact molecular mechanism involved in the oral carcinogenesis. And oral microbiome transfer therapy, also known as the oral microbiota transplantation, is a potential treatment where the healthy oral bacteria from the donor can be transferred to a patient having the unhealthy oral microbiome. So these are the future direction whether only very few publications are there. Okay, so that can be a good lead for that. And what we did it, that is like, I can tell you in a very good way without any data. If anyone purchase any kind of SLT pouch, you cannot see any kind of analysis. Nothing is there. So in India, no products are getting really analyzed. So this kind of work is really needed, because as being in the ICMR, we also having that National Tobacco Testing Laboratory, where I am the scientist in charge for that. Because based on these data, they also want to do the Indian guidelines. Okay, because our analyzed product of the each pouches should be there, which is not there. Okay, and whatever data I showed, we come out with a series of publication for the Sajid is a lead for that, and so these are the publication we come out the study what I showed you, and to some extent bioinformatics data we generated to find it out whether we can come out any kind of analog or inhibitor for that. So I am not going in detail of that. This kind of work we also published recently, that we can see that tomatidine as a putative inhibitor for the CIP1A enzyme, because as you know, P50 metabolic cytochrome P50 is a alkaloid inhibitor based on this identification. So all these bioinformatics work, I am not going in detail right now, and this is a group worked on that, majorly done by Dr. Sajid and Sonal, and bioinformatics was by Dr. Ved, and technical assistance is done by the Lata Joshi, and scientific officer is Dr. Pragya Sharma, mainly the clinical sample she collected from our local clinic. And this project is funded by the ICMR Task Force project, and for the other part of the work is by the DHR. And thank you.
A nice talk. So, any questions from the audience?
Thank you so much, ma'am. Whenever I listen, you always feel like yeah, बहुत अच्छे से हमें समझा दिया. Ma'am, one question I have, like which kind of bacteria is abundantly present in oral microbiome, like whether it is gram positive or gram negative?
Yeah, actually we did. Look, that is a very fundamental question, positive or negative.
Yes, ma'am.
Because because this kind of information in Indian context is not available at all. So what we did, we just did no matter positive or negative, because we just picked it up all. Because we did the NGS platform, it's a very minor amount. Because if you do the, gram positive, gram negative, that is a classical way to do that. If your amount, even it is a gram positive or gram negative, very small quantity is present, you cannot pick it up. So that is a highly sensitive way we picked it up. What are present?
Yes.
Whether positive or negative, because you cannot tell that is a negative or positive right now, because both are there. Okay. First, because India perspective of this SLT product, you won't find this kind of molecular information at all. That is the reason from a single project we could come out 9 publications. Okay. So that is the reason. That is a very classical work. Few is a positive coming out in the plate. Some dissertation student is doing it, but we use the NGS platform to pick it up, no matter which amount it is, and present, then it will going to show up. Okay, even for the fungal. Otherwise, you have to do separately plating for fungal, positive, negative, and all this kind of things. You understand that?
Yes, ma'am. Thank you so much, ma'am.
Okay.
Any other question from the audience? Okay, so.
Question. Do you think using antibiotic first to eliminate the microbiome and then transplantation is a good approach, or no need of antibiotics and just do the transplantation of good bacteria?
Yeah, of course, because here is also we got lot of antibiotic resistance gene also we got it. Okay, from the bioinformatic analysis, whatever picked it up. Of course, antibiotic using and then transplant, of course, it's not a good idea. But in the same time, probably in a few cases that also needed, because to inhibit, few of these, harming bacteria not to get transplanted.
Yeah,
like
that was my
there. Yeah. So actually, this area still is lot of infancy. Speculation of future projection is a lot. Hope, but still 1 or 2, publication is there in a systematic way for the transplantation. So that way, of course, to inhibit the antibiotic will be good, mainly for the few cases, and then transplant the good bacteria basically.
Okay, thank you. Any other question? Thank you.
Pardon me.
Thank you so much, ma'am, for your insightful talk. Now, I would like to call upon stage Dr. Manoj, Dr. Shinjini. You also want to join your ma'am, and Dr. Shilpi also, to come upon stage and felicitate Dr. Maushmi Bhardwaj, ma'am. Please, Dr. Purnima, you can also join them.
Yes. I was thinking that even my PhD is from IIT.
Thank you so much. Now, I would like to call upon stage Dr. Archana ma'am to give a small token of appreciation to our session chairs who have conducted the session marvelously. Please, Dr. Manoj Garg and Dr. Shinjini Dasgupta. At least they also need a big round of applause from you guys. I know everyone is exhausted, but we will take few more minutes. Thank you, everyone. Dear esteemed guests, distinguished speakers, and fellow delegates, on behalf of the organizing committee, I would like to extend my heartfelt gratitude to each and every one of you for making the sessions so resounding and successful. I would like to thank our speakers, panelists. Thank you for sharing your expertise and insights, which have enriched our understanding of cancer research, diagnosis, and treatment, specifically utilizing immunotherapy. Your contributions have been invaluable. I would like to thank our session chairs also in different sessions who have taken that pain and conducted these sessions very successfully and remarkably. To the organizing committee, volunteers, and each one of us for their hard work and efforts. As we are concluding the last session of today, now I would like to give you a brief information regarding next your whereabouts. So you guys can move to I2 moot court, the same place, and there will be a small Indian traditional culture.
So we are going to have a cultural event from 6:30, followed by the dinner at Wall Street.
So thank you all. So you guys can move towards I2 moot court.